এই দেখ না, একটা আওয়াজ হচ্ছে কেন রে? হুম। 1 নম্বর আর 2 নম্বর, ওইটা আমাদের এখানকার আওয়াজগুলো নিচ্ছে কিনা বুঝতে পারছি না, কারণ আমি তো এখানে সাউন্ড অফ করে রেখেছি। একবার একটু একবার একটু এতে কিছু মিটে কিছু বলে দেখ তো।
Good morning, sir. Good morning, Professor Mondal.
Good morning, Professor Mondal.
Said you are not audible. Your mic is off.
Good morning.
Good morning.
Good morning, sir.
Good morning, sir. Good morning, sir.
Morning.
Morning.
Good morning, sir.
Good morning. ইনসোর্সে জয়েন করেছেন?
করেছেন?
তাহলে শুরু করে দাও, একদম টাইমে সব আছে তো।
হ্যাঁ, অবশ্যই।
Principal, sir, should we start now?
Yeah.
So, good morning and welcome everyone. Yes, sir. So we are starting, Professor Mondal.
Okay, please.
So, good morning and welcome everyone. Once Carl Linnaeus, in his book Linnaeus' Philosophia Botanica, quoted that in natural science, the principle of truth ought to be confirmed by observation. And from this very minute, we will start our journey together to seek this truth through various aspects of research in the field of botany. Welcome everyone, and thank you once again for joining us for today's webinar. My name is Dr. Sandeepan Ray, and I am the Assistant Professor at Department of Botany, Durgapur Government College, under Kaji Nazrul University. Along with me is Mr. Anish Bhattacharya, Assistant Professor at Department of Botany, Durgapur Government College. We will be your host for this 2 days webinar.
Today, we all gathered here to witness how plant science are evolving at this critical juncture on 24th and 25th June 2022. Through 2 days national webinar on botany through modern perspectives, algae to angiosperm, organized by Department of Botany in collaboration with Internal Quality Assurance Cell of Durgapur Government College. We have participants from all over India, like from Andhra Pradesh, Gujarat, Maharashtra, Jammu and Kashmir, Odisha, Tamil Nadu, Telangana, Uttarakhand, West Bengal, and Rajasthan. We cannot miss the person whose continuous support and valuable advices shows us proper directions for successfully doing this webinar. Their presence is enough to motivate a huge mass of botany lovers. Let me welcome and thank our members of National Advisory Committee, Professor Sudhendu Mondol, Department of Botany, Vishwa Bharati, Professor Surinder P. Hullar, Emeritus Professor of Department of Botany, Punjab University, Dr. Ashwini Kumar Srivastava, ex-scientist G, Birbal Sahni Institute of Paleosciences, Professor Alok Bhattacharya, Department of Botany, the University of Burdwan, Professor Chandan Sengupta, Department of Botany, University of Kalyani, Dr. Probir Kumar Das, ex-member secretary, West Bengal Higher Education Council, Professor Probir Kumar Saha, consultant scientist, Division of Plant Biology, Bose Institute, Dr. Bharati Nandi, ex-associate professor, Department of Botany, University of Kalyani. It is my pleasure that I got the opportunity to welcome them. Thank you so much. So now let me go through the schedule for today. This webinar has been divided into 4 main sections. And to start with, we will have an inaugural session where our esteemed keynote speaker, Professor Sudhendu Mondol, UGC Professor of Botany, Department of Botany, Vishwa Bharati, will share his thoughts, followed by plenary session where 2 eloquent speakers, Dr. Yogesh Mishra, Department of Botany, Institute of Science, Banaras Hindu University, and Professor Krishnendu Acharya, Department of Botany, University of Calcutta, will deliver their thoughts. After that, we will have a lunch break for 30 minutes, followed by poster presentation session 1.
Our intent is to spend most of the time to nurture the nature through this 2-day scientific gathering in virtual mode, while leading academic scientists, researchers, and students will exchange and share their experience and research on various avenues of botany, covering diverse group of plant kingdom, starting from algae to angiosperm.
So now without further delay, we will turn the time over and I want to request our Honorable Chief Patron, Dr. Devnath Palit, Principal, Durgapur Government College to deliver the welcome address. Sir, please.
Good morning to you all. Thank you, Dr. Sandeepan Ray and Sri Anis Bhattacharya. Very nice introduction of the today's national webinar on botany through modern perspectives, algae to angiosperm. On behalf of the Durgapur Government College family, I like to welcome you all in this today's national webinar. So, my colleagues, Dr. Abhijit Mondal, Coordinator, Internal Quality Assurance Cell, Dr. Swapon Ghosh, Secretary Teachers Council, then the Joint Convener, Dr. Ashok Bhattacharya and Dr. Prashanto Saha, Joint Organizing Secretary, Mrs. Moutushi Sen and Dr. Sandeepan Ray, Joint Coordinator of this seminar, Sri Anis Bhattacharya and Sri Dibyendu Medda, and my colleagues, distinguished participants from all over the country, and my deep respect and profound respect to all the, as mentioned by Dr. Anis Bhattacharya, so all the National Advisory Committee members. So I think it is the galaxy of botanists today. Though I am the principal, but my basic subject is botany. So I feel it's my pride privilege to be with such a gathering, august gathering of galaxies I mentioned earlier. So without delay, I would like to express my deep sense of gratitude and profound respect to Professor Sudhendu Mondal, who is deliver his keynote address within few minutes. He is basically the UGC Professor of Botany. He is my teacher. Sir, my deep regards and pranam to you. My deep sense of gratitude and regards to all the other resource person like Dr. Jogesh Mishra, Department of Botany, Institute of Science, Banaras Hindu University, Professor Kishendu Acharya. Basically, when I was in Darjeeling, he's my dada, mentor, friend, everything. So Kishendu, my welcome to our college. Then Dr. Sandeep Kumar Behera, Senior Scientist, NBRI Lucknow, Dr. Ashalata D. Rosario, Department of Botany, Nursing Datta College. My teacher, Professor Shobhun Mukherjee, retired Professor, Department of Botany, University of Kolkata. Distinguished participants from all over the country, and the participants, ladies and gentlemen. So without wasting time, I just request you, please, this is the platform as mentioned by Sandeep Konde and Onis. So where the distinguished and renowned professor, those who are working in the field of botany from algae to angiosperm, despite their busy schedule, they will spare their extensive knowledge and experience with us. So my request, please, you will be enjoy the 2 days in very befitting manner, and I think this is the one of the most interesting webinar they have chosen the topic, and please interact with our resource person, and they will highlight the different perspectives of the botany as mentioned by the title of the seminar. And if you have any questions, please write in the chat box, actually mentioned by the host later on. Anyway, and I think they have mentioned one thing, that e-poster session is there. So many abstracts we have gathered, and I can say only one thing, that for the last couple of months I have seen the radical changes in the teaching, learning, evaluation and research perspective in the Department of Botany under the able leadership of Dr. Ashok Bhattacharya head. And I think in the near future they will organize some national level seminar in the physical mode, and international seminar obviously in the physical mode. I expect from them. So without delay, I would like to conclude my inaugural speech by saying my deep respect and profound respect to all the resource person and everybody. So please enjoy. I can say not listen. Please enjoy this session, 2 days session. Thank you very much. Once again, thank you to you.
Thank you so much, sir, as your speech have enlightened us about various aspects of life and development. And thank you for your continuous support. Now, I want to request our Honorable Vice Patron, Dr. Abhijit Mondal, Coordinator of IQAC, Durgapur Government College, to kindly address the gathering. Sir, please.
Thank you, Anish. I think I'm audible to all of you.
Sir, you are.
Yeah. It
Yes,
gives
sir.
me immense. Okay. It gives me immense pleasure to present the inaugural speech in this 2 days national level webinar on Botany through Modern Perspective, Algae to Angiosperm. I would like to express my gratitude to our respected Principal Sir, Dr. Devnath Palit, and our Teachers Council Secretary, Dr. Shapan Kumar Ghosh. I am pleased to welcome everyone here in this webinar on behalf of the Durgapur Government College family. It is at the same time an honor for me to become a part of this webinar. A very good morning to all of you. We feel honored to have with us eminent resource persons from all over India. I want to convey my deepest sense of gratitude to them for accepting our invitation and giving their valuable lecture in this webinar. I feel privileged to convey my thanks and regards, especially to the Department of Botany for organizing this wonderful webinar in collaboration with Internal Quality Assurance Cell, Durgapur Government College. I would also like to convey my gratitude to HOD and the whole team, conveners, organizing secretaries, organizing committee members, who all are committed to this program to make it a success. And more this type of webinar or seminar in physical mode will be expected from the Department of Botany. I am thoroughly pleased to address the participants who have participated from all over the India. I welcome all the participants in this webinar. I will request all the participants to make use this platform to gather knowledge to enrich themselves. So today, we certainly have a wonderful session and interactions between the participants and the resource persons ahead. So without consuming any time further, I should stop myself here. To conclude, I wish all the best for the success of this webinar. Thank you.
Thank you so much, sir. Your presence and wise word will help to magnify the trust for a better world in the best possible way. Now, I humbly invite Dr. Shapan Kumar Ghosh, Teacher Counseling Secretary of Durgapur Government College, to greet the gathering. Sir, please.
Thank you very much. Am I audible?
Yes, you're audible.
Okay. So, good morning to all those who are present in the present webinar. Our respected Principal Sir, Dr. Devnath Palit, our IQC Coordinator, Dr. Abhijit Mondol, the organizing committee, a head of the Department of Botany, and all the faculty members of Durgapur Government College, and our honorable speakers, Professor Mondol and others, those who are going to deliver their lectures in the two-day national seminar. So it's my colossal pleasure to be asked for to say something in this wonderful webinar organized by the Department of Botany, and they are doing regularly in a regular interval in the Department of Botany, Durgapur Government College. So, as mentioned by Sandeepan, that we all know that conference or webinar or seminar, whatever we call it, actually it's a meeting point, and it provides an opportunity to share the knowledge in the concerned subjects and fields among scientists, botanists, teachers, young researchers, and students working in every nook and corner of the world. And the main aim of the webinar is to bring together and highlight the recent discoveries and trends in various fields of science and biological sciences. And it is also encourage the collaborations to augment the interdisciplinary research work. And the goal of the meeting is a cross-fertilization of ideas from different fields of science, especially biological sciences, for this particular webinar. And we all know that though it is a virtual webinar in online platform, but we miss the mingle during the lunch break or during the tea break or dinner when physical seminars are organized in the department. So we miss that, but still we are not losing everything, because pandemic had it has taught us that how to tackle the problem if the entire world is going to be seized in this pandemic situation, but we are not actually stopped or by in anything. But there is there is a wish, there is a way at all. So in spite of the above, there is also a social impact here because we know that Durgapur Government College is one of the leading colleges in the industrial region of Durgapur, Raniganj, Asansol, etc. with its undergraduate and postgraduate program. So this meeting will be helpful to motivate the young researchers and young students to carry out their research works. And also we know that actually it encourages the students to write dissertation papers or some seminar papers. So it will help the young students to encourage so that they can think about what is going on in the present situation. So it's a I think it is a very fruitful to organize such webinar in this platform. So I would like to not stretch this discourse because already 50 minutes have gone. So the teachers and so I'd like to the webinar. So thank you very much for giving me the opportunity. Thank you. It was a great success of the webinar.
Thank you so much, sir, for your wish. And thank you for your valuable advices and continuous motivation. Thank you, sir.
Just a little housekeeping before we get started. If you have any questions during the presentation, please type them into the question box and we will bring them up after the presentation and we will have more time for question at the end. And I would request everyone to keep your microphone in a off mode during the presentation. So today, we are very fortunate that we have a great keynote speaker with us for you. Actually, in a true sense, he needs no introduction. He is an institute by himself. Please welcome Professor Sudhendu Mondol. Securing first-class position in BSc Honours and MSc in Botany from Bishwabharati, Professor Mondol received his PhD degree in Science from University of Calcutta and has been awarded Commonwealth Academic Staff Fellowship at UK. He has above 37 years of experience in PG level teaching, 42 years of research experience, 23 years of administrative experience. Professor Mondol is a recipient of numerous prestigious awards like ISCA Young Scientist Award, Millennium Scientist Medal in 2009 by the Honorable Prime Minister of India, Dr. Manmohan Singh, in a view of his significant and lifetime contribution to the development of science and technology in the area of plant science. He also received Professor S. Siddhartha Memorial Award, Paul Johannes Bruce Gold Medal, and many more. He has received Professor E.P. Odum Gold Medal from the International Association for Ecological Communication. Professor Mondol has successfully supervised 35 PhD students in the field of plant biodiversity, systematics, palynology, aerobiology and allergy, detection of aeroallergens and biochemical analysis of allergic pollens, ethnobotany, pollination biology, and environmental botany. Professor Mondol visited a number of research institutes and universities in countries like Germany, USA, Bangladesh, UK, Thailand, Malaysia, Japan, Russia, etc. in connection with his research work and as a visiting scientist. He has completed 23 research projects financed by CSIR, UGC, DOEN, DST, DBT, and Vishwabharati. Professor Mondol has also, to his credit, more than 275 research papers published in various national and international journals and 20 books, including one Tago and Flower by CSIR publication. He is the former director of National Library, Kolkata, Government of India. He also served as Pro Vice Chancellor Academic in Sikkim Skill University. He was also the president of Indian Aerobiological Society Delhi and also worked as an working president of Natural Disaster Management Cell Vishwabharati and many more. During his work period at the National Library, Professor Mondal received VVIPs like ex-Honorable President of India Dr. APJ Abdul Kalam, ex-Honorable Prime Minister of India Dr. Manmohan Singh, ex-Honorable Speaker Sri Somnath Chatterjee, ex-Honorable Governor of East Bengal Sri Virenjay Shah and Sri Gopal Krishna Gandhi, ex-Honorable Chief Minister of West Bengal Sri Buddhadev Bhattacharya, and many other state ministers and senior government officers. All of them praised about the activities and the functions of the National Library. Apart from all these achievements, his beloved students, admirer, his peers, known him as a great storyteller. And now he is going to give us some actionable idea about our own legacy of Indian system of medicine. Welcome, sir, and the stage is yours, sir.
So thank you very much. Shall I start?
Yes, sir. Yes, sir.
Start.
Sure, sure.
Hello. Hello. Am I audible to you?
Yes, sir. Yes, sir. You're audible.
Just a second. Just a minute. Can you see my slide?
Yes, sir. We can say it properly.
So good morning to you all.
So there is an echo coming from your end.
Hello. Hello.
Yes, sir.
Okay. Just a minute, please. Hello.
Yes,
Hello?
sir, you are audible. Yes, sir, you are audible.
Okay. Is the slide visible?
Yes, sir, absolutely. It's clear.
Hello?
Yes, it's clear.
Okay.
It's
Thank
clearly
you. Thank you. So I would like to thank you very much for giving me the chance to meet you academically. Actually, to tell you frankly, Durgapur is very known to me, and I was associated with this college for last few years. And presently, one of my students, Dr. Ashok Bhattacharya, is now heading the Department of Botany. And also, personally, I know one of the very good researcher of your college, Dr. Devnath Palit. I know him personally for his research work. He is carrying out research. Current a teacher in a government college having transferable job from this college to that college. It's very difficult to carry out research projects and to guide the students. But still, Dr. Debnath has taken the trouble for his own inquisitiveness and his initiative. He is still carrying out research work, and we are all very happy for his academic endeavor. I thank you, Dr. Debnath, for your academic interest and to highlight your college to the befitting community in a befitting manner. Thank you very much. Now, in today's scenario, as you have your national webinar on botany through modern perspectives, algae to angiosperm. I tell you that the topic is very interesting because whenever we study botany, we have to start from the algae or angiosperms. Cryptogams and the phanerogams. These are the two groups from where we can function our physiological or biochemical or any other functions. All are materials starting from algae to angiosperms. So the theme you have chosen is very right in today's scenario or even in the 21st century. So I thank you all very much. And I have decided that, the plants are most important. Without the plants, there is no possibility of the survival of the human being. That's why I thought that I will make some genesis of the old, the way the plants have come to us and our India's scientific heritage. That's why I have given the title of my talk, India's Scientific Heritage with Reference to Medicinal Plants. As you know, for the first time in the history of science, Plato is the person that he laid the foundations of the philosophy and the science. He's a great mathematician, and his student Aristotle, he developed integrated scientific concept that is based on the observations. Without observing, he could not say anything else. And now we found our father of botany, that is the Theophrastus, the Greek naturalist. For the first time, he had the inquiry about the plants and their causes, other different activities. That's why he is known as the father of the botany. Now, as you know, today in the 21st century, or in the late part of the 20th century, we are all dependent on www. But for India, www is important academically. Sir William Jones, William Roxburgh is a botanist, and William Carey, the first professor. And William Jones, for the first time, established the first academic institution in India, that is the Asiatic Society. He was a Pusne judge during the British India period in the Calcutta High Court. But in the Indian scenario, if you see the genesis, he from India first time named the plants in Sanskrit language and published in 1792, 93 and 95. He published 4 research papers in the Asiatic researches only related to plants, being another field and scholar, historian. But for the first time from India or from Bengal, specifically from Bengal, West Bengal, specifically from Nadia district, because Krishnanagar and the Nadia are the Pandits, Sanskrit Pandits who are there. So he had an interaction with the Pandits and he collected information and he named the plants with Sanskrit. I will show you the slide. That's why I give him the is the pioneer, not only a botanist or the researcher, academician who worked in India with the plant system. And William Roxburgh, you all know, is the leading botanist. He studied in the Coromandel coast, then to Bengal. And William Carey is the first professor and the printing press, everything. And now I come to 2 persons, Paul Johannes Brull and Shankar Purushottam Agarkar. Paul Johannes Brull, basically a German scientist, he came to India during the British India period to teach natural sciences. He was a BSc graduate. He served in Rajshahi and other parts of Bangladesh, now present Bangladesh. But he was inducted by the then Vice Chancellor of the University of Calcutta, Sir Ashutosh Mukherjee, and initially he appointed Paul Brull as the controller of examinations, then registrar, and in 1913 he given him the responsibility to establish the biology department, giving the chance of the head of the department of botany. That was the first botany position of the teacher in India. That was in 1914. Paul Johannes Brull was the first professor of botany in the University of Calcutta, appointed by Sir Ashutosh Mukherjee, the then Vice Chancellor. Ashutosh Mukherjee was a magnificent. the dynamic person, because he inducted Sir C.V. Raman, he appointed Satyen Bose, Meghnad Shah after completion of their MSc, and also Acharya Prafulla Chandra Ray, he appointed him in the chemistry department, because these are the stalwart people's vision of Ashutosh Mukherjee to improve the science faculty in the Calcutta University. After Paul Johannes Brühl, we could find the Shankar Purushottam Agarkar. Agarkar from Maharashtra, Pune, he came to Calcutta Museum to study the botanical objects, having MSc degree, and it came to the knowledge of the Ashutosh Mukherjee. Then Ashutosh inducted him and appointed him to join the Department of Botany, Calcutta University, to work under Professor Paul Johannes Brühl, as it is a botanical science. Then you will be very happy to know in the today's scenario, if you join in a job, if you don't become the permanent, you can't get the leave. Ashutosh sanctioned leave to Agarkar after 6 months of his appointment to go to Germany to study further research. And Agarkar went to Germany and worked in the field of plant taxonomy under Engler and Prantl, and returned to India and ultimately headed the department after Paul Johannes Brühl. That's why I thought that you should know the botanical concept in your country. And this is the Sir William Jones. This is William Roxburgh. This is William Carey, who was the first professor in the first college in India, Fort William College, which was established in 1800, and he came from Sirampur and joined there as a professor. And plants are important. They are the man's prime companions in this universe, because without plants we cannot survive. That is, being the source of oxygen, food and energy, shelter and clothing, drugs and beverages, and also in today's scenario, the aesthetic environment. As you know, we, the human beings, we learn to identify, describe, name and classify different types of foods, clothes, books, games, vehicles, religions or professions, and any other objects that they come across or that influence their life. The process begins and ends with life, when a baby is born and ultimately when there is the death. All of it starting, when a baby is born, you go to greet with a bunch of flowers. When there is the death, you go to greet again with a bunch of flowers. So the plants are essential in everyday life for the human beings. And the life involves a continuous expense of energy. If you survive in the world, you have to have the energy. And the study of life processes evidently presupposes a thorough understanding of the structures of the organisms, the environment in which it lives, its hereditary makeup. Structures and functions are so intimately related that it is impossible to separate one from other. Like if you see the structure of a chloroplast, function is photosynthesis. If you see the structure of a mitochondria, functions are the respiration. So that's why each and every structure is related to the functions of the either in the plant system or even in the structures of the human body. Now, as a botanist, the characterizing, naming, classifying and identifying all plants, whether actually or potentially useful to man, are professional duties of the botanist, especially to taxonomists or systematists. We have to know it as a botanist, whether the plants are useful or in otherwise it is important. And why we give the scientific name? Because scientific name is the symbol of communication to which the information is attached. If you go to the other parts of the world, you have to tell the scientific name. If I say neem or if I say arm, nobody will understand. So that's why it is the symbol of communication in which the information is attached. And what is the role of the taxonomist? If you have a descriptive language for precise and useful characterizations of the organisms. When you see a plant or when you see an animal, you have to give a precise and colorful characterization of that particular organism. And finally, to devise a simple scheme for identification. How to identify them? And also to work out a system of classification to show the relationship. How we are all related, a botanist or a geologist or a physicist. In a community, we are all related in a society. Likewise, in the plant system or even animal system, we have to find out a system of classification to show their relationship. And you know, the systematics is a dynamic science. The duties of the taxonomists are never ending. It never ends. The flow of data about the plant material and the plant's necessities. They are reordering the information, revising schemes, revising schemes, revising schemes, and also are important re-evaluating the systems of classification and perceiving new relationships for a better understanding of the plants. As long as the plant world exists, there will be more to learn about the plants, plant products and plant taxa, both for practical and theoretical purposes. Because plant products are important for all of us in our everyday life. For the first time, the Rudolf Jacob Camerarius, for the first time on 25th August 1694, he published his own work, that is careful and painstaking experience that then said that the stamen and pistil must work together to produce ripe seeds, and these two are the sexual organs of the plants. In a plant, in a flower, sepals, petals, stamens, carpels. So the stamens and the carpels, they work together so that we can get the fruits and the seeds. Ultimately, the plant eat. Who told it? That is Rudolf Jacob Camerarius for the first time in 1694. And these two are the sexual organs. And now, who is known as the father of pollination biology? Christian Conrad Sprengel. Christian Conrad Sprengel, he said that the secret nature revealed in the structure and fertilization of the flower. He published in 1793, almost after 100 years. Like Rudolf Jacob in 1694 and Sprengel in 1793. Almost after his observations were with the nectar protection and the function of the corolla as an organ of advertisement and the difference between day and night flowers, mutual adaptation of flower and its pollination. That's why they are attracting the insects for pollination. That was his observation, that the nature revealed in the structures. That's why he is known as the father of pollination biology. Now I will just briefly, I will tell some of the leading taxonomists. We forget always, we ignore them, but they are the pioneer works on which we are standing today. Andrea Cisalpino, on which name Cisalpinia, the plant. He for the first time, the Italian physician and a botanist, he for the first time, first scientific classification of plants in 1583. De Plantis Labri, 1500 plants he named. And he was the first person who developed herbarium. And in the world, for the first time, herbarium was established in Italy. That is Padua Botanical Garden in 1545. That was the first herbarium in the world. That's why Andrea Cisalpino is famous, because he developed first time the idea of herbarium. The genus of Cisalpinia was named by him by Plumeria. And then John Ray, another scientist, he's a British, he established the classification Historia Plantera. But he for the first time coined the word species. Whenever we call genera, genus and the species, but he for the first time coined the word species. Now Tournefort, he classified he for the first time the concept, making the definition of the concept of the genus, botanist, the French botanist. So this is and he's for the first time, John Ray's concept, separation of the phanerogams and the cryptogams, algae to angiosperms. So cryptogams are there, phanerogams are there. In phanerogams is division, flowering plants with a monocot and the dicot. That was the Joseph Pitt and Tournefort's discovery, and they are the pioneer botanists in our field. Now we come to the Carl von Linné, the Carolus Linnaeus, the Latin name. He's a Swedish botanist, physician, zoologist. He laid the foundations of the modern scheme binomial nomenclature. He is known as the father of modern taxonomy and is also considered one of the father of modern ecology. Carolus Linnaeus, Sweden, Stockholm. I had a chance to visit Sweden, Stockholm also. His book, System and Activity in 1735, where he has been classes, orders, genera and species particularly. This is his most important book. And also finally, the global, that is the important species plantarum in 1753, and also the general plantarum 1791. So these are the books important botanically. And this is his personal garden at Uppsala. You can have a good idea of the picture. Following this idea, the Tagore-san, Rothindranath went to Japan and he made certain gardens in our Uttara and Tagore's garden in the same pattern like Linnaeus's garden at Uppsala, and it has a similarity with some of the Japanese garden in Tokyo. And these are the books, and the Linnaeus collected and named world plants 7,700 and animals 4,400, giving binomial nomenclature, generic name and the specific name. Now we come to the post-Linnaean period, that is Michael Adamson, with the numerical taxonomy of the phenotypes with the Scottish botanist in 1673. He proposed that numerical taxonomy. Then we have the classification Bentham and Hooker, the natural system of classification, named 200 families with the 97,200 species, 7,500 genera they explained from the Royal Botany Gardens. These are the scientists. Now we got to the first time major phylogenetic classification, Engler and Pruntle, then Hutchinson from again Royal Botany Gardens Kew, families of flowering plants, 1973, and finally we get the top botanist, Armen Takhtajan, the Russian, Soviet, Armenian botanist. He, in the 20th century, is the most important figure in the plant evolution and systematics. His classification is still important. And you know, he could not survive one more year, otherwise he could have been century years. 1910 he was born, and he had his death in 2009. His other interests, apart from taxonomy interest, morphology of flowering plants, paleobotany, and the flora he has been. And I had a chance to meet him either in Tokyo International Botanical Congress and also in the Royal Botany Garden Kew. And finally I had a chance to visit his laboratory in the Russia, that is his own place in his laboratory also. That is, he was there. He major contributions of Takhtajan in Potom in 1967, systematic in Moscow, then Takhtajan's and conquest floristic regions of the world, and finally during his death year 2009, flowering plants, that is the most significant in the plant system. Conquest also, as you know it, I'm just showing you some of them. Then the Thorne, another recipient of Asa Gray Award from California, from USA in 1920. He was born, and he also given the significant phylogenetical concept. And nowadays, with the advancement of science, so many molecular data is coming, so we call it the molecular phylogenetics. Advances in molecular biology have yielded profound impact in taxonomy. As molecular data, especially genomics data, become available. The term molecular phylogenetics is the study of evolutionary relationships among the organisms using DNA and RNA, and on the basis of which a tree can be constructed using DNA sequences and also with the relationships of the different organisms. And lately we have the Angiosperm phylogeny group. You all know that APG, nowadays we consider with APG. APG1 in 1998, and APG2 2003, APG3 2009, and APG4 2016. And you know, recently their flora of the world flora, there are so many scientists attached with the world flora, and we have 4 Indians attached, one Bengali S. Banerjee and A.K. Pandey, and 2 others, Saminathan, 4 persons associated with the world flora, and we feel proud from India that 4 botanists are associated in the publications of the world flora. It came very recently. So this is, you know, still today there is no precise figure about the number of species. As you know, there are so many scientists, they are giving different data. Now I will go to the next. And you know, the first Indian botanic garden, it was established in 1787. Why the botanic garden was established? The main objective of the Britishers were there. Garden was established to take a stock of the disseminating such articles which are prove beneficial to the inhabitants, the plants which are important to the inhabitants. That's why Robert Kidd was the first person, or the superintendent or the founder of the Royal Botany Garden, Howrah, which is known as the Indian Botany Garden. You know, in the Kidd's name, in Calcutta, you know that the Kidirpur is known as in the name of Kidd, because Kidd Sahib used to travel by boat from the botanical garden to other part, that is the Kidirpur, by boat. That's why on his name, Kidirpur. Kidd Sahib, Lieutenant Colonel Robert Kidd, was the first scientist who established the botanical garden. Now we come to William Roxburgh. He succeeded Colonel Keir as the next superintendent of the Royal Botany Garden, Shibpur. He toured India and Nepal extensively on explorations of the plant and returned to England in 1850. But he surveyed initially at the Coromandel coast, I said the South India, and the plants of the coast of Coromandel in 3 folio volumes, 1795, 1798 and 1819, containing 300 color plates with description and names in Latin, Telugu and English. You see a person coming from Britain to India, collecting plants, giving their drawings, everything, description, and in 3 different languages, the tenacity and the devotion, Latin, Telugu and English. So all botanical names, English names and the Telugu names, as he was associated with the South India. And finally, he published Hortus bengalensis from Bengal in 1814 with 3,500 plants, and also published Flora Indica in 3 volumes during his time in 1820, 1824 and 1832 with 3 volumes with 2,533 color illustration. You know, some of the top scientists, the botanists, you see at this stage, William Roxburgh made color illustration of 2,533. You see Rabindranath has written so many things, the late life from 70 to 80, 65 age to 70. He has drawn 2,500 color drawings. And that was his successful. These are the persons who were stalwart in their own field and the devotion. It can be compared with the Roxburgh and Rabindranath. Roxburgh himself that drawn the plants, his own handwriting, that are still preserved in the Indian Botany Garden, Shibpur. If you visit, you can see it. And if you visit the Uttarayan Garden, the Tagore's Museum, you find the Tagore's hand drawings. 2,000, almost 2,500 colored drawings, which he drawn from the age of after 60, 65 to his death 80. So these are the persons to be remembered like that. Now, as I have said, Sir William Jones, the first person who came to India as a judge, you know, he came to India on 23rd September 1783 as a Pusne judge and the Supreme Court of the Judiciary at the Fort William in Bengal. And you know, he thought that we have to establish an institute, academic institution. That's why he established the Asiatic Society of Bengal. You know, he came to India on 23rd September 1783 and he took a decision and only in 3 months established Asiatic Society on 15 January 1784. That was his target. You see the administration, how it functions. Coming to September, only October, November, December. Only in 3 months, he established that institute, Asiatic Society of Bengal, on 15 January 1784. That was the institute having considered, and he is having main interest in natural history, particularly botany. You know, he's a Pusne judge, Supreme Court, but he's a botany, natural botany. Why? Because that most lovely and fascinating branch of natural knowledge. For him, botany is the most lovely and fascinating branch of the natural knowledge. He published 4 papers on Indian plants, 2 of which appeared posthumously in the Society's journal Asiatic Researches. In 1795, that's I said that is the first person who published the botanical paper in before anyone else, before the botanist, William Roxburgh, earlier to that. That's why 2 research papers came out after his death, posthumously in 1795. And the most important is that the botanical observation on selected Indian plants published in the Asiatic Researches. The most extensive study on the subject and the result of a decade or more botanical exploration that the research in and around Krishnanagar. The observations describe 78 plants which are all selected by the author for their novelty, beauty, poetic fame, reputed use in medicine and supposed holiness. I have selected my topic, medicinal plants. You see, the William Jones selected that plants reputed use in medicine, medicinal uses, and which were mentioned in the sacred poems of the ancient Hindus. You know, some of the ancient Hindus, they have there in their poems some of the plants. You know, Gurudev Rabindranath Tagore has written so many poems, so many plants are selected by him with their name associated with that. So these are the pioneering workers who related with the plants. The botany has a history in everyday life. And you see that William Jones, he said that Sanskrit language is a wonderful structure, more perfect than the Greek, more copious than the Latin, and more exclusively refined than either. Because that is William Jones, Anglo-Welsh philologist and scholar of ancient India, as he established. Because he said, although Linnaeus and others, we give the Latin name, but he said, it is a wonderful, Sanskrit language is a wonderful structure, more perfect than the Greek, more copious than the Latin, and more exclusively refined than either. So he has given the high position of the Sanskrit language, of the Indian language, to the world community, not only as a scholar, but also a plant lover. That's why I thought I should tell it to you so that you have in your idea it's a botanical concept. Now we come to the Joseph Dalton Hooker. He was from Edinburgh, assistant professor, botanist. Finally, he became the director of the Botanical Garden and the Museum Kew from 1865 to 1885. He visited India and his works, Flora of British India, is still famous. 7 volumes in 25 years. He also made a monograph, Rhododendron. As a botanist, I can tell you, the most prestigious post globally is the director, Royal Botany Gardens, Kew, and highest and the coveted post and the highest salary as a botanist. I had a chance to visit Kew Botanical Garden many times. Even in 1992, when I visited, Dr. G.T. Prance, or Gillian T. Prance was the director. I got an appointment, I met him and discussed many things with the Indian things, and G.T. Prance was very helpful and he knows about the Indian plants. That's why the most prestigious post, Joseph Dalton Hooker availed, and he worked on the Indian plants, and he is also, we can call is the guru of Indian plants, because he has the 7-volume book. Still no one can beat him, Flora of British India, during that time. It took 25 years to publish for his whole life work. Being the botanist or the director, it was possible for him to publish this botanical exploration. We salute to him and Joseph Dalton Hooker. Next, there are different institutes, as I said, Asiatic Society associated with the plant, Royal Botany Garden associated with the plant, Fort William College, William Carey, Hindu College, Presidency College, that is also related to plant, I will come. Scottish Church College, they are also plant scientists, Medical College, plant scientists, and Calcutta Public and Imperial Library, the National Library, where the depositions are there, Calcutta University, Botanical Survey, Bose Institute, Jagadish Bose established. When the botany was not studied, Jagadish Bose worked on plants. That's why I will show later on all these. Jagadish Chandra Bose, the first Indian celebrated scientist, although he is a physicist or the father of biophysics, because he selected, he proved by experimentation that the plants or the animals much share in their common. And he demonstrated that plants are also sensitive to heat, cold, light, noise, and various other external stimuli. For the first time, Jagadish Chandra Bose told that the plants are also very sensitive. And he was born in 1858. You see, today we send our children to the English medium school, but his father, he got his elementary education from a vernacular school because his father thought that Bose should learn his own mother tongue, Bengali, before studying a foreign language like English. And initially, he studied in Kolkata, Bangladesh. Then finally, he attended Cambridge after studying physics at Calcutta University. He returned to India in 1884 after completing a BSc degree to Cambridge University. And, he delivered his first lecture on May 10 in the Royal Society in 1901, when there is no subject in botany in India. No one is teaching plant sciences, but he delivered a talk on what the plants have feeling like other living beings and humans. That was in 1901. That is his Royal Society in London, jam-packed with the famous scientist on May 10. First lecture in the Royal Society by Jagadish Chandra Bose. And you see, then others, he's communicated. His first paper came out in the Royal Society of London on the determination of indices, electric refraction published in the Pathology, and one paper has given him the award of the doctorate degree from London University. London conferred doctorate degree in 1896. And for the first time in 1897, Gurudev Rabindranath Tagore calls on Jagdish Chandra on his return home to offer him congratulations on his successful mission abroad. Gurudev Rabindranath Tagore was a fond. He was having a friend, two scientist friends. One is Gurudev Rabindranath Tagore and another one is Albert Einstein. Rabindranath never visited to any other scientist or any other top scientist of the world, but he met all over the world many different personalities. But personally he visited Jagdish Chandra Bose's house after his doctorate degree in 1897, and he visited Kaput, Germany, Berlin, to Einstein. He met with Einstein 3-4 times, and he went to Kaput to meet his residents in Germany. And I had a chance to meet that house of Albert Einstein in Kaput. I went to Germany in 2003 and also in 1985 for the first time. I visited that. So these are the associations of Tagore and Jagdish Chandra Bose, because Jagdish Chandra worked on plants when there was no botany department. And he presented a demonstration lecture, a special meeting at the Linnean Society of the London. What was that? Electric response in ordinary plants under mechanical stimulus. So that was in 1902. He also given a lecture at the Paris, lecture Society de Physique. Also, the British Association of Bird Food, electric response in animals, vegetable and metal. So taking the plants as a model, Jagdish Chandra delivered so many lectures in different parts of the world. And in Linnean Society, when he delivered in 1992, I was thrilled because I had a chance and I got an invitation to deliver a lecture at the Linnean Society in 1992 after 90 years. And I delivered a lecture in the Palynology specialist group of the Linnean Society in 1992. I was thinking that I did my research work at the Bose Institute, Calcutta University, and I salute Jagdish Chandra Bose and Rabindranath, from where I have my own academic bread and butter. So I remembered that day that when I was delivering the lecture at the Linnean Society, Jagdish Chandra Bose delivered the lecture there at the Linnean Society. And I feel proud to announce that that is the society where a botanist or the physicist can deliver a talk. And in 1908 to 9, he has many other papers, mechanical and electric response in plants, electrophysics and plant physiology. We don't know plant physiology during that time. Botanical Society of America at Baltimore, he delivered talk. Chicago Academy of Sciences, Torrey Botanical Club, University of Illinois at the lately. Illinois, Illinois is the university when Gurudev Rabindranath sent his son, Ratindranath Tagore, first, and the first Indian agriculturist from USA trained is Ratindranath Tagore. Rabindranath sent his son in, I think probably 1909 or 10, I could not remember exact, but the first graduate agriculturist from Illinois University, where Jagdish Chandra delivered talk in 1909. And in 1914 to 1915, Plant Autographs and the Revolution, the Royal Institution of London, again third Friday evening discourse. He visited many other plants. And also, lastly, you see that the lecture before the Royal Society of London, action of drugs on plants. Jagdish Chandra Bose became the fellow of the Royal Society in 1920. So you had to deliver the lectures in the Royal Society so many times to prove his academic merit. Otherwise, he could have not been related to fellow of the Royal Society. You see, these are his books from 1902, Response in Living and Non-Living, Plant Response as a Means of Physiological Investigation. So many, and the last in 1929, Growth and Tropic Movement of the Plants. 1923-94, Physiology of Photosynthesis, published by Jagdish Chandra Bose. So we don't know botany even during that time, but the book on plant physiology, the Physiology of Photosynthesis, was published. So these are by Jagdish Chandra Bose. And in 1918, he started the publishing the Transactions of the Bose Research Institute, where he published Life, Movement and Plants. And after 2 years, on 13 May 1920, he was elected a Fellow of the Royal Society of London. Demonstrated magnetic crystallograph at the Physical Laboratory of the University of College, London. Before that, C.J.K. Ramanujan in 1918 became the first scientist FRS, but before that, Another person, engineer Ardeshir Kursetji, in 1841 became the first FRS, Fellow of the Royal Society. After that, Sujika Ramanujan, mathematician, and as a scientist, biologist, biophysicist, Jagadish Chandra Bose, who worked with the plants and proved his merit, became the Fellow of the Royal Society in 13 May 1920. And he published his another research paper, Physiology of the Ascent of Sap, in 1923-94. And later, life, and he established Bose Institute in 1917. And I mentioned Rabindranath because he is the great sons become the Asian Nobel Prize in 1913, and the two countries' national anthems, Sonar Bangla, Bangladesh, and Janaganam of India. And I give him because he said the highest education is that which does not merely gives us information, but makes our life in harmony with all its existence. And Rabindranath first time said, sun lights the world and book lights the world, life. Why that book lights the world? And he said that India must use the gift of science to catch up with the advanced nations, failing which he will fail to reap the harvest of the present day. So if India has to co-op, he has to take the use of the gift of science of the West. And his friendship with Albert Einstein. And in India, as a botanist, I feel proud that no other branches, either physics or chemistry or mathematics, although Aryabhatta and others are there, but in the form of printed, the first book came out in India by Garcia de Orta on 10th April 1563. That is related to drug plants or the medicinal plants. Exclusively the plants, Coloquis, Simples, the Drogos, Ecology, Medicinas do India. The first printed book by Portuguese language, on 10th April. And this Garcia de Orta was the Portuguese pharmacologist, colonial administrator, explorer, and the botanist, and the first European to describe the famous double coconut, Cocos de Maldiva, double coconut, and give information on medicines used in India. He described bell fruit, Eagle Marmelos, Nirgundi, Vitis nigando, Neem, Melia directa, and Hersinger, Nyctanthus arbotristis, Conesi Batri, Hollerhana antidysentrica. These are all medicinal plants. And his work, Coloquis, 1563, is a landmark in the history of civilization. So that is the first botanical exploration in the form of medicinal plants or the drug plants. And the first research article published by a Bengali is Babu Ram Kamal Sen in 1829. A description of a common tree found in Bengal, that is bell or biloa, Eagle Marmelos. That was published in the Transactions of the Medical and the Physical Society of Calcutta. Babu Ram Kamal Sen, he was not a botanist, but he published the first article. And the second article, the research article by Radhanath Sikdar, that was a new method for drawing common tangent to two circles. It was published in Gleanings in Science, on the basis of which Radhanath Sikdar was appointed in the Trigonometrical Survey of India. He calculated the peak 15, the highest peak in the Himalaya, the Mount Everest, and Radhanath Sikdar, the pioneer scientist, and his calculation height of the peak turned out at 29,000 feet. And then Himalaya, you get a number of plants flowering in different alpine zones or temperate plants. Now, I come to another paper. As I said, the Paul Johannes Brühl is the first professor in the Calcutta University. And J.C. Sengupta was his PhD student in plant physiology, published from the Department of Botany in 1927 on the production of ripe foods by artificial pollination. So pollination biology has a history from 1927, Eissornia Piciosa. It was published by Paul Johannes Brühl and J.C. Sengupta. Seeing the tenacity and devotion of J.C. Sengupta, Paul Johannes Brühl sent Jyotish Chandra to Germany, and he did his doctorate from Germany. Coming back, he joined Presidency College, and finally he joined the Presidency College and worked there as a botanist, finally became the head of the department, and Jyotish Chandra Sengupta became finally the principal of Presidency College as a botanist, Jyotish Sengupta, during his time. And in today's scenario, in 21st century, we are all associated, or we are all seeing with the plant, very important plant, most important and a sophisticated plant, Arabidopsis thaliana. the first plant to have its genome sequenced, and is popular tool for understanding the molecular biology of many plant traits, including for development and light sensing. The plant belonging to the family Brassicaceae. You know, during earlier time with the genetics, we see the Mendel's pea plant, Pisum sativum, and now in 21st century, we are fond of Arabidopsis thaliana. What is this plant? You see the flower, beautiful flower. Now it belongs to the scientific plantae, Angiosperm, eudicots, rosids, brassicles, Brassicaceae, Arabidopsis thaliana, botanical name, and lineage described and hence, and synonyms is Arabis thaliana. How the name came, I will explain it. Arabidopsis thaliana, you know, a German scientist, a physician, German, Johannes Thal, he was a 1542 to 1583. He for the first time detected this plant in 1577 in Harz Mountains and named Pilosilla silicosa. First time from Germany. But later on, Linnaeus in 1753 renamed the plant Arabis thaliana on the basis of thallus, because Johannes Thal named the plant, but he didn't describe it and he didn't publish it. But he gave published it, and honor of Thal, he put his Arabis thaliana. But Gustav Henol erected the new genus Arabidopsis and placed the plant in that new genus. And the genus name Arabidopsis comes from Greek meaning and resembling Arabis of Linnaeus. That's why Arabidopsis thaliana came into being in today's. And you see the Arabidopsis book, what is that? The use of Arabidopsis as a model plant has provided opportunities for plant scientists from all over the world to network, collaborate, and share resources. Genomic research has significantly changed the landscape of plant biology in recent years. Almost all genome sequencing projects from Mars to Polar have resulted from extensive international collaborations. If one marks on a world map the affiliations of many authors involved in genome project publications, large areas of the globe will be highlighted. Probably the most far-reaching global effort in plant biology research is the dialogues and the collaborations established between the plant scientists in developing and developed regions, because India is still a developed nation. It's not a developing country. So it's all other, USA, Canada, UK, England, Russia, China, we are all collaborating in this group of research. Now you see the medicinal plants. How many species are there? Medicinal plants belonging to around 386 families, 200 to 2,200 genera. Asteraceae is much more common, 419, Euphorbiaceae 214, Lamiaceae 214, Fabaceae 214, and finally Apiaceae. These are the proportion of the medicinal plants selected from the Indian plants. I call it botany is the mother of science, which has given the birth to several different branches, each being pursued as a specialty. And now there has undergone tremendous change for the last 50 years and presently called the plant sciences to cover its wide scope. Now normally you go to the Oxford or Cambridge, never find the botany professor of plant sciences. So presently called the plant sciences. And you know, one of the pioneer scientists in India, Professor Birbal Sahni, who studied in Cambridge, Jagadish Chandra Bose, C.V. Raman, Birbal Sahni, Pandit Jawaharlal Nehru, who studied also botany, is the father of Indian Palaeobotany. He also worked and he made significant contribution. William Sonia, Sivadoyana, Glossopteris, you know, Pentoxylon, and he made on his name, Birbal Sahni, Birbal Sahnia, Dibbodarshan, one of a student, D.D. Pant of Allahabad University, Dibbodarshan Pant, who made one plant discovered, Birbal Sahnia, Dibbodarshan, the top one is that. So these are the stamps published by the government of India to celebrate or to honor the Indian botanists. And I name another Bengali botanist, that is Professor Amiya Kumar Ghosh, who worked himself in Calcutta as an eminent palynologist and paleobotanist, who worked from 1937 to 47 and 61 to 7 as a botanist in the Calcutta University. You know, he is known as the father of Indian palynology. Working individually in Calcutta Department of Botany and the Bose Institute, he himself published 3 research papers in Nature in 1947, 1952, and 1983. And you know, it is a very sad to mention, or it's very pity that although he worked himself, he submitted his thesis individually to the Calcutta University for doctorate degree. But there is a coalition with Birbal Sahni, with Amiya Kumar Ghosh as a botanist. You see it still during that period, there is a conflict. And the paper which he published in 1947, that occurrence of microflora salt in pseudomorph beds in Punjab, it published in the Transactions of the National Institute of Science by Nature, and that is, and also the 52, that has given the confusion with the Birbal Sahni. And this thesis was submitted to the Calcutta University, and examiner was Birbal Sahni. And you see, on this basis, with the personal click or hit, Birbal Sahni rejected his thesis, and Ghosh could not get his doctorate degree from the University of Calcutta. Although he himself has published 80 research papers, established the Department of Paleobotany in the Calcutta University, and one of the pioneer paleopalynologists. But you see the fate with the quarrel between the two top scientists. But you see, there is a God who always believes in that. That's why Birbal Sahni, although the first Indian botanist who was selected to become the president of the International Botanical Congress, still today no Indian could become the position of the got the highest honor International Botanical Congress, but that was to be held in 1950, but Birbal Sahni could not join. He had his death in 1949. That's the fate of Birbal Sahni. Although he was studied in Cambridge and a good friend of Jawaharlal Nehru, established his Institute of Paleobotany, later renamed as Birbal Sahni Institute of Paleobotany, but he could not hold that chair of the president. He could not have that occasion to chair as the president of the International Botanical Congress in 1950. That is the fate. One should not take revenge in otherwise. And today I can tell it because I have seen more than 125 PhD theses. I have never objected or never rejected any thesis in my life. I always encourage the young researchers to do research. There may be some true, false, or anything, some lacuna, but we should encourage the young workers to be upright to their future activities in the different ways. Next, I tell another person. Professor Pankishnu Parija from Odisha. He is regarded as the botanical father of Odisha. He from Ravenshaw College. You know, he was not having the BSc degree in botany, but he studied in Cambridge University, ranked the Frank Smart Scholarship, first class typos, Cambridge School of Botany. He worked under the supervision of Professor Blackman in the field of plant physiology and respiration of cherry leaves and apples, and published in his papers are published in the Proceedings of the Royal Society. But if you publish anything initially, the Royal Society, it is easier to become the fellow of the Royal Society. If you see that he was the professor of botany and the first vice chancellor of the Utkal University from 1943 to 48. Pro vice chancellor, Banaras Hindu University, pro chancellor, vice chancellor, again Utkal University. He became the president of the Indian Botanical Society and also botanical section of the Indian Science Congress Association. And finally, he became the general president of the Indian Science Congress in 1960. You know, after that, Arun Sharma became the general president as a botanist. Archana Sharma became the general president of the botanist, and Professor Parija became, and he got the Padma Bhushan award by the government of India, and he was also the fellow of the Royal Society. And you know, he was the secretary and the president of the Indian Student Majlis at Cambridge. Who were there? Pankishnu Parija was the secretary, finally became the president. Who were there? You see, during that time, Bal Gangadhar Tilak, Sarojini Naidu, Muhammad Ali Jinnah, C.V. Raman, Birbal Sahni, and many others. And also he became the president of the Utkal Sahitya Samaj. So these were there, and Pankishnu Parija was there as the secretary and finally became the president, the stalwart scientist in Cambridge. And who was his teachers in Kolkata, University of Calcutta? He studied BSc, Acharya Jagdish Chandra Bose physics, Acharya Pufullo Chandra was chemistry, Bankim Das Mukherjee and D.N. Murli mathematics. Studied mathematics honors and no provision for honors in botany during that time, because in 1913-14 botany was not there. Very close friends, Birbal Sahni and M.O.P. Iyengar as botanists. Birbal Sahni named a fossil, that is Enigmocarpon Parijai, after Parijai. That's what to has their friendship and others. Vivalsani named Enigmocarpon Parijai. And you know, as I said, medicinal plants. Plants are very useful and important for us. And you know, the importance and usefulness of trees, plants, creepers, climbers came out first time in the mind of Lord Buddha, an ancient saint about 2005 years back. When science was in paradox, the role of green coverage in maintaining oxygen and carbon dioxide equilibrium in the environment was unveiled. However, forests were protected since time immemorial for various cultural, religious, and social reasons. And Lord Buddha, he said, tree is a living symbol of boundless benevolence. It does not demand anything from anybody for its own nourishment. But during his lifetime, it goes on giving enormous quantities of useful material to the world. Not only this, but it goes on giving shade even to a woodcutter who has come to fell it. So trees. And Rigbed, what said? The plants, the water and the sky preserve us, and woods and the mountains with their trees and tresses. Emperor Ashok, who said the foremost disciple of Lord Buddha, is recognized as the pioneer conservationist by the International Union for the Conservation of Nature and Natural Resources, communicated the edicts of his master through inscription on rocks, polished stones, etc. He was the first initiator of roadside plantation. You see the highways, national highways, GT road, the roadside trees, was the first initiator was Emperor Ashok. Before that, no one could do it. That's it. And India is a rich indigenous medicinal tradition from the ancient period. Now I give it the ancient traditional Indian traditional medicine. It consists of different types of cure: Ayurveda, Unani, Siddha, Yoga, folk medicine. India's earliest indigenous medical systems, Ayurveda, that thrived on two classical medical compendiums. What are those? Charak Samhita and Sushruta Samhita. Ayurveda forms a holistic healing science dealing not only with treatment of some diseases but projecting the system as a complete way of life. Now, Unani organized and at Unan or Greece, and the Greek philosopher and the physician Hippocrates was its founder. This system of healing is in touch with India since 1st century AD, but regularly practiced by 13th and 14th century. Unani medicine. The ancient Indian literature, like the Vedas, Upanishads, Puranas, Aranyakas, and Jatakas, sing the glory of the nature of the Creator. And this lays down the percept of conduct of man to live virtuously. Many of the hymns of Rig Veda speak about the beneficial properties of plants, that is the Oshudi. It is pertinent to mention that the entire Indian literature, right from the Vedas, Puranas, epics, Sutra Shastra scriptures, the works of Kalidasa, even the Rabindranath Tagore, are teamed with praises of various plants and animals, like biodiversity in general and their contributions for the sustenance of the living world in particular. We see the herbal heritage of the India. There is a vast herbal heritage, natural resources. The history of medicine in India, that starts with the Rig Veda, where it has been mentioned 67 medicinal plants in Rig Veda. In Atharva Veda, 290 plants which are used as charms for curing the diseases. Then with the Lord Dhanvantari, they have given the originator, Lord Oshiri, the Ayurveda. And there are Ayurveda, the two Samhita, Charak and Sushruta. About 700 plant species have been mentioned here as medicinal plants. So these are the earliest Indian herbal heritage. Now if you see the tribal medicine, or the ethno medicine, or the folk medicine, 1600 species are newly identified as drug-yielding plants, and they are well known for their use. About 4,000 drug industries of various Indian systems of medicine like Ayurveda, Unani, Siddha and homeopathy. It is about 12% of the present Indian flora, about 80% of the raw materials of medicinal plants, which are collected mainly from 75 million hectares of forest coverage. Some of the forest species like Mamera, that is Coptis tita, Rauwolfia serpentina, Sausoria lapa are endangered and threatened. A good number of plant species and their products such as Plantago, Acacia, Asparagus, Sina, and Myrobalan are of having export value. We are exporting these. Even the medicinal plants, like different species of Lycopodium, Rauwolfia, Podophyllum, Dioscorea, Colchicum, Gloriosa, they are all overexploited for their medicinal value. Santalum album, Pterocarpus santalinus, overexploited for their scented and colored timber. Some of them are botanical curious, Nepenthes casciana, Helioingia himalaya. There are other beautiful plants, primula and orchids. A large number of plants are endangered due to agricultural pollution, Drosera, Uticulidia, and also the agricultural action, Nipah fruticans in Sundarbans. And view of the so many destructive forces working together on the biosphere, it will be our prime duty to save our floristic heritage at any cost. India, with her 55,000 plant species, with 550 total communities of 160 linguistic groups in varied geographic and climatic zones, with varied culture, rich traditional knowledge system, and wisdom possesses an ethnobotanical emporia. That's why it is very important for phytomedicine or ethnomedicine. Professor Robert Schultes of Harvard University opined that India, with her many living groups of people having diversified ethnic culture, history of rituals and performance, who are more or less isolated from modern world, are closely associated with their ambient vegetation, which is the emporia of ethnobotanical research. That is the findings of Schultes. And the plants are useful, may be grouped in the different groups, that is medicinal, timber, musical, house building, mats and baskets, religious purposes, oils and oilseeds, dyes, fodder plants, edible plants, and wild cultivated, and all are for other miscellaneous uses. You know, the tribal people had certain beliefs and taboos of medicinal plants. Plants with latex have medicinal value. Green plants before flowering are more effective as curative agents than the dried plants. Roots are considered more effective medicine than the aerial parts of the plants. Plants for the uses of medicinal purpose in various diseases should be collected in different times, as in the morning, midday, or in the afternoon. Medicine prepared in combination with the fruits becomes more effective. The barks in the medicinal uses are taken from that side of the plant on which the rising sun shines. The tribals have a belief that the violation of any taboo makes the medicine ineffective. So we have to strictly follow these of the tribals. Now, about 7,500 plants of medicinal value have been used. Besides, 3,900 are food plants and 525 species are still used by tribals for different purposes. But in this context, I would mention another narcotic yielding plant, that is cannabis or hemp. The cultivation is banned almost all countries. But you know, the oldest surviving piece of paper, more than 2,000 years old, discovered in China, was made from hemp. And today an indigenous method of producing paper from hemp is still practiced there in China, from hemp, 2,000 years old. Now this may turn an alternative, because now we cultivate eucalyptus. Eco-friendly, would be more eco-friendly than planting eucalyptus. Hemp is also a source of valuable medicine for treating glaucoma and alleviating the pain of certain cancer patients. And that's why, for this purpose, hemp is being cultivated in a protected area in Amsterdam, Netherlands, still being cultivated for medicinal purposes. And you know, the plants are used in different purposes. As I have mentioned, there are many medicinal systems. There are in India about 465,000 registered medical practitioners in Ayurveda, 240,000 conventional doctors as compared to 10 lakh doctors of modern systems, including homeopathic systems, and about 10,000 licensed pharmacies of Indian traditional systems. So economic value and sustainability development, we have to see the income of the rural women and also the tribal communities. Now, if you see the economic value and the importance of the biological diversity in relation to human health, one every 125 plant species studied at the Harvard Research Foundation in the USA has produced a major drug. Market value of these drugs in the US is at least $200 million per year. 118 out of top 150 prescription of drugs in USA, 74% on based on plants, 18% on fungi, 5% on bacteria, and 3% on vertebrates. And the top 10 prescription drugs in the USA, 9 are based on natural plant products. In 2015, sales of prescription drugs with active ingredients of plant origin were about 25,000 million. World Health Organization estimates that 80% of the world population relies on traditional and plant medicine. Losing a tree species a day means losing 3 to 4 potentially valuable drugs every year. Compounds from Ginkgo biloba, preserved by Chinese monks, used 80% Europeans older than 45 years for different purposes. So this is the economic value, and the herbal products are obtained for different drugs like atropine, quinine, morphine, reserpine, and digitoxin, which has great value to establish the important herbal medicine. Now I give you another example, a very serious problem. Although the drugs developed came out from 35,000 plants, creating bioactivity of 20,000 extracts, but a serious problem due to overexploitation of the medicinal plants. The marketed drug Taxol, Taxol is developed from Taxus babyphilia, which is used for the ovarian cancer, isolated from this plant. And 24 kg of Taxol is required per year in the United States alone. If you see 25 kg, so 1 gram of Taxol is produced from approximately 13 kg of dried bark, equivalent to about 1.5 mature trees. So if you have a mature tree, 1.5, then only you can get 13 kg of dried bark, then only you can get 1 gram of Taxol. But you see, in the United States itself need 24 kg. The trees can be grown for the production of active components, but there are certain difficulties. The brick trees are slow growing, and such harvesting cannot be sustained indefinitely. The other way to obtain extracts of the preparation for medicine from the plants is obtained through micropropagation and culture of plant cell tissue organs. But these are the alternative sources, definitely offer a number of benefits, including uniformity of productions, freedom, climatic factors, seasonal variations, diseases, and other restraints. So this is a problem for this particular plant for overexploitation. Now Himalaya, as I said, that that Himalaya is very rich in diversity. Eastern Himalaya harbors about 8,000 species, starting the 8 states, Arunachal Pradesh, Assam, Manipur, Meghalaya, Mizoram, Nagaland, Sikkim, and Tripura, consisting of 7.6% of geographical area. And Western Himalaya, 5,000 species of the flowering plants. It is the home of the precious genetic library maintained by the natural ecosystem. The number of genes from about 1,000 in bacteria and 10,000 in some fungi to 400,000 or more in many flowering plants and few animals. So that is the center for the Himalaya having reached biodiversity. Today, we are losing at least 1 higher plant species per day from the tropical plant forest. And if the present trend continues, about the 25% of the total higher plant species will be lost in the next decade, and another 25% by the end of the 21st century. So nowadays, the biotechnological procedures involving genetic engineering are also being developed for the production of the useful plant drugs to avoid overexploitation of bioresources. So now I say that the trees are the earth's endless effort to speak to the listening heaven, what Rabindranath Tagore said. Tagore also said, the winds of grace are always blowing, but it is you that must raise your snails. So you have to plant the trees. And that is the great banyan tree. It is not only one of the wonders of the plant kingdom, but it gives the shelter of hundreds of other living elements, including plants, animals, insects, and microbes, either as epiphytes, saprophytes, symbiotics, or parasites. And it is an ideal, most congenial for conservation biodiversity. And this is the sacred Bodhi tree of Ficus religiosa. This is the tree, Ficus religiosa, and this is the Stylotum nudum, a relic of the past, most important plant, botanically very significant in the phylogeny or the evolution. And one of the rare endemic species, the very high, narrow, and restricted distribution in Maharashtra, the Junar and the Purubandar hills of Pune district. It is well preserved in the Experimental Botanical Garden of the Botanical Survey of India. And Tornado, that's the Sophora toromiro. It is one of the commonly growing shrub or small tree of eastern island, became the extinct, and even a single live plant was found in the island. A hunt into various botanic gardens was made, and with concerted efforts, the species was multiplied from the specimen growing at the Bonn Botanical Garden, and the lost extinct species was again brought back to this world. So this is in the Botanical Garden of the Bonn, Germany. And even in the cold desert, you find, the Arctic Circle. In the snow-cladding Arctic Circle, there exists vegetation. During the warmer days, some of grasses at the very dwarf spiny shrubs providing food to the endemic reindeer. Cultivation of vegetables also in practice during summer months. Till now, the ecosystem and the ecological balance are not disturbed, and one can breathe happily and merrily. So this is the cold desert, and that is the Arctic Circle. This is the state tree of the West Bengal. Alstonia scholaris Chatim. That's one of the important Gurudev Rabindranath Tagore's father, Mahorshi Devendranath, meditated there with his own ideology. That is Jatra Vishwam Bhavataka Nidam, where the world makes a single nest. That is the Chatim Stala in Shantiniketa. And this is the state tree of West Bengal, Pictanthis arboreus Shivli. And this is the wonder drug plant. You know it, but still I want to just to recapitulate you, Rauwolfia serpentina, the wonder drug plant of the India. Gloriosa superba, the most important plant in the botanical curious, the significant. You see the anthers and the petals, fantastic in structure, beauty. And this is Paphiopedilum, beautiful petals, structure itself. And it is the insectivorous plant. Piphanthis casciana is the Meghalaya. It's growing luxuriantly on our roof garden, even it is the roof garden. This is the endangered species. And you see Rafflesia arnoldi, the largest flower, and Amorpha, largest inflorescence, and the Venus trap, insectivorous plants. These are botanical curious. Starting from BSc undergraduate, even from the school level, even to the postgraduate level, in the college interview level, sometimes the questions are asked whether you know it. And the Helsinki University Botanical Garden, this is the preserved in Finland, and in our botanical garden in Victoria Regia, the giant water lily, the Acharya Jagadish Indian Botanic Garden, Shivpur, is well preserved. The Nilambur nucifera, the most important plant, medicinal value. And the medicinal plants, some area, some of the medicinal plants I will show you have medicinal value, Stipania japonica, and also important medicinal plant. Mesozoaferia, the most important plant, a dicot plant having the parallel venation, Mesozoaferia. In monocot, we always find the parallel venation. This plant, you get the parallel venation. Curcuma amada, the most important medicinal plants. During the rainy season, you will get the flowers. This is the calamas. You find the flower of bamboo. And these are some of the, Ginkgo biloba, mirabilis, that is in Lucknow, preserved. And this is the living fossil, Ginkgo biloba. And this is the Satya. Those who are in Darjeeling, they know it in Darjeeling. And this is the invasive species, Isonia. And this is the Parthenia mystophorus, Lantana camera. These are not Indian native plants, but it has come and now is growing luxuriously. And this is the invasive species. Now it is our duty to conserve the plant wealth of this planet for our happiness and prosperity and the generations to come in future. That's why natural species are the library from which the genetic engineers can work, and biodiversity is the greatest treasure we have, and its diminishment is to be prevented at all costs. So these are always my students who have worked and helped me to visit around the world with their work. What I have told them, they have carried out satisfactorily with the devotion and intelligence, and they are now throughout the world well placed and doing their best in the botanical academic fraternity. I always praise them, and I congratulate all my PhD students who are present working still today. This is all my publication. This is the word I have got it with the works of my students. Visited abroad, these you know, and finally I got the Millennium Scientist by a botanist. To get an honor from the Prime Minister of India is more prestigious in the Science Congress in presence of so many FRS scientists. When MS Swaminathan was there, Govardhan Mehta was there, CNR Rao was there, even the Ashok Sen FRS, these are all there in presence of that to get a scientist. A small Indian fellow, a botanist, got the Millennium Scientist Medal. That gives me satisfaction. Starting from a village life, a young boy from a village, being the first graduate and postgraduate from a village of Birum district, studying at Vishwabharati, working at Bose Institute, or working in England as a Commonwealth Fellow for 2 years, visiting around the world, getting the most prestigious award from the Prime Minister, having a book published by CSIR, Trigger and Flowers. No West Bengal botanist, even Arun Sharma, Archana Sharma, or B.B. Vistas, no one having a CSIR publication. Still today, the botanist, I have it in my mind. And I was helped and backed for my life by non-Bengali friends and the global friends, not by any Bengali fellow who have helped me to climb up. You know, I tell you these words because it is with the Bengali fellow we never reciprocate our friends. I am the person, I always encourage the young talents to come up to help them in every respect. Be bold and to help your colleagues. This is my spirit to young botanists. So these are visits, awards, sorry. And with this, I thank you botanically giving me the chance and allow me to speak you the botanical scenario, not only in India, the global picture. Thank you very much for your patience hearing. Thank you.
Thank
Hello.
you so much, sir. Thank you so much. And what a wonderful journey we have just covered through the pages of history of botany in India and the world to the present scenario. And I must say, yes, our life begins and ends with the plant. And in terms of Gurudev, Mallika Chameli Beli, Kusumatulaha Balika, Gath Juthi, Gath Jati, Gath Bakula Malika. In every song, in every writing of Gurudev, there is botany. And personally, I guess this lecture is quite close to me because there is Gurudev, Bose, and Einstein in a single thread. And who doesn't know about the connection between Gurudev and Professor P.C. Mohalanavis and his discovery of sauropod and naming after Gurudev Barasaurus tagore, Barapasaurus tagore. And clearly pointed out by you, sir, that what we have with us in India as a natural resource and what can we do with it and what is the present scenario of the natural resource and the Ayurveda in India. Thank you so much, sir. And this session is open for interaction. If there is any questions by the participants and the guest, you can put forward.
Sir, with your permission, if you allow me, then I would like to ask something to Professor.
Of course, sir. Of course, sir.
Sir, I'm Dr. Yogesh Mishra from Department of Botany.
Yes, please.
Banaras Hindu University.
Oh,
And sir, it was wonderful to listen your talk. But since you have a very wonderful, successful and painful journey, because that was reflecting in your last sentence. So I would like to ask you 2-3 remarks because these are not the question, because since you are a very elderly person and you have gone through all the ups and downs of the academia, so that's why I thought you could be the right person to answer this question, and I was very impressed by your honesty. Sir, first thing is that if you look at the botany department, you have shown the many paper in Nature. But you know that it's very hard to find a single Nature from India after 1980s. It's very hard.
Yes.
And the second thing is that people working in the Department of Botany, they are very reluctant with the modern changes. Very reluctant. And even though we are having so many universities, so many BSI, but if you look at the global plant botanist age, in the molecular biology, in the classical taxonomy, Even though
Yeah.
we are such having historical resources, historical knowledge, we are not up to the mark.
Yes.
And one of the basic reasons which I fail to, which I understand, because sir, I have been visited a lot. I was in Max Planck Institute, Cologne, Germany for 2 years, and Umia Plant Science Center, Sweden for 5 years.
Very good.
So I have seen the world and seen the legendary people, because you were introducing the Arabidopsis thaliana, and
Yeah.
I was working with the Kornif. He was the one person who developed
Very
this
good.
as a model system. You might
Yes.
heard about it. He was
Yes.
the director of the genetics department of Max Planck. So
Yeah.
I was talking a lot with him and all this journey that how Arabidopsis was developed. So sir,
Yeah.
my question is that what we should do as a youngsters to change the mind, gentlemen, now it's time to come, equally respect that this is a natural biodiversity which you have, but please acquainted with the modern knowledge, otherwise you cannot sell this information to the modern platform. What we should do to deal with these reluctant minds?
To tell you frankly, I can tell you that
Yeah.
even today's scenario, the persons who are heading the chairs or who are in the top positions, they want everything that only I will get. I will
Yes.
not allow any of my colleagues to get this or that or to help him, because now even the dedication or the devotion is lacking. Even the youngers are sometimes become frustrated when they see that the seniors not helping. The attitude, how you develop yourself. If your senior colleague or if your director is not placing properly, even I can tell you the history. In Delhi University, Professor Mohan Ram was very much helpful to me. But even in Calcutta, when Arun Sharma was there, he was not helping me. He was not nominating me for any other award or anything else. That is the senior. Even the DD Pant, I said Dibbodarshan Pant, for the student of Birbal Sahni Institute. He helped me a lot because I did initial works in palynology. But it is very difficult to tell you the mentality of the person. You see, in Bengal, I will give you the example. Arun Sharma became the president of botanical section. He became the general president. He became the FNA. Archana Sharma became FNA. But they never did any of his students taking up or pulling them, although some of them did good work. They can be of that quality standard, but never helping that attitude. But in earlier, you see Acharya Jagadish Chandra Bose, Prafulla Rai, Prashant Chandra Mahalanobis, Meghnad Saha, they were all helpful. They are pulling their students to place them. Even the Prashant Chandra Mahalanobis sent some of his colleagues to abroad to have the study, to help them. That is the lacuna in today's scenario. Besides, nowadays in Indian scenario, the promotional system is very easy. If you do some this or that stray work, you can be promoted to the level of the professors. No one is very serious about the research work. But research is your own thing. If you don't commit from your own heart, then you cannot do it. You can do it casually. But there is a lacuna, I tell you. If you don't get the inspiration from your, I tell you, like your academic parents, father and mother has given you the birth, but you have to get benefit from your academic parents. That's why, otherwise it is very difficult to come up to this level. That's why we are not giving. As I said, that AK Ghosh published, even still today among the botanists, Shoyaram Bose, SR Bose, you find it, he is having 27 research papers in Nature. He worked on polyporocy in the fungal mycology. Shoyaram Bose or SR Bose. He is the only botanist from India. He is having 27 research papers in Nature, Science and other relevant publications. Even in the Prafulla Chandra Rai, he is the topmost chemist of India. He did his DSc from Edinburgh University, but he could not become the fellow of the Royal Society because I told to some of my chemist friends that he was not having a paper in the Royal Society journal up to the age of 60 or not having a paper in Nature up to the age of 60. What he has his paper in Nature after 60 to 69, Prafulla Rai had 3, 4, 5, 7 papers in Nature. So he could not become the FRS, but his students, Meghnad Saha, Satyen Bose, all Prashant Chandra Mahalanobis, all become fellow of the Royal Society. How you do it, that is the thing, and which journal you publish it, it matters. Even after that BP Pal, or even that no one become now in India. One of the greatest Indian is K.S. Bawa, Kamal Bawa, who is now in USA. He became FRS. He became the member in the USA National Academy of American Academy of Sciences, become the fellow. The one Indian now is there in the USA who become the fellow of the Royal Society for his work. Okay, so these are the, so who is helping? Bawa is helping so many Indian students. So how is the attitude matters for your survival and the identity? That much I can tell you, Professor Jogesh. I know some of your work. You are doing very good work. You have to keep it up to be at par with the other senior stalwart people of our India. Okay, thank you. Best wishes.
Thank you, sir.
So is it clear? Hello?
Yes, absolutely. Yes.
Thank you very much, sir.
Thank you, sir. So now we'll move to our next session, that is a plenary session 1. And I will request Mr. Anish Bhattacharya to continue with the session, please.
Thank you, Dr. Ray. Our next eminent, am I audible?
Yes.
Yes, you are audible. Yes.
Okay. So our next eminent speaker is Dr. Yogesh Mishra from Department of Botany, Banaras Hindu University. His specialization is in molecular biology, physiology, genomics, proteomics, and metabolomics of cyanobacteria and plants. Dr. Mishra pursued his Bachelor of Science degree in Botany from Uday Pratap Autonomous College under Purvanchal University with First Division, and Master of Science in Botany from Banaras Hindu University with First Division also. He also did his PhD in Botany from Banaras Hindu University, Varanasi. He pursued as prestigious postdoctoral researcher in several overseas institutions like Max Planck Institute for Plant Breeding Research, Department of Plant Development and Biology, Cologne, Germany, under the supervision of Professor Csaba Kons, then as a postdoctoral researcher in Umeå Plant Science Center, Department of Plant Physiology, Umeå University, Sweden, under the supervision of Professor Stefan Jansson and Professor Wolfgang P. Skroder, Deutsche Forschungsgemeinschaft, Germany. He formerly was the assistant professor at Department of Botany, Punjab University, Chandigarh, India, from December 2014 to 2015 November. And as I mentioned earlier, currently he is developing numerous future botanists at Department of Botany, Banaras Hindu University, Varanasi, as assistant professor from November 2015 onwards. In his career, he had numerous international visits in different countries like China, UK, France, Germany, Denmark, Finland, Switzerland, Sweden, Norway, and other European countries. He was awarded with Early Career Research Award by Science and Engineering Board, India in 2016. He was shortlisted among final candidates called for presentation by Indian National Science Academy at 2011. He has professional membership with International Plant Proteomics Organizations and International Society of Photosynthetic Research. So it's a kind of a short but highly effective motivational story for many students. And we all can already guess that what we are going to have within a few minutes. So without any further delay, I would like to request Dr. Yogesh Mishra to deliver his precious lecture to us. Over to you, sir.
Thank you, Ashish, for introducing me. And it's a great pleasure for me to share some of my research with this audience today. Because when I returned back to India, there was a big challenge for me that what should I start? So, I did my PhD in the cyanobacterial system and postdoc in Arabidopsis thaliana. And as professor in my previously, it was very well deliberated by how important model system it is. And I am very satisfied today to announce that when I joined the Department of Botany, India, Banaras Hindu University, this model system was not, nobody was working. And I started this model system from 7 years before. And I'm very happy to share that even yesterday one of my paper was accepted in BNMC genomics. So in the 7 year of effort, apart from the cyanobacteria, I established the two model system. One is the Arabidopsis thaliana here, and we start in the process of journey to developing another model system, that is the liverwort, Demorsera hirsuta. And some paper has come, and I believe that next year some new paper will come. So since many young people are joining with me, and if you look at a lot of good papers in the bryophytes, especially with the Physkometrella and this Markensia polymorpha is coming. So I believe the Indian biodiversity having the around 20,000 bryophyte species, we can do wonders. We can do a lot of good things. But today's talk is not based on either the Arabidopsis or the bryophyte. Today I'm going to tell you something different, because if you look at the, now I'm going to share my slides with you. So today I'm going to give a talk. So let me first start my presentation. So can you see my slides?
Yes, it is visible.
Okay. So, today I'm going to discuss something completely different, because if you look at the cadmium stress, many people working with higher plant, bacteria, yeast, cyanobacteria, they have been doing the cadmium toxicity research across the world. And if you look at the 60s, 70s, 80s time, a lot of people in the cyanobacteria working with the cadmium toxicity, either using the unicellular or the multicellular organism. But I was thinking that why we are talking too much about the impact of the stress. But if you look at the nature, in nature what happened, if you look at a pond system or freshwater system where these cyanobacteria are naturally growing, what happened that if there is a continuous inculcation of the toxic heavy metal come into that area, that they will accumulate, and when the rainy season will come, their concentration get diluted. It means organism is going to saying the two things. One, they are getting impact of the stress and they are going for the recovery. But if you look at the research, abiotic stress research across the world, there is too much focus on the impact of the stress. But we should also look into the scenario that these plants have a tremendous capacity to recover out of it. So I'm going to talk a very important thing, that how does Enabina species PCC deal with the acute cadmium toxicity at molecular scale, and what are the molecular mechanisms by which it gets recovered. Because we should not forget that these tiny organisms are they are present on the Earth's surface over 2.6 billion years longer time. So they must have a unique strategy, what they are, how they are dealing with that. So here, why it is important to study the acute cadmium toxicity and why it is important to understand the post-recovery strategies. As we know that cadmium is very important contaminants which affects the growth of the organism ranging from prokaryote to eukaryote. And another feature of this cadmium is that it has a bioaccumulative nature that is going to accumulate in the system. And it is very important to mention here, even at a very low concentration, it can induce the oxidative stress and affect the growth, metabolism, photosynthesis, and respiration in several organisms. A lot of studies are available in the literature. But if you look specifically the cyanobacterial research concerning the cadmium toxicity, you may see there is a range of concentration has been used. So after looking to literature, we found that the maximum cyanobacterial studies pertaining to cadmium toxicity has been investigated at the LC50 dosage, where the 50% growth was inhibited for that particular organism, and that concentration ranging from 2 micromolar to 15 micromolar. So When I look into this problem around 7 years before, then I thought this is the undermining of the cyanobacterial capability to deal with this stress. And if you look at the research related to cadmium toxicity in the higher plant, you will surprised to know that now people are trying to understand that how these plants get recovered from this heavy metal toxicity, where they have used the concentration from 50 micromolar to 1 millimolar in the various organisms. The reason behind taking is that even in the cyanobacterial system, in the aquatic system, that when the summer come, the cadmium concentration increase very high in these dishes and plants, and after that they get diluted in the rainy season. It means there is a chances that organisms are exposed to very high cadmium toxicity, and after that the dilution come and it goes for the recovery. Taking these things into the consideration, we try to mimic that situation in the lab and try to understand how this organism, Anabina model organism, is dealing with the acute cadmium toxicity. But the most important challenge for us, that what dose we will call as acute dose and how we are going to select. So what we have done in the beginning of experiment, that since it is very well known fact that cadmium has a genotoxic impact. And what is the genotoxicity? Genotoxicity is that they break the DNA in the organism. So the first aim was that to select a cadmium concentration which can induce the DNA break, and after that organism can survive. So when we put in the fresh media. So we started from 20 micromolar and reaches towards the 200 micromolar, and we found that the 100 micromolar cadmium after the 24 hour and 48 hour induce the different kind of cadmium toxicity, DNA break, and we found that the 100 micromolar cadmium exposure after 40 hours, we found half of the DNA get breaks. Here you can see clearly with this image. So we took this 100 micromolar cadmium exposure 48 hours as a acute cadmium toxicity dose for Anabina PCC710, and after that we did all our experiment. And please keep in mind, if I am taking say called ACDS, means I am talking about the acute cadmium toxicity, it means that 100 micromolar of cadmium exposure for 48 hours. So this was our experimental setup. What we basically done, that we took a culture, we let them grow to the grow them to the mid-log phase, and after that we give the 100 micromolar cadmium toxicity 48 hours. We centrifuge that culture and put into the fresh media and try to see how the organism is recovered. On the basis, and we found that after the 15 days, almost culture is attaining the unstressed one, so they are almost recovered. So we, on the basis of this growth curve, we took the three phases on which we focus for the next study, that early recovery, mid recovery, and late recovery, and we given the term ER, MR, and LR. These are, and the first one is acute cadmium, and the early recovery, mid recovery, and late recovery, and we perform rest of our experiments. And it is very important, and many people ask the question that if you given so much high concentration of cadmium, how much it is going to accumulate inside the cell, and what was the level of intracellular cadmium concentration. So we measured the intracellular cadmium concentration after the acute cadmium toxicity, and we noticed that 39% of cadmium, you can see here, it retained inside the cell. And after that in ER, it retained only 29, after MR 12%, and LR. And we also try to look at that if this high amount of cadmium toxicity, does it create the oxidative stress? Yes, you can see by measuring the acid-induced intracellular ROS using oxidant, using DCHF dye, which gives fluorescence after binding with the ROS. So here you can see in the early ACDS, if it is not showing the red color, light yellow fluorescence, it means there is a huge amount of ROS is that, and after the ER, LR, the amount of ROS get declined and argonium get recovered. So this experiment given us two important questions is that if there is a retention inside, what are the mechanism by which either it is chelate or it is going to throw cadmium outside the cell? And I will discuss in the end of my slide where we talk about the transporter and binding. In between, our interest was that how this cadmium toxicity affect the physiology, ultrastructure, and how the reprogramming of the molecular machinery takes place to cope up with this acute cadmium toxicity. The first and foremost important thing we measured that how does this entire journey of Enabina goes with the acute cadmium toxicity followed by the recovery at the level of morphology and the ultrastructure. If you see here, if it is unstressed, so dumbbell-shaped cell, well organized, so cell you can see here. But if you put the acute cadmium toxicity, the cells started shrinking. Here you can see, and sometimes there's a breakage of the filament also, and after that it recovered. But the important thing is that, and by using the transmission electron microscopy, here you can see that there is the deorganization, not the proper assembly of thylakoid membrane was noticed just after the acute cadmium toxicity. ER, there is a recovery started, and finally at the LR. So it suggests that cadmium is affecting the thylakoid membrane organization, and as from the earlier knowledge we know that it affects the photosynthesis, especially PS2. And apart from that, we also notice that it is also affecting the external architecture of the organism and creating the stand break, and it is going to recover. But the question arises that what are the molecular mechanisms lying behind this recovery? So we started because many physiological studies has also been done, but not on the acute cadmium toxicity. So we measured the chlorophyll fluorescence parameter of the Enabina subjecting to acute cadmium toxicity followed by the 3 recovery phases, and we measured that the ATP, NADPH, ATP, NADPH, and chlorophyll fluorescence parameter. And the fluorescence parameter basically tell you how the photosynthesis and what complexes of photosynthesis is get affected. So we measured the fluorescence parameter like FB over FM ratio, PS2 yield, QE and QL. And I'm not going to discuss all these details. Those who are very interested, please read our paper. We published a very nice story in the Journal of Hazardous Material 2021. It's a very high impact factor journal like 10.5. So you can read all the story. But in the crux, I would like to tell you that most of the PS2 related parameters get affected. So it is similar story which has been earlier reported that PS2 is the main target of acute cadmium toxicity, and PS1 showing much robustness in this cadmium toxicity. And one of the important thing is that if you look at the snapshots of events what are taking place inside the organism, so most important thing is that try to look into the proteome dynamics, because the most of the biological phenomena, metabolic changes are governed by the proteins. So in order to look into the proteome dynamics, we look into the cytosolic proteome of Enabina after exposing the acute cadmium toxicity and looking into the post-stress recovery stages like early recovery, mid-recovery, and late recovery. You can see the nice 2D gels in these 4 sets of condition. So we noticed that after the gel analysis, we found 88 protein spot showing the significant changes, and we given the terms that is called DAPS, differentially accumulative protein on the gel, and we try to look at that these differentiation are statistically significant or not. Here you can see these proteins are showing different trends. These 88 trends out of showing 76 showing the differential circulation, and some proteins showing up and down. And I will tell you about the proteins in the upcoming slide. The important thing I would like to bring your attention here on this PC analysis. So here you can see after the acute cadmium toxicity, the proteomic changes find in a one corner, and after that they following a trend. It means there's a continuous reprogramming of the proteome is followed, so that it help into the organism to get rid about the cadmium toxicity. Further, in the different PC analysis, we found that how these proteins are forming the cluster, and these proteinic changes get attestation by the statistical measurement, and we say that whatever changes we are showing is a differential accumulation of these 80 spots. So after that, we identified these 88 proteins by mass spectrometry. This is the list of the protein, which I'm not going to discuss. But the important thing is that to bring your attention here, that how to make easy to audience that what these 88 proteins are doing. So in order to make the easiness for the organism, we differentially classified this protein on the basis of functional category. These 88 proteins belongs to different functional category. Say, for example, photosynthetic and pigment and light dependent protein, and the Kelvin-Benson cycle related protein. You can see the respective percentage: protein biosynthesis, folding and degradation, purine and pyrimidine synthesis, carbohydrate, lipid metabolism, regulatory function, and hypothetical protein. So if you look at the category of the protein, these proteins are involved into the important metabolic pathway which helps the organism to survive. So here the protein basis, protein dynamics offering a possible phenomena what is going to affected by the accumulation of this protein pattern. Many times people may feel that these proteins are artifacts, are the real product of the gene. In order to make sure that these proteins are not the artifact, they are product of the gene, we selected certain proteins from each category and performed the semi-quantitative PCR analysis, and we found in the maximum cases they are following the trend, accumulation trend, as it is demonstrated by the two-dimensional electrophoresis, except for the few cases because cytochrome 550 where it not showing the same accumulation pattern. And you know, said sometimes this phenomena is happen that all the time your transcript accumulation is not showing the same accumulation pattern in the protein. There are many reasons for that. I'm not going to explain that. After that, since it is very important to mention here, in our SEM and TEM analysis, we noticed that the thylakoid deformity was noticed after the acute cadmium toxicity, and there was a recovery in the thylakoid also. So, and the photosynthetic parameters also get affected. So we try to ask the question that if there is a dimorphity and if there is a changes in the photosynthetic parameter, what are the protein complexes are responsible for that? In order to answer that question, we use the thylakoid membrane protein dynamic study by using the Blue Native page. For the students for which we are not aware about this technology, I would like to just introduce this technology. Blue Native is a native page electrophoresis technique. As you know, in the thylakoid membrane, your photosynthetic electron transport chain is lying in form of different complexes, PS1, PS2, cytochrome, and a lot of protein. And we simply isolate the thylakoid membrane, solubilize this thylakoid membrane with the detergent, and run on the native page, native gel from low percentage to high percentage. Here you can say the upper is the 4% and lower is the 18%. So this is a very tough technique because the isolation of thylakoid membrane is very important, their purity is very important, their solubilization is very important, and the last is the separation. So this is the technique which is very good if you want to see the dynamics of the photosynthetic complex. So we applied this technology in the cyanobacteria, where we look into the dynamics of the photosynthetic complex protein. And if you look at this PN page, you can see the PS2 is heavily affected by the acute cadmium toxicity, and they almost damaged, and the recovery you can see. And when we look at the second dimension, all these photosynthetic subunits you can see. So we also measured that how the photosynthetic complex dynamics are changes and how they recovered. After running that to a second dimension, we pick the proteins, and here you can see this is all the listed protein belonging to the different photosynthetic complexes. So here, say for example, there are protein for the photosystem 1, photosystem 1 reaction center subunit 2, uncharacterized protein PS2 chlorophyll A binding protein, ATP synthase, different unit alpha, beta units, delta. So we find all the proteins, D1 protein, D2 protein, cytochrome 6, cytochrome B6 in this thylakoid preparation. It means our purity of the thylakoid membrane was very good and isolation was very good. So further, we also characterizes that these proteins must be the real product of the gene. So we again verified them by the semi-quantitative PCR, and most of the multiple protein complexes, so in hand-to-hand tested with the real-time semi-quantitative PCR, that they are showing the similar accumulation pattern as on the BNSGS page. So as I said, after looking into that, okay, yes, this photosynthesis is governed by the dynamics in the photosynthetic complexes. We also look into the possibility that if the organism is retaining so much cadmium inside after the acute cadmium toxicity, and there is a slowdown export is possible, what happened with the two properties of the chelation as well as the export? In order to answer that query, since the transporter are very low abundant protein and it's very hard to get into the 2D gel, so we applied the alternative techniques to see their dynamics, to understand the molecular mechanism and the acute cadmium expression pattern. As you know, those who are aware about the metal toxicity, that metal first get chelated by the protein, so they are not get exposed for the organism to attack. So the two proteins are very important for that. One is the phytogelatin and second is metallothionein. So we measured the expression pattern of these two genes that is responsible for their synthesis, like phytogelatin synthase, which is responsible for the phytogelatin, and metallothionein gene, which is responsible for the synthesis of the metallothionein. And apart from that, we also measured the expression pattern of three exporter, like three component CZC, which involve into cadmium, zinc, and cobalt efflux, and CZCB, and which is ATPase type transporting. And these are the expression pattern of these three exporter in the acute cadmium, ER, MR, and LR during the post-stress recovery. And I would like to bring your attention here that once the acute cadmium toxicity was exposed to organism, you can see the expression of phytogelatin synthase was very high. It means organism want that these cadmium should be first chelated. And the early recovery, since you know that phytogelatin is not the direct product of the gene, however, the metallothionein is direct product of the gene. So metallothionein is showing the different expression pattern as the phytogelatin. So phytogelatin synthesis immediately get enhanced so that it can chelate. And the next strategy was to induce the metallothionein so that it can be chelated at the early phases, so that it should not hamper into the recovery of the organism. But the interesting point is that apart from these two chelating capabilities, the organism induced the exporter to exclude the cadmium outside the cell. Here you can see that after ACDS they induce, and they are very high expression into the mid-recovery, and yet recovery almost the expression was very low, similar in the other three cases. It means the organism first try to chelate and then try to go for the next level of chelatin by expressing the metallothionein, and in the same time they induce the exporter to export the cadmium so that the toxicity can be minimized. So, you know, if you have such a huge amount of data, it's very hard to make it convenience for the audience. So in order to make the convenience for the organism, we develop a hypothetical model where we try to bring all the information together, where we bring all the morphological, physiological, and the molecular data together, and we develop the model where we can explain that how the organism in the unstressed condition, what was the situation, when you put the acute cadmium toxicity, how it going to affect, and what happened in ER, MR, and late recovery. And that was appreciated a lot by the reviewer. And what is the take-home message, and what is the conclusion of our study? On the basis of these results, we found that NABNA respond to acute toxicity and recover during the PSR the following adaptive strategies. What they are alerting, they are altering its morphology and ultrastructure. They reduce the photosynthetic ability. They activate the PS1 dependent cyclic electron transport system, which we tested both by the physiology as well as the dynamics in the thylakoid membrane complexes, and stimulate the cadmium chelator and efflect the transporter in time dependent manner. They rapidly do the reprogramming of entire proteome, especially related to the cellular defense, carbon assimilation, regulatory protein, hypothetical protein. In view of the dynamics of all the parameters examined, we found that NABNA have many metal detoxification mechanism and functional in a synergistic way to cope up with the acute toxicity followed by the post-stress recovery. And what is eventually we can see from this type of study, we concluded that this study give a new dimension to the ecotoxicoproteomics that can be used for the selection of suitable marker, that is genes of protein, and these molecular candidate can be used as for the engineering the organism, which can be used for the better metal tolerance capability and can be applied biotechnologically for the metal removal for the contaminated area. And this talk will not be completed if I will not acknowledge the people who helped me a lot. So I'm really very thankful to my PhD student, Miss Akanksha Srivastava, and I would request audience in the afternoon, she is going to talk about a very wonderful story of the photosynthesis. That paper was published in Algae Research last year. So she will tell you about the other dimension of the cadmium toxicity. Apart from that, I am very thankful to Subhankar and Sandhya. They helped me a lot into the other experiment. I am very grateful to one of my collaborators from his working as a scientist in KTS Sweden for the mass spectrum analysis. I am very thankful to one of my colleagues, Professor Ram Sagar sir in the Department of Botany, who helped me a lot for the statistical analysis. And I would like to thank the Program Coordinator for Center of Advanced Study, School of Life Sciences, and the University of Potential Excellence for the financial support. And thank you very much. And I would be very happy to welcome the question related to this study. And please feel free to discuss and ask the question.
Thank you so much, sir, for enlightening us by such a beautiful lecture. Heavy metal toxicity is indeed a major problem nowadays. It not only harms us economically but also ecologically. And finding such groups of organisms against it is indeed a major step towards bioremediation. The more they come to us by such excellent findings like you, sir, the more we will surprise and the more we will be benefited. Really, nature never fails us to surprise. So now the session is open for interaction and we will take few questions from participants. So please
ask.
Please ask.
I will be very happy to answer.
Anyone from the participants, if you have any question, please ask. Feel free to ask. Actually, your lecture was so much compact that I think they have all their doubts in a clear way.
Okay, no issue, Ashish, and I'm very thankful to organizer for giving me this opportunity to speak, because I think this is my second lecture in any webinar, because I'm a bit reluctant person to give the online talks. So in the last 2 years, I was just engaged in papers and all these things. But I thought now it's time to come to participate and give lectures about our findings. But it would be great that if I can present in offline mode, so where you can connect audience in better way rather than online mode. So I hope that participant will. So I would request that if those who have any query, please go through that article which I have shown in my slide. That paper was published in Hazardous Material, and it is very long paper, 20 pages published article. So anybody who is working in the metal toxicity, he or she must be benefited out of that article.
Same here, sir. We will hope that in future we will get you in as offline mode.
Yeah.
Let's see. Let's hope for the best.
Yeah.
So I
So
guess.
thank you very much. If you allow me, then I may leave now.
Okay. Okay. You may, sir. Thank you.
Thank you.
Okay, now we will move to our next speaker, and I guess today undoubtedly the most memorable day in my life because the next speaker whom I am going to introduce now is my teacher since 2009, and I'm quite fortunate enough to work with him, and also fortunate to introduce my teacher, one of the leading mycologists of the world. Please welcome Professor Krishnendu Acharya from Department of Botany, University of Calcutta. Professor Acharya.
Am I audible? Am I audible,
Yes, sir.
Sandipan?
Yes, sir,
Okay.
you are
Thank you.
audible.
Thank you.
Professor Acharya pursued and obtained his MSc degree in botany from University of Calcutta with distinction in 1992. He then completed MTech in biotechnology from Durgapur University in first class. After that, he pursued his research work in the field of classical and molecular mycology, macrofungal diversity and medicinal prospects, innate immunity in plant and myconanotechnology, fungal biotechnology, plant pathology, etc. He has over 25 years of research experience with over 436 research articles, 7 books, and 3 patents in his name. The most remarkable feather in his cap is that he was entitled as top scientist in Stanford University list among world's 2% scientists in 2019. He is a fellow of Mycological Society of India and recipient of awards like Sir Edwin John Butler Memorial Award, Patel Memorial Award, Global Achievement Award in the field of Phylosphere Biology, and Professor K. Natarajan Memorial Award. Professor Acharya is an academic editor of PLOS ONE and associate editor of the Journal of Environmental and Sociobiology. He has also participated as a resource person for different national and international seminars and different refresher courses. He is also the reviewer of reputed journal published by Elsevier, Springer, Nature Publishing Group, Wiley, Royal Society of Chemistry, and more. To me, he is an outstanding human being, a guide, a teacher, and last but not the least, a great storyteller. So, dear participants, please buckle up your seatbelt as he is going to take us in a roller coaster journey on how we can change the scenario of our Indian organic farming through biotechnological approach. Over to you, sir. Please welcome.
Okay, thank you, Sandipan, for your nice introduction. But forget about all these things, what I have done and what is my publications, because all these things are ornament because it's required to get this platform where I can share my story in front of this August gathering, where I can share my story with the future budding scientists of the world. So these ornaments are basically for that. Today I'll talk something different. First of all, definitely I would like to thanks Dr. Devnath Palit, principal of the Durgapur Government College. DJC, I actually, he is a good friend in during my initial career at DJC. Again, that is also Darjeeling Government College. Now he is the principal of DJC, Durgapur Government College. And after a long time, we have actually a discussion regarding this seminar and all these things. Few days back, I talked to him. And many senior professors are here, Professor Sudhendu Mandal sir, Professor Pravisha sir. But what I feel that during the last talk, the participant comes down from 57 to 38. So I will try my level best to keep the participant be within this particular session. Uh, uh, and another important thing, two of my students right now is a faculty of the same that institute. It's basically an honor for me as a teacher. When students actually reach to the high level or reach to the better position, it's always a pride for a teacher. And also thanks to both of them, and I hope they have taken the baton from us, and they will complete the this lab to reach to their destinations, okay, future destinations. Okay. Also, I'm thankful to the organizer because they have selected the main focus or theme of this particular seminar, actually nurture the nature. So whatever I will speak today, basically to with this particular motion, that is the nurture the nature. And whatever the research things we are performing in my lab, that will be presented by my students in the second half of this session. Somanjana Khatwa is there, Sudeshna is there, Rituparna is there. They will talk about the hardcore science, what we are doing. But here I just like to share what I am doing with the people who do not have completed their school education system, or have not completed their school education system. So first of all, I would like to share my slides. Okay. Is it visible?
Yes, sir, it's visible.
Yes. So basically, my story of the journey actually is a organic farming system. It's a that is based on the organic farming system. And this organic farming system, we have tried to convert it the inorganic way of cultivation through our biotechnological intervention. Why? Because to empower the farmers. Okay. So that's basically the major objective of my topic, that a biotechnology-based organic farming system for empowerment of farmers. It's basically a journey from lab to land. Before entering into this thing, if we think a little bit 1960s and early of 1960s, we have introduced that inorganic way of cultivation because of the food security. For inorganic cultivation system, we so-called scientists, we discovered that different types of hybrid plants. Okay. And as you know, the hybrid plants needs higher energy, or because of their higher metabolic rate, they need SOS chemical fertilizer. Again, we so-called quote unquote scientists, we discover different types of chemical fertilizers. And as you know, after taking all these chemical fertilizer, the cell size increases, water content of the cells increases, and it's become a good platform for the pathogen to sit over it and to cope up from this pathogen. We again, so-called scientists, we discover toxic compounds. Okay. So as you know that pathogen can generate resistance against this toxic compound. So again we discover more toxic compound, and more toxic compound. And due to that, standing in the 21st century, what we are seeing, our lands are drying, our soils are dying. It's dying, literally. All the natural resources are contaminated. Environment pollution level hiked. Also, global warming increases. Also, we introduce many elite diseases within us. Angina pectoris, to diabetes mellitus, to cancer, to Alzheimer. Now, standing in this scenario, we are crying for go for organic, and go for organic. whom we are again asking to go for organic? The poorest among the poor, who are surviving from hands to mouth. The poorest farmers of the country have a small land. We are asking them, go for organic. Just think about these poor people, these thin people. They are actually feeding us every day. 150 crores people actually depends on these thin people. But they are not getting benefited from their farming system. So how many days they will go for farming? We have three important priority for survival. One is food for survival, clothes to wear, and house for shelter. Besides house and cloth, we can survive for our years. But without food, how many days we can survive? The people who actually feeding us every day, they don't have food for their survival. So this is the condition, this is the scenario. Those who are actually from the village area, they can understand it well. So, and we are now asking for go for organic and go for organic. We introduce so many actually farmers meet and all these things, and we ask them, you give carbon input in your soil, fantastic. If that farmers ask you, can that carbon input makes our land to become an organic one? You will say, no, it cannot be. Because we, during these last 60, 70 years, every day we applied chemical fertilizers and chemical pesticides and made our land sterile, because we are the killer. We kill all the microbes present in the soil, and we have destroyed their habitat. So now you have to introduce azotobacter in the soil. You have to introduce PSB in the soil. You have to introduce PGPR, and to tackle the pathogen, you have to give trichoderma, pseudomonas, BT, Beauveria, Pachyllomyces. So many things are required. If they ask you from where I will get this thing, yes, some company will produce this thing. They make it. They keep it in a packet and then say that they will sell it to you. Just think, microbes grown in the laboratory, preserved in the talcum or charcoal, then keeping it for 6 months. We do not know the birth certificate of this organism. And when you transferring that organism after 3, 4, 5, 6 months, do you think they will parrot? If I keep you for 5 days in a closed room, if I release you after that, can you see sun properly, light properly? How do you expect that they will reach to the land and fight for you and give you good food? And the cost? 300, 400 rupees per kilo. So this is the major objective, that how I have made a journey to make the farmers to become entrepreneur. Another objective of my today's talk is think outside the box. My dear friends, dear teachers, dear colleagues, and the PhD scholar and MSc, BSc students are there. So just try to think outside the box. Because during this, actually now we are gradually coming out from these pandemic times. You know, we loses our many friends, many relatives during this COVID time. Many people loses their job. Global economy goes down gradually all over around the globe. Standing in this scenario where after passing this graduation, master degree, PhD, if you ask us what I will do, it's very difficult to give you the answer. But if we can think outside the box to become an entrepreneur, why shouldn't we can create job? Those we are actually engaged in certain job, we are feeding our family, no problem. But when you create a job, you feed many people. During this pandemic time, I made more than 530 family to become entrepreneur. Now they are become sustainable, and I will tell you that story how they become an entrepreneur. So that is as I stated that my talk actually circles with nurture the nature. So entrepreneurship to promote organic farming is the fundamental objective of my talk. Before entering into the success story, I like to clarify what does it mean. What does it mean means how you convert your inorganic land into the organic one. So to convert the land into inorganic one, that has three strong hand. One is you have to think about the soil quality and fertility management, because soils are dying. So you have to revive the soil. So you have to change the soil quality, and you have to think about the fertility management of the soil. So that for that reason you need compost. Carbon is important, otherwise no other microbes will survive in that particular, because you have to create the habitat, okay, for that microbes. So my farmers are all very much familiar with the vermicompost, compost, biogas slurry, and here we have given certain technological intervention how you can improve this compost into a value added compost. So during in this compost mostly you will get N and P, but to enrich the compost with potash, that is K, you have to add either you can add the banana stem part, also you can add the wood ash. That wood ash contain large amount of potash, also it contain all the micronutrients required for this organic farming system. So amend your this thing with this material. Also amend is egg cell, so enrich your compost with calcium too. So that way you can convert your normal compost into an enriched compost. So this is the fundamental thing. They can do it by themselves only. Beside this, another thing is required for soil quality and fertility management. That is the use of biofertilizer and biopesticides. So biofertilizers means nitrogen fixing bacteria. They need phosphate solubilizing bacteria. They need plant growth promoting rhizobacteria, which actually supplements the soil with some enzymes and other hormones. And also they are able to fix the nitrogen from the air. They are able to solubilize phosphate, which we added in the soil for last 60 years, because 60% phosphate still bound to the soil. Only 40% utilized by the plants only. So they solubilize it, and they actually give this material to the plants for better growth and development. So that is required. Beside this, as we are working with plant, the crops mostly we are used, they are the hybrid plants. So for hybrid plants and other plants also need certain foliar nutrient, and in market there are lots of foliar nutrients are available. That is amino acid, humic acid, and this amino acid, humic acid, and vermi was this amino acid cost around 1600 rupees per liter. Please note, my dear friends, those who are actually studying masters, graduation, and PhD, please note this thing, because I'll give you I'll show you how my farmers are producing all the things in a cheapest cost. This amino acid nowadays available in the market, which cost around 1600 rupees. I have given a technology for the farmers. They are able to producing this amino acid at a cost of 40 rupees per liter. Write it, 1600 and 40 rupees. Now in the market, you will get these only two products in case of biofertilizer, that is Azotobacter and PSP, which cost around 300 rupees per kg, where no birth certificate is there. You do not know the CFU, how much CFU present there. You do not know the health condition of this organism, but you have to purchase this thing from the market at a cost of 300 to 400 rupees per kilo. Here I have given a technology for my farmer. They are producing for this in this biofertilizer section. They are able to produce by themselves only. Because if they ask you this question, sir, can I produce this thing in my premises? You will say yes, you can produce it in your premises. How much cost is needed? You know, you have to purchase 3-4 fermenter. That fermenter, one fermenter comes around 5 to 8 lakhs, the small one. Then you need 3-4, so you need a laboratory of 50 lakhs rupees, so you can able to produce this thing. Then that farmer will tell, sir, take my land, you just cultivate here, I'll be the happiest one. Okay, so this, and in the market you will get only two products. One is, please visit any retailer shop, you'll get more, there is a very little chance of getting this thing, because nobody bother about this product, because they don't believe that organic can be a solution for this thing. Everybody talking about the Sri Lanka factors, but I will show you the real story. Because if you add these azotobacter and these PSB, have no birth certificate, no quality, can it work? Can it be an organic one? And also you will get at a cost of only 300 rupees. But only these two cannot play a role for the changing of soil quality. You need different types of PGPR. So I have given technology for my farmer. They are able to produce azotobacter by themselves. They are not at all a scientist. Okay, they are not at all a scientist. Their maximum education level is class 8, class 9, class 10, like that. And most of them just know how to sign. Okay. So they are able to produce azotobacter by themselves. PSB, they are able to produce different types of PGPR, plant growth promoting bacteria, the Bacillus subtilis, Bacillus amyloidosis, Bacillus lichenoformis, Bacillus valigensis, and many more, Megatherium, and many more. They are able to produce by themselves, which is not available in the market. Okay. So if you think about the organic, you have to think holistically. Otherwise, don't go for organic, because don't suck the blood of these poorest among the poor. Because they are the real people who is actually converting their blood to sweat every day. Right now, we are sitting in an ambient condition. I am talking here, you are listening. But my Bhagavan, those who are feeding us right now in the field, doing something agricultural practice to feed us good food. Okay? So don't try to cheat this Bhagavan God, who's actually feeding us every day, every moment. So these things are required. Besides this, they need the amino acid, humic acid, and all these things. In case of pest management, okay, and all these products, what I have shown here, that is nitrogen fixing bacteria, phosphorus soluble bacteria, I have given a technology. My farmers are producing at a cost of 15 to 20 rupees per kg. Also write that thing, 15 to 20 rupees per kg, and with a birth certificate live, and they directly transfer to the field. And beside this, the pest management system is there. In case of pest management, one technology I have given to the farmer, that is the prevent the disease rather than cure at the end stage. So disease prevention. Then there are certain herbal formulation through which you can manage the disease to some extent, but you need a holistic approach for pest management. You need trichoderma, you need Beauveria, you need Pseudomonas, you need Verticillium, you need Bacillomyces. So disease prevention and disease protection techniques are required to manage the pest at the field level. And last but not the least, the marketing strategy. So lab to land, and the marketing strategy, land to directly consumer. No intermediates are there in my this agricultural practice system. So when they ask you this question, that so for production of this thing they need. So I have developed a technology for the farmer. Therefore, they need a fermenter which costs around 8 to 10 lakhs rupees. So I have developed a fermenter for my farmer at a cost of only 10,000 rupees. Okay, this is my fermenter. That is, I have not patented, although I have 3 patents, but I have not patented anything for the farmers, because it's for the God. So I will not go for patenting anything. Whatever I will show here, that the instrument what I have designed for the farmers. And this is my fermentational vessel. So these are the tablet supplied from the lab. Cut any one tablet, put it here, then pour the water. For that reason, I don't think you need any degree. Just put the water here, shake well to mix the media. Then you need to sterilize it. You need a autoclave. What is the price of the autoclave? It's around 75,000 rupees. So I have also changed these 75,000 rupees to 3,200 rupees. Big pressure cooker, fast city, then wait for 20 minutes, take out your material, that's sterilized. Okay. So after getting the sterilized media, then you have to inoculate it. For inoculation, you need laminar airflow. Fantastic. What is the price of laminar airflow? It comes around 1 lakh rupees minimum. That is also a local made. So that 1 lakh rupees, I changed that 1 lakh rupees into only 100 rupees. That laminar airflow. That 1 lakh rupees we generally practice for an inoculation. But here I have devised a technology for the farmers. This is the master culture media which supplied from the laboratory. This is dispovan, autoclavable. You can use it repeatedly. 50 ml dispovan, and the needle, I just elongated that needle, make it bigger one, and you just put it, draw the culture, and just inject it. So this is a very simple thing that you can easily inoculate the organism in the sterilized media. Then after that, put it in the fermenter, that the biomass production unit, wait for 3 days, 72 hours, 10 to the power 13, 12 to 13 cells per ml. So in a single batch, you are able to produce 30 kg of azotobacter, 30 kg of PABSB, 30 kg of all the PGPR. Also, beside this, you can produce BT, Bacillus thuringiensis. You are able to produce Pseudomonas. These two are biocontrol agents because the bacteria can be grown in this particular fermentation system. And 30 kg, if I sell that 30 kg in the market, what will be the price? 30 kg into 300 is 900 rupees. And my farmers are producing all these things at a cost of 400 to 600 rupees per kilo. Per 30 kilo, means 15 to 20 rupees per kilo. And with a birth certificate. Once the fermentation completed, you take the material, dilute it in the water, and just spray it in the field. That's a very simple thing. They can do it easily. So that technology I have given, and using this technology, they are able to produce so many different types of organisms by themselves. Besides this, they need the foliar nutrition. For foliar nutrition, they need different types of vitamins, so-called vitamin by the farmers. So this is vermibed was now available in the market. It comes around 800 rupees per liter. But here we have designed a very simple technology that only one time invest in one plastic drum, and this is the layers you have to make. One stopcock is there. Then layer of bricks, then you use compost, then use cow dung, then earthworms, then cow dung and compost, and shower the bed with the water. Then after 10-15 days, you will get this type of solution. Then gradually the color of the solution increases, and keep it in the bottle and use 5 to 10 ml per liter every 15 days interval. And you just see the composition of this vermi-wash, that is rich in carbon, rich in nitrogen, rich in phosphorus, potassium, also contains so many types of micronutrient, also contain different types of beneficial microbe, which is important for the growth and survivability of the land plants. So just think about the investor. In market, you will get only 800 rupees per liter, but you just invest only one time, then you get this vermi-wash for lifetime only. Besides this, in market, you will also get amino acid. and you just see the benefit of amino acid for growth and development of plants. It improves protein biosynthesis, resistance to abiotic stress, photosynthesis, stomata activity, chelation, phytohormone, pollination, and fruit formation. It also increases the microbial activity. So for that reason, we use generally normally the fish, the very low cost fish, any low cost fish or fish waste, and you need jaggery, chitagur, that jaggery, that is 1 kg of fish. Keep it in a jar. Then you have to ferment it. For fermentation, you need incubator. Am I right? For incubator, it costs around 75,000 rupees. But I made an incubator for my farmers. That if you see that today morning there is a temperature little bit low. Now it's humid and high, and evening it's again goes down to low. If you think about the temperature of the soil, day and night, more or less ambient temperature, it varies from plus minus 2 degrees centigrade. So I utilize that very simple knowledge, and I made a fermenter. Just dig the soil, keep the fish and that jaggery in the bottle. Okay. Now put it this way in the soil. Okay. So only 15-minute investment of time. You reduce the investment of 75,000 rupees. Every day morning you just stir once, because aeration is needed to get better fermentation, and covered by a net, mosquito net. And after 45 days, please take the screenshot. Those who are actually from the village area, please tell your parents, tell your farmers, try to make this thing in their home. At least they can reduce the investment of 1600 rupees to 40 rupees only. Okay. And this product is at least 10 times higher quality than the product available in the market. So please help your Bhagavan, those who are actually feeding you every day. So take a snapshot of this thing and tell them that you prepare this thing and use. After 45 days, you will get a blackish color solution. Then keep it in the bottle. Just filter it, keep it in the bottle, and use for 6 months. 5 to 10 ml this phase amino acid per liter. This is the dose for application. Okay. Beside this, also they are able to produce different types of humic acid for the better growth. You can see the impact of humic acid on plant growth and development. Then next one is the disease prevention. So what I have done during this process, that this is basically a new strategy, that is improvement of innate immunity in the plants. So we develop certain small molecule, biological small molecule, which mimics the pathogen. Okay. So in spite of pathogen, when you spray this small molecule over the surface of the plant understood that pathogen is there, but no pathogen is there. So once they understood that pathogen is outside, they will boost their immune system, that innate immune system, so that when the real pathogen sit over it, they can immediately fight against this pathogen. So you can prevent the disease rather than cure at the instead. That simple strategy we have utilized. What we have used the prawn cell, crab cell, and the big mushroom cell wall that contain chitin. We make the chitin into chitosan to chitosan nanoparticle, and we use this chitosan nanoparticle as a pathogen mimicking elicitor molecule. Thereby we can produce, we can induce the innate immunity in the plants. That actually we have made a product on that, and this person actually did PhD on that, Swarnendu Chanda. Now he actually got a job in a scientist post at Delhi. After 1 year, he left that job. Now he become an entrepreneur. at Bangalore. This guy. And that was published in scientific reports. After that, Nature India actually make it a actually news, and it's an important invention that can be utilized. After that, Rajya Sabha TV came to our laboratory, make a documentary on that. So initially, if I tells farmers to do all these things, it's a tough job. So what I try to do in case of this farming system, you just put all these things, that crab cell or spawn cell, okay, or this big wood rotting mushrooms one, in your compost. In the compost, there are lots of bacteria who release chitinase. They can destroy chitin to chitosan. So if you use this compost in the rhizosphere, they will boost the immunity in the plants. ISR, induced systemic resistance. Okay. And for disease protection, you need Beauveria bassiana, that can for controlling of insect. For pathogen, I just consider only bacterial pathogen, fungal pathogen, and insect borne pathogen. I have not considered any viral pathogen, virus as a pathogen, because virus has to be carried by something, either through seed or by insect or by other thing. So if I cut down that entry point, so no viral disease will be happen. So you can control this insect by Beauveria bassiana, aphids, whitefly, psyllids, termites, grasshopper, mealybug. So you can control it. Caterpillar, thrips. So you can control by this Beauveria bassiana. You can control whitefly and other insect by Verticillium leccani. You can control different types of caterpillar with the help of Metaherginium. You can control nematode by Pacillomyces linacinus. So for a holistic approach, you need to grow all these organisms for make the land inorganic to, or convert the land into inorganic organic one. But in market, you will not get anything. So mostly you will, in the market, you will get only trichoderma, which can control fungal diseases or some insect disease, some nematode diseases. So I have Palaeobotany harzianum, Trichoderma viridi, Trichoderma aspergillum. So here you will get 3 different types of trichoderma. For insect control, we have 4 different types of fungi. 1 bacteria is BT is there. So 5 plus 3 is 8. And for bacteria, that is Pseudomonas is there. So if you use all these things in your land, so your land will protect herself from any sorts of pathogenic attack. Otherwise, if you only use a certain amount of Pseudomonas, if you only use certain amount of Trichoderma, again they don't have any birth certificate, and they are selling at a cost of 250 to 500 rupees per kg. Okay. And you do not know the viability. You do not know the CFU. You do not know the health condition of all these organisms. So do you think they will work for you? They will not work. So for that reason, actually, farmers are reluctant towards the organic cultivation, because nobody is supporting farmer all sorts of with all sorts of weapons. Okay. So some formulation also I have given. I have 100 different types of formulation through which they can SOS manage certain crop diseases. And after that, I generally, we scientists, when we work, generally after completion of our works in the lab, we generally tells the other people, I have done this thing in my lab, you can test it in your field. But lab to journey from lab to field, it's really a hectic schedule. There are certain parameters you have to understand. So generally, in my cases, when I go for any translation research, I use this thing by myself first. Then I tells my farmers, you can utilize this thing in your cropping system. So I have tested this thing in different cropping system. This is the this is a rice cultivation system. My father also cannot understand that it will work because they are habituated with this inorganic way of cultivation. They cannot understand without fertilizer, without pesticide, can you able to produce a grain? But I have taken this challenge. He has given me a very small land. Just see, this is one less than a katha. I put it my plants here. I utilize SRI technology. This is conventional. After 19 days, just see from a single plants, I got 8 to 10 tiller. Meeting just boy. Okay, I got 8 to 10 tillers. Okay, then just during that flowering time, you just see the changes in number of tillers. This is the conventional, only 10 to 12 tiller per bunch of the plants. Here from a single plant, I got 47 to maximum 65 tiller. 47 to 65 tiller from a single plant. Just see the changes in root. This is the conventional root and this is my system of cultivation root. It's a thick and white color root. This is thin brownish color root. And just see single plant. Just see the panicle size. It is 185 seeds per panicle and this is 165 seeds per panicle. Everybody talking about that organic is not giving good productivity. Just see here, or it's 1.273 times to 1.5 times. I am getting more productivity than that of inorganic one. So after that, now this is the part how I have transferred this technology to the marginal farmer. The people, the students and other researchers, if you think about to enter into this particular area to become an entrepreneur, so you have to make a cooperative, use your farmers in your locality. So convince your farmer to your locality, make a cooperative system and try and help them to produce this organic food and then after that the produce or try to accumulate every day and sell it, and from that thing you will get the sustainable, you can develop a sustainable model for cultivation system. So that way you can think about the transfer of this technology to the marginal farmer and to become an entrepreneur. So this is the first model I have introduced at Balagor, and this is the model at the Balagor. This is the laboratory of the farmer. That is the 6 feet by 8 feet, a small laboratory for the farmers. And in this particular laboratory, I first introduced my technology. And one day I brought them to my institute, talks with these people. Highest qualification in this particular team is class 12 pass only one, and many of them class 6, 7, 8 pass, and rest only know how to sign. Just 1 day training, okay? And you just see the educational qualification. After getting training, now see the masters. They are producing all the product by themselves only. They are able to inoculate. They are able to produce media. They are able to produce different types of fertilizers, biopesticide, azote vector, PSB, BT, then Bacillus subtilis, then Bacillus megaterium, Bacillus lichenoformis, Bacillus amikfaciens, Bacillus valenciensis, Pseudomonas fluorescens. They are able to produce with the help of this fermenter, and they are also know how to inoculate in a solid culture system. They are inoculating. Just see. And for liquid culture, we generally use conical flask. Okay, one conical flask comes around 175 rupees. Okay, so for liquid culture, if so 175 rupees is needed for liquid cultivation to get a conical flask. Again, I reduce that 175 rupees to 2 rupees only. Just see, my farmers are producing fungus and bacteria by themselves, all the fungus, and you know what is the price of this fungus they are producing, and single bottle is equivalent to 1 kg of the product. Single bottle is equivalent to 1 kg of the product, which cost around 250 rupees to 500 rupees. Okay, But my farmers are producing all these products at a cost of only 2 rupees. Please note, they are producing live organism, live fungi by themselves without that so much degree. They don't bother about degree. Okay. They are able to produce of all these things by themselves in their premises at a cost of only 2 rupees. After complete growth, they pour it in the bucket, mix it water, filter it, keep it in their spray drum, spray it in the field. That's all. Now I talked about amino acid. Just see, one of my farmer becomes so interested about this technology, he made 1400 liter, 1400 liters of amino acid by himself only, and just see the color. No smell of fish, no smell of that molasses. It's a sweet, beautiful, thick brown color liquid. You can see it. The brown color liquid decaying from this vessel. So this is the amino acid produced, and now he is actually giving this amino acid to all the farmers in his village. Ajao lelo. This is the product. You can use it and you can see the magic of this solution. Okay. And this is pheromone trap for fruit borer. And just see the product. Now see the real the result in the land. Not a single molecule of chemical fertilizer, not a single bund of any chemical pesticide. No disease. Just see color. This time potato, actually potato is the pride of Hooghly. This time because of the rain condition, potato, most of the potato field become destroyed. But see my field. We got 80 basta, 80 bags of potato from our bigha. And another condition I always, when I work with these people, I give one condition to my farmers, that when you go for organic one, then everywhere that the price of the organic product become high. Okay, if you purchase this oil at a cost of 190 rupees to 210 rupees, but if you go for organic, it will comes around 700 rupees per liter. Okay. But one condition I always give to my farmers, that you shouldn't sell organic product not more than the market price, because I made your production at a cost of, at a very low cost. The production cost actually in my system at a cost of 7 rupees per day per bigha only. 1 cup of tea cost only. 7 rupees per day per bigha only. So you shouldn't ask a single penny higher price than that of any other vegetable available in the market. Okay. So lab to land, now lab to land to consumers. Within 2 years, my farmers become suited booted. Just see the smiling faces. Within 2 years, they purchase one small car. Every day they put their harvest in that small car, reach to the different places, housing complexes. Within they sell for 2 to 3 hours. Before reaching there, there is a queue of people interested in their product. They sell all their product within 2 to 3 hours. And got more than 20 to 30,000 rupees per day, earn 20 to 30,000 rupees per day. That way, they become right now sustainable. Okay, this is the whole team. Besides this, during pandemic time, when everybody kept yourself within the room, I have tried to visit different corners of West Bengal. That is, this is Adghara, this is Canning, this is Jhargram, this is Jhalda. This is Bolpur Santiniketan. Just see, in Bolpur, now we have established one laboratory there. Now the farmers are producing all these things by themselves. This is Santiniketan. This is Hirband. that is in your nearby places, Bankura, in Hirvand area. Mostly you just see the people who is working with me. Hats off these people. These people this year got a award of 5 crore rupees from HCL company. That HCL actually asked project from different groups for sustainable development. 1280 project actually they got 1280 project, out of which they selected 3. One is from Bandana Siva, 2 is from Krishnendu Acharya's group, one is Hirvan, another one is Kultoli. And the 2 got 25 lakhs and 1 got 5 crore rupees. That is the first one. Hirvat got 5 crore rupees. Kultoli got 25 lakhs rupees. So these people, just see, they are producing Jivamit. They are after production they immediately transfer this thing to the land. They are producing during night time all sorts of bacteria by themselves. Just see the faces. Just see the faces. They are really the man of the soil. They are really the pulse of the soil. Okay. They don't have any vanity like us. So they are able to produce all these things. Once they produce, immediately they transfer these things in the land. Just see, they're spraying in the land. They are producing different types of fungi by themselves. Just see. They are producing Trichodama. They are producing Biberia bassiana, Verticillium lacini. Just see, here they are producing the product which comes around cost of 5 to 6 lakhs rupees. But they are producing in few hundred rupees by themselves with a birth certificate. Just see their product. Okay. So do you think that organic is not the commercially viable one? You have to give appropriate technology. You have to stand behind these people. Otherwise, you cannot convert your land into organic one. It's a very simple thing. You have to transfer this technology to the ground level. It's a duty for a college teachers and university people, because all the institute made for the manifestation of mankind for the society. So this is the Kultoli. They got 25 lakhs award. Just see their product. This is Naton Hirvan. They are also producing all these things by themselves. And during this lockdown time, I have made more than 300 people to become entrepreneur utilizing this technology. So those who have land, they can become an entrepreneur. But those who don't have land, how I can create jobs? So that actually that time I just think that how can I create job for the people, those who do not have land by themselves. Okay, in their family, they don't have any land. So I try to convince certain people, why should not you, the people having other company or other industry, I try to convince them that why shouldn't you think about to invest your money into the organic one or agriculture. Most of them immediately told me, yeah, that's not a profitable one. That's not a profitable one. So I told them, I will give you the technology. All the technology I have provided to the farmers. Actually, it's in my most of the technology I've actually given for my own cost. Actually, to create a confidence within them. So I told them, I will develop your laboratory. No investment is required for that laboratory. Plus try to invest in that thing. So one fellow came to me and told me, I have a barren land in Bakura, Saltora area. Okay, probably the people, many of you know that place, that the Saltora area. So can you make that land into a fruit orchard? I told him, yes, if I get the soil, then I can convert it. So this is the land. Actually, never ever there has been cultivated by any people earlier. So this is basically a barren land, And if you see the health card of this barren land, no nitrogen, no phosphate, no potash, and more or less. And carbon is very poor in that particular land. So I made a small laboratory there. Just say, I have converted that land to a gold mine, only 10 acre of lands. This year, this company is thinking they will earn more than 45 lakhs rupees from this small land. So this is the dragon fruits, 35,000 dragon fruits plant. This is intercropping. This is guava. This is guava and intercropping. These are the pomegranate, Malta, seedless lemon. So they are able to produce all these things without utilization of any molecule of chemical fertilizer and chemical pesticides. And productivity definitely higher than that of any other system of cultivation. And most important part, that in this particular type of organic cultivation, at least 15 to 20 family now feeded every day from this agricultural system. So they got job. So that way you can create job. If you have the land, you can create job for the people. Also, you can become an sustainable entrepreneur. So beside this, I have transferred this technology to the polyhouse cultivation system. In a polyhouse cultivation system, they actually now government gives 50% subsidy. So again, I have tried to motivate these people who are taking fund from the government. Why shouldn't you think about your polyhouse cultivation system in an organic one? Because polyhouse cultivation system is a closed cultivation system. If you closed cultivation system, if you use the inorganic salts and pesticide, within 2 to 3 years, the soil becomes so toxic, you cannot grow anything there. So just see, this is a polyhouse of 1 acre of land. It is in Hooghly, that is Govindapur area. They are producing different types of high-end vegetable. They are producing that is English cucumber. They are producing broccoli, bok choy. Now they are producing baby spinach. They are producing that is coriander, because it is costly right now. So in this particular type of entrepreneurship, at least 20-30 family now become got job in this particular polyhouse system, and they are now become sustainable. Okay, this is the polyhouse system. Another polyhouse, I have tried to produce this capsicum, because as you know, this plant is very much prone to disease. So it was really a challenge for me. When I talked about I will grow this capsicum without any fertilizer and pesticide, most of the people actually laughed at me. Are you a mad one? So, but I have taken that challenge. And from this small polyhouse, we have produced 3.2 ton of this bell pepper without any damage in that bell pepper. Okay, no pathogen attack, and the health, you can see the health of the peppers too. And also, once you grow all the vegetable in an organic system, it will increase the survivability or the post harvest, improves the post harvest time. So you can manage your crops for a longer period. So beside this, also I have tried to make entrepreneur in the to promote horticulture. One person here in Kolkata, he is actually selling flowers, cut flowers. So he had a land at Deganga region, and that mostly the salty land. He tried to cultivate the leaves required for bouquets. So he asked me, sir, can I produce these important these leaves in my land, because I have tried for last 3-4 years, but I didn't get the result, because most during the summer days these plants are burned because of the high increase in the ratio of sodium chloride in the salt. So this land I have taken care from last December, 4th of December. I have developed a laboratory there, and just see the result. Just see the health and growth of the plants. Okay. Now, entrepreneurship to promote organic farming in the ABA model. One interesting abstract I have found in your this that abstract book, that principals are also working on the rooftop gardening to promote this thing, strawberry production. It's really good, sir. And I hope my lecture will give you some food for your thoughts so that you can utilize it for further development. So this is actually, if you look into the Durgapur area, if you look into Calcutta, all the cities and metropolis actually increasing vertically, and all of us always crying for global warming and all these things. But if we can produce organic food in our small 10 foot 10 feet by 10 feet means houses. Now we are living in 10 feet by 10 feet. So I have developed a model. If I get only 4 feet by area in any house or any flat, so here I can I have a model through which that family can get at least one poison free vegetable every day for their dishes. So this is my model. This is the model where we can cultivate 13 different types of crops in this single platform, and the length and breadth is 4 feet by 4 feet. You can keep it in the rooftop, you can keep it in your veranda. So that way we are producing different types of crops for the family. These are the different types of crops we produce in this vertical gardening system and organically. And every day you will at least get at least one product for your dishes without any poison, without any fertilizer. Actually, if we think truly, every day morning we carry back and visit any local market, purchase some poison from the market, bring it to my home, and my wife or maid made it delicious dishes, and every day morning we feeding our beloved one teaspoon of poison, and evening 1 teaspoon of poison. That's the routine is going on. Actually, it's very difficult to remember name 5 vegetable in the market which is poison free. So at least you can create a small area where you can get certain poison-proof vegetable, rich in antioxidant, rich in fiber, rich in vitamin, rich in minerals, and also the green fresh vegetable in your dish. So we can think differently. We can think our pattern of living in a different mode. Not to blame anybody. You should do. Try to start from your home. Okay, so then this is also another area that is for urban area, that is the organic hydroponic system. in case of polyhouse, I got the hockey area where blank places where we can utilize this hydroponic system to get extra product from this polyhouse system. So initially this company actually taken a quotation to making this hydroponic system. The quotation actually came 15 lakhs rupees. So I told them I will help you to make this hydroponic system, and I made this hydroponic system because I work as a carpenter, and I have shown them how to make this hydroponic system. So they made it. I just shown them how to do all these things, and I made this hydroponic system in this hockey area at a cost of 2,50,000 rupees. So 15 lakhs rupees goes down to 2,50,000 rupees, and every week they are harvesting 5,000 fresh harvest of different types of leafy vegetables like bok choy, lettuce, romaine, then then basils, sweet basils, so pudina. So with the help of this hydroponic system, they are getting extra money from this vacant area actually. Here I would like to give the example for my young friends that these two guys, that is Moinak Gupta and Lalit Jawar, they actually IITian. They had a very good job in US during lockdown. They came back from US and they make a aeroponics or hydroponic system near about Gujarat, in an area of Gujarat. And you know the turnover of these two fellow? The turnover is 80 lakhs rupees, 40 lakhs rupees per month. 40 lakhs rupees per month turnover. Okay. So beside this, I also insist these people, when you are producing this value added compost, you can sell it. They are also selling their compost in a good price and also earning their money. Beside this, I would like to enter into another area where through which you can become an entrepreneur. So nowadays, everybody now become health concern. Okay, now 124. Okay, so everybody concerns about their health. If I provide you one food product which is rich in vitamin, rich in minerals, rich in antioxidant, then rich in fiber, then this food can protect your body from free radical stresses. It can reduce, it can be a very good diet for diabetic patient. It can reduce cholesterol level. Also, it can increase the peristaltic wave, and it is a diet means it's a low fat diet. Okay, it can protect you from it can protect your liver. It can protect from cardiac problem. It can be a very good immunomodulator. It can protect your body from the cancer. If I sell this product to you, how much price you can give for that product for 1 kg? 4 to 5,000 rupees per kilo. Am I right? But if I tell this is a mushroom, oh, that's a mushroom. Okay, mushrooms gives all sorts of benefit. Okay, and in your area, that Durgapur area and Bankura area is a really a big, good hub for different types of mushroom. You know kurkura chatu, you know kent patra, you know jam patra, you know murgi chatu. In the next session, my student will talks about this kent patra, jam patra. What is the health benefit of this kent patra, jam patra? How it is acting as an immunomodulator. One of my student actually worked on isolated one compound from your kurkura mushroom, kurkura chatu. She will talks about the anti-cancer effect of this kurkura mushroom, that is Astrias hygrometricus. So please listen this talk that will boost your or thought you will get a new direction to think about your science. Okay. As you know, we born and brought up in a state like West Bengal. It's a mini India, the only state in India where you will get the Himalaya at the north and the sea at the south. Go to west, you will get the dry area like India, centrally alluvial, and Tarai Duas, and one gift is this Sundarban. So all this geoclimatic condition of this state made it a treasure house for the luxuriant growth of flora and fauna. So you'll get numerous types of edible mushroom. This is Astrias hygrometricus. You will get in your locality, Kurkura chatu. Okay, you will get an enormous number of wild edible mushroom that can be utilized for development of drugs and many other purposes. And some of them you can domesticate it, but most of them you cannot domesticate because they have a mycorrhizal association with other organism. So this is called as Kent Patra Rasula. This is called as Jampatra, the color of the Pilias. This is called as Jhal Chatu. These are commonly found in this the Durgapur area also. Also Bakura. This is Karange's mushroom, Chota Karange, Bada Karange. This is Termitomyces. This is Bali Chatu. This is Wi Chatu. Okay, this is Termitomyces microcarpus. So beside these, you will get different types of mushrooms from your area too. So these are the new mushroom we have discovered from lateritic soil. This is Losula Kanadi, Sadapatra. This is Losula intervenosa, Murgi Chatu, and this is also Murgi Chatu. That is Losula elato reticula. This is Losula savalpina from hills, all edible. So you can think about to make an industry on that. So you can think about canning of this wild mushroom. You can think about the product, like different types of product from this wild mushroom, and you can become an entrepreneur by using this mushroom for value-added product development. So you can think about the papad, you can think about the tea, you can think about the soup, you can think about the cosmetic also. Beside this, mushroom, as you know that mushroom generally found during the rainy time. Okay, so you can think about the germplasm collection unit from the mushroom, and mushroom cultivation is a technology through which you can easily convert your investment 3 to 5 times within 15 to 30 days. I'll show you that thing. You can convert your, suppose you invested today 100 rupees, within 1 month you will get at least 300 rupees. Just show me any other business. As a biologist, we never think about that. Try to think differently. Try to create job. I'll show you that how my peoples are engaged in this mushroom cultivation technology and how they become self-sustainable right now. So spawn production. So it's a very easy technology. So in spawn production, you need wheat and you need to make spawn. So you can easily produce spawn from this, that is seed for the mushroom production. And in case of spawn production, you can easily convert your rupees 3 times within 15 days. How many days? Within 15 days. Just see, you can easily produce spawn. So here we have developed a particular strain of Pleurotus. That Pleurotus you can cultivate round the year. So generally we cultivate Pleurotus during the winter time, but to become an entrepreneur, you have to supply chain for round the year. So in my laboratory, I have developed a strain which we can cultivate and grow for round the year. So this is the person who actually BA pass graduate, who actually act as an laboratory assistant for last 7-8 years in my lab. Then during lockdown time, I asked him, why shouldn't you think about to become an entrepreneur? So I developed a laboratory at Andul. So now this person actually earning every month 50 to 60,000 rupees by selling spawn only. This is only the scale of 2 years. Only within 2 years, he scaled up the and earning every month at least 50 to 60,000 rupees per month. And from him, he also giving training right now, and some of the people actually taking spawn from him. They are producing mushroom in their area. This lady actually earning by producing mushroom every month more than 1 lakh rupees, but just by producing and selling the mushroom. Also, she developed certain product for the certain commercial product too. And just think about the mushroom spawn production. Can you predict if you enter into entrepreneurship in only in mushroom spawn production, how much turnover you can get in life or year? I'll show you one person. This is Tushar Mehta. He actually a graduate from commerce department from Rajasthan basically. 1996 he started mushroom spawn production unit in Bhawanipur. Right now, if you ask any mushrooms from this person, you have to wait for at least 1 month. Then you will get the spawn. So much so demand is there. And his turnover is only 7 crore rupees annually. Please note, 7 crore rupees annually. Now he purchased a big house in Behala and converted the whole house into industry. So the cultivation process, again, it's a very simple, and in cultivation process, within 30 days, you can convert your investment into 3 to 4 times. Okay, so this is the product you can get from the mushroom. Okay. So different types of mushroom strains we have, different species we have. Now we are actually promoting this mushroom cultivation in different rural area. Also, we are motivating graduate students, those who are interested to go for entrepreneurship after their graduation, so they can take it as a challenge and they can utilize this technology to become an entrepreneur. So all the so this is Calosivy, this is Volvariella, this is Pleotas zamo, this is my strain, that is Pleotas, which you can grow these Pleotas for round the year. So I was engaged with this system of mushroom cultivation at Narendrapur Ramakrishna Mission for a long time, where I am giving training to the blind boys to become an entrepreneur. Okay, many person from Bankura also getting actually trained by our team to become an entrepreneur. So all the people just see these people. All the blind boys, they are able to produce spawn. They are able to inoculate the thing. They are able to produce the bag. They are able to harvest this thing. They harvest this thing, sell this thing. Also, they dry the mushroom and sell it for the longer time period. Okay. So you can dry it, you can keep it for a longer period, and you can sell it. So all these persons now getting training from this Narendrapur Ramakrishna Mission, and now they become entrepreneur. So during the last international conference, one of my first entrepreneur, he's Baburam. He's a blind one. Okay, he doesn't have eye, but he has the inner eye. Actually helps him to become entrepreneur. Every day this person producing 25 kg of mushroom by himself only and sell it, and now he become a entrepreneur, sustaining by himself, and he has a good family. Just see, he doesn't have eye. Just see, we have so big eyes, but we don't have the wisdom. He has the wisdom. And now I am trying for this medicinal mushroom. This is 6,000 rupees per kg. This is 2 lakhs rupees per kg. This is cordyceps. This is Ganoderm. I'm trying to domesticate it here. So I am right now reaching towards the end. Here are some snapshot of some young entrepreneur. Just see, some of you like you, some of them like you. Okay, they during this period they become entrepreneur. Now they become the millionaire. Okay, so try to take a snapshot of this slide. Please visit them in Google. Okay, and try to get inspiration from them, and try to become an entrepreneur, or try to think differently what you will do after your graduation, what you will do after your master, what you will do after your PhD. Okay, so try to think differently. Okay, so these are the organization from where you will get the different, they have different scheme. You can get fund from this thing. National Horticultural Board gives 50% subsidy for making the greenhouse or polyhouse. Okay, so also you try to take a snapshot of this one. So this is my concluding slide. So thank you very much for giving me so much of your valuable time. This is a blank slide for you. I am just keeping a blank slide for you. Please write something here. That is your future dream. Please write something that actually help you to achieve the dream. And after 5-10 years, in some seminar, you will present your talk about your achievement and how you achieve that thing. Maybe I will be there in the audience. After your talk, definitely I'll jump from my seat. I'll give a clap for you. Well done, my dear friends, well done. With a great hope, I just like to say, don't think outside the box, lead outside the box. Thank you very much. Thank you very much for giving me this opportunity. Thank you very much for your patience too. Thank you.
Thank you, sir. Thank you so much. And oh my God, what we have just witnessed. Actually, truly, sir, I am speechless. Actually, it's beyond any compliment, any praise. But no wonder, actually, who know you from the very beginning, we know that you work with the people at the grassroots level, sir. And truly, the agriculture to me is not a science. It's rather an experience. It's rather a nature itself.
Yes.
I sometimes wonders how this traditional farmers who told them to use cow dung, the leaf litters, the urine as a manure. I can bet it's no single scientist can told them to use this as a manure. It's the mother nature that taught them to use this. So yes, it's greatly pointed out the different avenues of entrepreneurship, the different avenues how to develop the socio-economic structure. So your lecture truly emphasized the importance of the conservation of these indigenous things, of this indigenous resource, and to grow on a zero industrial input, and would be a necessary and essential step to address the food and nutritional security of this large democracy of the world.
Right.
And for the people who are still deprived to access this cheap and nutritional staple, mainly our farmer who are feeding us day and night from the time immemorial. Thank you, sir. Now this session is open for the interaction, and I will request everyone to put their
এই
questions.
সবাই বোঝা গেছে তো?
Yes, sir, absolutely clear, sir. There is no doubt, sir.
না, আমি টাইমটা দেখছিলাম, অ্যাকচুয়ালি অনেকগুলো স্লাইড তো অনেকক্ষণ সময় লাগছিল, তো আরো খিদে পেয়ে গেছে। স্যার থ্যাংক ইউ। হ্যাঁ স্যার, কেমন আছেন?
হার্টিয়েস্ট কনগ্রাচুলেশনস।
থ্যাংক ইউ স্যার, থ্যাংক ইউ।
অসাধারণ, এতগুলো আসপেক্ট যে তুমি ডিল করছো, মানে যে বটানিস্ট আমি ভাবতেই পারছি না, এরকম আমি কাউকে দেখিনি বটানিস্টদের মধ্যে।
থ্যাংক ইউ
ইটস
স্যার।
রেয়ার কোয়ালিটি। আই মাস্ট কনগ্রাচুলেট ইউ। বিউটিফুল, এক্সেলেন্ট, এক্সেলেন্ট।
আপনাদের বোলপুরে করেছি স্যার, বোলপুরে।
কনগ্রাচুলেশনস। আই এম ভেরি হ্যাপি টু লিসেন।
থ্যাংক ইউ স্যার, থ্যাংক ইউ স্যার, আপনাদের ব্লেসিং।
একবার এসেছিল, এতটা বলনি, ছোট করে বলেছিল, অল্প একটা আসছে।
হ্যাঁ হ্যাঁ,
মনে আছে? ভেরি হ্যাপি, এক্সেলেন্ট স্পিচ, এবং এতগুলো ফাইন্ডিংস, এতগুলো দিক খুলে ধরছো। ইয়াং জেনারেশন যারা এন্টারপ্রেনিউর এইসব দিকে যেতে চাইবে, they will be মানে benefited with this aspect if they take it seriously.
হ্যাঁ স্যার, এখন সময় এসছে টু ক্রিয়েট জব স্যার। আদারওয়াইজ ওয়ার্ল্ডের ইকোনমি থাকবে না, মানে ইট নিডস দ্য ক্রিয়েশন অফ জব। আর এইখানে যারা এসছে তারা তো সবাই বাচ্চা ছেলেপুলেরা, তোমরাই আগামী ভবিষ্যৎ। অ্যাকচুয়ালি আজকে আমি এতক্ষণ যাদের সঙ্গে কথা বললাম, আমি কিন্তু কোনো ইন্ডিভিজুয়ালের সঙ্গে কথা বলিনি, আমি কিন্তু ভবিষ্যতের আমিগুলোর সঙ্গে কথা বলেছি। সো উইথ দ্যাট
Right.
হোপ, হ্যাঁ স্যার, থ্যাংক ইউ স্যার,
Sure. All the best. All the best.
থ্যাংক ইউ স্যার।
So any questions from the audience? We'll happy to take few questions. I think sir, it's quite clear to them.
Hey,
Absolutely crystal clear.
where is Devnath sir? Is there Devnath sir?
No, sir, he's not here, I guess.
আচ্ছা আচ্ছা।
And one more thing, you can, I guess, and you can contact any time to Professor Krishnendu Acharya for any help regarding your agriculture farmings and all. Anyone?
স্যার আমি অঞ্জন বলছি।
হ্যাঁ এই তো, আরে অঞ্জন আছে, অঞ্জন আছে, আরেকজন ডাক্তার ইয়ে ছিলেন। হ্যাঁ বলো অঞ্জন, কেমন আছো?
হ্যাঁ স্যার ভালো আছি।
হ্যাঁ হ্যাঁ,
আমি তো পুরোটাই শুনলাম আপনার।
ইয়েস।
তো স্যার আমার একটাই বক্তব্য আছে যে বাঁকুড়াতে যদি আপনি একটা হ্যান্ড সাউন্ড ট্রেনিং এর জন্য একটা ছোট সেন্টার খোলেন, তাহলে আমার যে চাষিরা আছে তারা, আমি ওদের সঙ্গে কথা বলেছি, তাহলে ওরা একটু উপকৃত হতো আর কি।
না আমি তো এই এখানে তো তোদের পাশের সেই কি যেন মঠটা রে,
হ্যাঁ হ্যাঁ হ্যাঁ,
ওইখানে তো একটা করার কথা আছে, জানি না কবে হবে। কারণ
না ওখানে আমার না হলে স্যার আমার কাছে করুন, আমি তো একটা এস্টাবলিশমেন্ট বানিয়েছি।
সেটা দেখে এলাম তো।
হ্যাঁ স্যার,
করা যেতে পারে আগামী দিনে।
হ্যাঁ ওইটা একটু সিরিয়াসলি চিন্তা করুন, আমি আমার সঙ্গে পরেও
না ওটা বেস্ট হচ্ছে ইফ দুর্গাপুর গভর্নমেন্ট কলেজ can take an initiative at that মানে that particular location is very near to your place too. তাহলে কিন্তু হ্যাঁ, তাহলে একটা আগামী দিনে হয়তো ওখানে বেশ করে কিছুটা জায়গা ছড়ানো যাবে।
ওকে স্যার। আমি স্যার ওই যেটা আপনার অ্যামিনো অ্যাসিডের ওটা শুরু করেছি।
ইয়েস,
ওটা হলে আপনাকে আমি খবর করব।
হ্যাঁ ছবিটাও পাঠিও।
হ্যাঁ স্যার।
হ্যাঁ।
Okay, anyone else would like to say something? So I think yeah, sure.
Okay, I have a question, sir. Prashanto Saha.
হ্যাঁ
স্যার বলছিলাম যে আমাদের এখানে আশেপাশের ল্যান্ডগুলো যেরকম মেটাল কন্টামিনেটেড, তো স্যার সেখানে ধরুন বায়োরেমেডিয়েশন টার্গেট করে আমরা যদি মানে কিছু এনার্জি ক্রপস যেমন আমরা যদি কিছু চিন্তা ভাবনা করি, কিন্তু এছাড়া আর কি ধরনের ক্রপসকে আমরা মোটামুটি ওখানে কাল্টিভেশনটা আপনি একটুখানি সাজেস্ট করবেন স্যার?
হ্যাঁ, ওখানে যে এরিয়াগুলো কন্টামিনেটেড আছে বা মেটাল কন্টামিনেটেড হতেও পারে, আর যেখানেই হোক না কেন তুমি যে কোনো ক্রপই করতে পারবে, এনি ক্রপ, ইফ ইউ অ্যামেন্ড ইউর সয়েল ইন এ প্রপার ওয়ে। আমি যে বাঁকুড়ায় তোমার দেখালাম সালতোড়া এরিয়া, ওখানে কোনোদিন গরু ঘাস খায় শুধু, ওখানে কোনোদিন চাষ হয়নি, এবং ওখানে জেসিপি দিয়ে ভেঙে নিচে মাটি বের করেছে। তো সেখানটায় যদি একটা 10 একর জায়গা, তার মধ্যে তুমি 2 বিঘে জায়গা বাদ দিয়ে দাও, মোটামুটি 28 বিঘে জায়গার মতো একটা জায়গা পেয়েছি, তার বা আর একটু কম হবে। তো সেখানটায় 45 লাখসের টার্ন ওভার হচ্ছে। হ্যাঁ, দুর্গাপুরেরও একজন আমাকে কন্ট্যাক্ট করেছিলেন, তারপরে আজ তিনি যোগাযোগ করেনি। উনি একটা প্রায় 200-250 বিঘে জায়গার উপরে একটা কাজ করবেন বলে বলেছিলেন। করা যাবে যে কোনো ক্রপ, এনি ক্রপ ইউ জাস্ট লাইক টু।
হ্যাঁ, মানে ফাইবার ক্রপস যদি আমরা টার্গেট করি, মানে বায়োডিগ্রেডেবল ফাইবার ক্রপস বা যাই হোক, তাহলে সেক্ষেত্রে মানে সেটার ফিজিবিলিটি রয়েছে বলছেন স্যার?
এনি ক্রপ, কারণ তুমি যখন হোলিস্টিক্যালি চাষটা করবে না, আসলে আমরা অর্গানিকের নামে কিছু ফ্যান্টাসি করছি সব জায়গায়, তুমি দেখতেই পাবে। কেউ তো আর তাকে কিছু দেবে না, সবাই বলছে আমি যেটা করছি টেকনোলজি ডিরেক্টলি ওদের ঘরে ট্রান্সফার করছি। কারণ একটা চাষি কেন হাত পাতবে? আমাদেরই তো উচিত চাষির কাছে হাত পাতারে ভাই, কারণ যিনি খাওয়ার দেন তিনি হচ্ছেন ভগবান। আমরা তো যে যাই চাকরি করি, অ্যাট দ্য এন্ড অফ দ্য ডে ভগবানের কাছে প্রার্থনা করি দুটি ভালো খেয়ে পড়ে থাকবো। আর দেখো তো, সত্যি চিন্তা করে দেখো, এই হাট জিরে ওই লোকগুলো 150 কোটির পেটের ভাত রেগুলার দিচ্ছে।
এক্স্যাক্টলি স্যার, এক্স্যাক্টলি।
আর আমি এই মুহূর্তে বসে কথা বলছি একটা ঠান্ডা ঘরে, তোমরা হয়তো একটা অ্যাম্বিয়েন্ট কন্ডিশনে শুনছো সবাই, আর আমার ভগবান মাঠে গিয়ে এখন ঘাস বাসছে, ধানটার বীজতলা কালকে ফেলবে। তুমি ভেবে দেখো, তারপর আর গামছা নেই, নোবডি বদার অ্যাবাউট দ্যাট। তো সেই জায়গায় এগিয়ে আসাটা সবারই দরকার। আমি আমার ক্ষুদ্র ক্ষমতায় চেষ্টা করছি তোমরা ইয়ং যারা আছো এগিয়ে এসো আগামী দিনে, দ্যাট উইল ক্রিয়েট এ রিভলিউশন।
Certainly, sir. Certainly, sir. You are already leading a revolution, sir. Thank you, sir. Thank you so much, sir.
Sir, one more thing. Actually, we are planning to take our student to visit one of your farm regarding the
Yeah,
mushroom
no problem.
cultivation and
No problem. Mushroom cultivation, one thing you can do. If it is possible for me, definitely I'll be there. কিন্তু সেটা শুরু কোনো সেপ্টেম্বর মাস নাগাদ ইউ অর্গানাইজ ওয়ার্কশপ ইন ইয়োর এরিয়া, মানে তোমার কলেজেই তোমরা করতে পারো ভাই। হ্যাঁ, তাহলে সেইখানে আমি যদি পারি ওয়েল, নাহলে আমার যে ছেলেটি এন্টারপ্রেনার হয়েছে সে গিয়ে ট্রেনিং দিয়ে আসবে। সো দে ক্যান স্টার্ট উইথ দ্য মাশরুম প্রোডাকশন। আর দুর্গাপুরের মতো জায়গায় সেল করাটা কোনো কঠিন ব্যাপারই নয়। কারণ হচ্ছে ওখানে গ্র্যাজুয়ালি এতটাই ডেভেলপ করেছে, আর প্রচুর মানে লোক আছেন যাদের পয়সা আছে প্রচুর, ফলে ইটস ইজি। এবং আমি ওর জন্য লিফলেটও করে দিচ্ছি, হোয়াই শুড ইউ, আই নেভার সে, মানে আমি একটুখানি সময় নিয়ে বলে নিচ্ছি, আই নেভার সে যে মাশরুম ইজ এ ভেজিটেবল। মাশরুম ইজ নট এ ভেজিটেবল, মাশরুম ইজ এ প্রোডাক্ট। এটা মাথায় ঢোকাতে হবে। মাশরুমকে ভেজিটেবল বললেই তোমাকে বলবে ওই বেগুন আলু পটলের দামে দাও। ওকে, মাশরুম ইজ এ প্রোডাক্ট। কারণ হচ্ছে মাশরুম এইগুলো করতে পারে। লোকে বলে মাশরুম নাকি মাংসের মতো, আই নেভার সে মাশরুম মাংসের মতো। মাংস খানিকটা মাশরুমের মতো, এটাও বলে রাখছি। মানে তোমরা যখন কোনদিন স্টুডেন্টকে বলে মাশরুম খানিকটা মাংস খানিকটা মাশরুমের মতো, কিন্তু মাংস খেলে যে অপকারগুলো হয়, কোলেস্ট্রল বেড়ে যাওয়া, হার্টের প্রবলেম, ফ্যাট বেড়ে যাওয়া, মাশরুম গেলে ঠিক তার উল্টোগুলো হয়, সবগুলোকে কিওর করে। সো এই মাশরুমকে কখনো মাংসের সঙ্গে কম্পেয়ার করা যায় না, মাশরুমকে কখনো ভেজিটেবলের সঙ্গে কম্পেয়ার করা যায় না, রাইট না?
Okay, sir. Thank you, sir. So, I guess the session is over now.
স্টুডেন্টরা কে আছে? স্টুডেন্ট, বিএসসি স্টুডেন্ট কে আছে?
Students,
বিএসসি কোনো স্টুডেন্ট নেই না? এখন পরীক্ষার জন্য ব্যস্ত? হ্যাঁ,
এখন নিশ্চয় সব পড়াশোনা হচ্ছে।
ঠিক আছে, চলো।
Okay, sir. So the first plenary session is over now. So we'll take a break here, a lunch break of around 40 minutes, and we will again meet you, all the participants, in the post session 1 from 2 p.m. From 2:30 p.m. onwards.
Okay.
So we'll take a break from here.
সন্দীপন, তাহলে আমার লাঞ্চটা কোথায় যাবো? একদম। ওকে, টাটা সবাই। হ্যাঁ, তোর গলাটা অনেকদিন বাদে শুনে ভালো লাগলো।
থ্যাংক ইউ স্যার,
টাটা।
So again we will meet at 2:30 p.m. onwards for the poster session. Thank you. Welcome back, everyone, and a very good afternoon. So we will wait for a few more minutes for other participants to join in. Thank you.
Good afternoon, everyone. Am I audible?
Yes, you are
Yes.
audible, but
Yes.
echo sound is coming from your end. Can you
Now
please
is it
check?
okay?
Yeah, it's okay, fine.
Okay. So hello everyone. Now we will start our e-poster presentation session. Over the past few weeks, we have received quite a lot number of abstracts. After minutely scrutinizing each and every abstract, we have selected 26 abstracts that covers every corner of plant science. To start with, we will call out the participant's name according to serial number mentioned in the poster schedule. After calling the name, they have to share their poster and have to present it in total time allotted as 7 minutes. That is 4 minutes for presentation and 3 minutes for interaction. We will not allow any of the participants to go beyond that time limit, and you will get a reminder from us after 3 minutes to conclude your overall work. So best of luck. Be prepared to make your poster ready to share. So we will move towards our first participant, Miss Ayushi Y. Purohit, PG Center in Botany, Srimati S.M. Panchal Science College, Talod, Sabarkantha, Gujarat. I would request her to present her e-poster and Miss Akanksha Srivastava to get herself ready for the next one. So Miss Ayushi Y. Purohit, please deliver your presentation.
Hello,
Hello.
am I audible?
Yeah, you are audible.
Okay.
Please start your. Okay, please present your slide and get started.
Okay. Okay. Can you see my poster?
Yes, it is visible.
Okay. Okay, good afternoon everyone. I myself Ayushi Purohit. I am from Gujarat and today I am going to present my poster on comparative stem anatomy of 4 species of Euphorbia from surrounding areas of Talod. Talod is situated in Sabarkantha district of Gujarat state. We are finding here a wide diversity of plants, and I am pursuing my PhD work in the wheat plants of Sabarkantha district. So I am presenting here the patch of my work. So here I want to share with you that in my study I have found many various types of wheat plants belonging to different families. Wheat plants are basically the plants which grow along with the crops, and they are known as unwanted plants. But according to the various previous researches, I can found that the wheat plants have also many more uses. Like they have medicinal purposes. Also, they are useful as ornamental or ethnobotanical uses. The main aim of my study is to focus the anatomy of the plants, as anatomy is the base of every research work we can say in the plant anatomy. It is useful in taxonomy to identify the plant. It is used for we can say that for the drug adulterant, that the plants are various other plants are used instead of the original plant. So this study can be useful to identify the real plant which can be used as a drug in various medicines. So here I have taken the 4 plants of the same genus, that is Euphorbia genus, Euphorbia heta, Euphorbia hyperacifolia, Euphorbia dracunculoides, and Euphorbia microphylla. I find these 4 species growing as a weed in the crops of our crops of Sabarkantha district, and it is found in more number we can say. So for the morphologically, this all 4 has many variations. We can say that in morphologically we can identify these plants. This image 1 shows the Euphorbia heta. Compared to image 1 and image 3 is dracunculoides, we can morphologically differentiate both the plants with their leaf structure. Similarly, other one, that is image 5, which shows Euphorbia hyperacifolia. This is also morphologically different. And image 7 that shows Euphorbia microphylla, that is also totally different plant. So morphologically we can differentiate this all the 4 plants, but I have taken here stem anatomy and compared the anatomy of all the 4 plants, and from that I can find that this all 4 plant stem anatomy is basically different. it does not shows more variation, but the stem shows secondary growth. And
Hello?
first of all, the texture, we can say that all the four the basic structure of the euphorbia stem, that they have hairy stem.
Hello?
But, hello?
Miss Ayushi?
Yes, sir.
Okay, you have 1 minute more, so please come to the conclusion.
Okay, okay, sir. So this is my conclusion. It is shows in a table form, sir. Wait. Okay. So we can conclude like that the shape of the section almost circular, but only hyperacifolia has such some ridges at some places. Epidermis is generally single layer found in all the 4 species. Hypodermis is also the same. We can find the lenticular cells present in this anatomy of the plant. Vascular bundles are also same, arranged in the ring-like structure. But pith is here somewhat different. We can say that some in 2 plants the pith is loosely arranged, whereas in 2 plants the cells are arranged medium sized and compactly arranged. The main conclusion of the study is to focus that the anatomy can be useful in Ayurvedic medicine. So it is possible to identify the dried raw drug sample of the species using these markers. Hence, anatomical studies are the reliable source to identify the genuine raw drug from their adulterant. So this is the main conclusion of my study. Thank you. Hello. Hello.
Hello, I'm Miss
Yes,
Purohit.
sir. Yes, sir.
Okay, so thank you for your demonstration. So this session is open for interaction. So I
Yes.
would like to ask the participants to if they have any questions.
Okay, sir.
Other participants can ask any question if they have any.
Okay, I have one small question.
Yes, sir.
Did you find any habitat forms of Euphorbia hitta? Are the different habitat forms, or have you studied any anatomical aspects of habitat forms?
Yes, sir. I can found the variation in the leaf structure. Actually, in one paper, I find that in Euphorbia hyperacifolia, there are no trichomes found. But in my study, when I have taken the plant, I found minute trichomes on it. That may be due to the habitat anatomical adaptations may have taken due to that. That I can say. Exactly, I don't know, sir.
And you can find the aspects of to find out adulterants and others.
Sir, from reviewing various papers, I have found that Euphorbia hitta and all the Euphorbia species are found in the ethnobotanical uses from previous years. So there are, we can say that this anatomy can be useful for adulterant plant. With the same appearance of the plant, sometimes we are misguided by the morphology of the plant. So if we take the anatomy, we can compare the stem anatomy or maybe root anatomy of any anatomical part of the plant. So in this way, we can compare and we can find out.
So I asked her just, have you done any histochemical study or just simple anatomy?
So it is simple anatomy.
Okay.
I have one question for you, Purohit.
Yes, sir.
So, this is the original work? Means this report was not published previously?
Yes, sir, it is original.
No report of this anatomical study has been done yet?
Sir, I cannot hear you properly.
The report of this anatomical work for this 4 plant haven't been done yet? So this is a new study?
Done, sir. Sir, it has been done, but not for all the 4 species. I have
all
found the work for Euphorbia hitta, but not found for Dracunculoides and other 3 plants, sir.
So it's a new report, right?
Yes, sir.
Okay, thank you.
Okay.
So Ayushi, I have a question. So other than anatomical study, do you think any other kind? Hello?
Hello?
Hello? Yes. Can you hear me?
Yes, yes. I can.
Ayushi, I have a question. Other than anatomy, do you think any other kind of studies can help you in this kind of study or in this kind of application, or in your objective? Other than
Maybe, ma'am, it
STEM
can be
anatomy, any other kind of?
it can be useful. Maybe we can go in a deep for we can say micromolecular anatomy and all that we can use for identification of the plant at a very small minute level.
Yes, because anatomy is not as much as helpful as other molecular studies, right?
Yes.
So you need to study, other than anatomy, you need to study any other kind of molecular
Biology,
things
yes, ma'am.
to see whether there are any differences there in the species or not. Huh?
Yes, ma'am. But for we can say that can be basically useful after morphology.
Yes, that's right. But only the anatomy will not be helpful.
Yes, I agree. Ma'am. Not only anatomy, but it can be one of the aspect which can be useful.
Yes.
So any other questions from the participants? Okay. Thank you, Ms. Purohit.
Yes.
Okay. So we will move towards our next participant. Our next participant is Ms. Akanksha Srivastava, Department of Advanced Study in Botany, Institute of Science, Banaras Hindu University. I request her to present her e-poster and Ms. Orpita Maiti to get herself ready for the next one. So Ms. Akanksha Srivastava, are you ready?
Yes, sir. Please give me a second.
Okay.
Can you all see my slides?
Yes, it is visible. So you can start your delivery.
Okay, so a very good afternoon to all of you. My name is Akansha Srivastava and I'm a research scholar in the Department of Botany, Banaras Hindu University. So first of all, I'd like to thank you to all the organizers of this webinar who has provided me the opportunity to present my work. And the work, what I'm going to present today is the title of the presentation is Ultrastructural Physiological and Thylakoid Membrane Proteome Analysis of Anabina for monitoring the photosynthetic responses under cadmium stress. So cadmium is a non-essential and toxic heavy metal which is commonly present into the water, atmosphere and soil. When cadmium accumulates inside the plant tissues, it can induce the serious phytotoxicity by interfering with several physiological processes, including photosynthesis. In higher plants and eukaryotic algae, it has been seen that the cadmium affects the photosynthesis at both the structural and functional level. However, in case of cyanobacteria, it is not known that how cadmium affects the photosynthesis and what adaptive strategy cyanobacteria adopt to protect their photosynthetic machinery under the cadmium stress. So in the light of this gap in the literature, we analyzed the photosynthetic response of Anabina under cadmium stress at ultrastructural physiological and thylakoid membrane proteome level via imaging, chlorophyll A fluorescence measurements and blue native S2 space respectively. So our findings showed that the cadmium exposure induced the ROS production inside the Anabina cell, and it also affected the thylakoid membrane organization, as we can see in the figure 1. Further, there was a question that if the thylakoid membrane organization are being affected under the cadmium stress, so the photosynthetic complexes which are present on the thylakoid membrane, their structure and the function must be changed. So to know the changes in the photosynthetic complexes, we conducted the chlorophyll A fluorescence measurements using the PAM fluorimetry. Chlorophyll A fluorescence measurement is a very sensitive and non-invasive technique which provides a lot of information about the structure, function, and the conformation of the photosynthetic complexes without distorting the sample. So our finding of the chlorophyll A fluorescence measurement showed that exposure of the cadmium inactivated the photosystem 2 reaction centers, which causes the decrease in the trapping of absorbed energy and increase in the heat dissipation. It also decreases the transfer of the electron from photosystem 2 to photosystem 1, which is also known as the linear electron flow. And decreasing the linear electron flow is a protective strategy adopted by Anabina under cadmium stress to prevent the over reduction of the photosynthetic electron transport chain, as well as also to prevent the photo inhibition of the photosystem 1. The recovery of the photosystem 1 is very slow than the photosystem 2. That's why photosynthetic organisms, they induces reduces the photo damage of the photosystem to prevent the photo emission of the photosystem 1. So similarly, it was happening in the Anabina 2. And at the same time, we observed that when the linear electron flow from photosystem 2 to photosystem 1 was inhibited, at the same time, the cyclic electron flow around photosystem 1 was operated to provide the photoprotection. So further to confirm our chlorophyll A fluorescence finding at the thylakoid membrane proteome level, we applied a very fascinating technology which is known as the blue native state space, where we separated the photosynthetic complexes first in their native form on the first dimension, and then in their respective subunits on the second dimension. So we observed that by comparing. Yes.
So Ms. Srivastava?
Yes, sir.
You have one more time, so please come to the conclusion.
Yes, sir, I'm doing. So by comparing the gel, we found that the most of the damage was happening into the photosystem 2 subunits, while the subunits of the other complexes which are required for the cyclic electron flow, they were quite stable. So again, the VNSDS page confirm our chlorophyll A fluorescence finding, and on the basis of our finding, we have made this model. So in nutshell, we can say that the shift into the cyclic electron flow from linear electron flow was a protective strategy adopted by Anabina to protect its photosynthetic machinery under the cadmium stress. So this paper has already been published into the algal research in 2021. Thank you all for the attention. Yes, sir. I have completed my presentation. If anybody can want to ask any question, then they can.
Hello.
Yes.
Ma'am, can you please elaborate your experimental setup? Means how much treatment cadmium stress you have given?
Yes,
just
I have taken the
single set
exponential
or?
Yeah. I have taken the exponential phase of the Anabina, which was approximately 0.5 OD at the 750 nanometer, and then I exposed the Anabina culture at the 10 micromolar concentration of the cadmium, which is the LC50 dose of for the Anabina.
Okay.
Yeah.
Okay. 10.
Is there any other question?
Do you have any other plans to study the cadmium stress other than this kind of study?
Sorry, ma'am, can you please repeat your question?
Do you have any kind of other plans to study cadmium stress?
Yes, ma'am. After that, what I'm doing, as I have studied the impact of the cadmium on the photosynthesis, and I have also assessed that how Anabina respond to the cadmium stress. So now our next planning is that I want to know that how these processes, how the responses of the Anabina are regulated at the transcription level. So I've just found that one transcription regulator, which is the LEXA, and we are in the process of confirming whether it regulates the photosynthetic responses of Anabina under the cadmium stress or not. But we have found that it has certain roles. So we are in the process of confirming that.
Okay.
Hello.
Yes, I guess it's quite satisfactory from your side. So our next participant is Orpita Maiti from Department of Center for Life Science, Vidyasagar University. Please share your slides.
Miss Urpita Maiti, are you there?
Yes, sir.
Okay. Please share your slide.
Hello.
Yes. It is visible. You can start.
Can you show? Hello.
Tell me.
Yes, it is visible.
Yeah, we can say it.
Sir,
But the size is quite small. Can you
good
please?
afternoon, everyone.
Yes. Can you please arrange the slide?
Actually, I am busy now on a training period, so I cannot carry my laptop.
Okay.
Now I carrying my cell phone.
So you can shift the slide from various points so that we can see the slides. Okay, you can.
Hello?
You can start now.
Good afternoon, everyone. I am Urpita Mahiti, research scholar Raman Kumar Mandal, Botany and Pathology, Vidyasagar University. Now I deliver my poster entitled Micromorphological Studies of some mangrove and mangrove associated plant, Orissa coast, India. Mangrove are divergent family of angiosperming plant. They are situated in high saline environmental condition. They are well developed in oxygen disturbing condition. Architecturally, parameter of mangrove leaves have a thick cuticle, sunken stomata, trichomes, salt gland, water storage tissue. So, distribution of mangrove actually Algae and Durgapur, located at Bhitorkonika, all various species in Abyssinia, Marina, Abyssinia, Rumphia, Silicea, also a associated plant, Exocaria, Agalosia, also associated. What is plant sample? Micromolecular. This is yes, light microscope in my department, and I conclude my psycho and thought at glad and also microscope as rotalia here is present only abaxial part, not on adaxial part, and salt gland
both at adaxial and abaxial site in the leaves. Plant Syria, Ipomia pescarpi, and Exocaria agalosa. And type diabetic parasite.
Hello.
Yes, sir.
You have only 1 minute more,
Sir?
so please come to the conclusion. Yep.
Agility, Eljogeria, and Ipomia pescarpi. Stomata and Psychom the plant leaf polia area I have done my
Okay, so I guess the demonstration is over. So we had lots of audio and visual problems in the presentation. So still, if anyone have any questions regarding her work, they can ask. Anyone have any question? Okay. Thank you, Miss Maiti.
Okay, sir.
Oh, so our next
Anushka,
speaker
you are
is
not audible. Okay.
okay. So our next participant is Mr. Chitranjan Noskar from Conservation Biology Department, Durgapur Government College, Durgapur. I will request him to present his e-poster and Miss Deepali Das to get herself ready for the next one. Mr. Chitranjan Noskar, are you ready?
Yes.
Okay, please deliver your lecture.
Is it visible and audible?
Yes, it is visible and you are clearly audible. Please start.
Okay. Good afternoon, everyone. And I myself, Chitranjan Noskar from Durgapur Government College, and I would like to present my e-poster here with the name Eco-Friendly Method of Strawberry Cultivation at Rooftop Garden. So the slide is divided into 3 parts: introduction, objective, and method. The strawberry is popular throughout the world not only for its characteristic aroma and attractive color, but also its nutritional and medicinal values. Like it is rich in vitamins, minerals, fibers, and antioxidants, and it has also anti-inflammatory properties. The antioxidants are polyphenols. The 2 types of polyphenols are L-arginine and ellagic acid. These 2 are proven that this can reduce the blood pressure and also the blood sugar level. And also the presence of anthocyanin that has linked with the lower risk of heart attacks. Quercetin had also anti-inflammatory properties and it can also resist atherosclerosis. So effect of different combination of potting mix on the fruit production, size, weight, and number of fruits. And decade ago, it was established that the strawberry is only cultivated in the hilly areas, but now the strawberry is state in any condition with special agricultural, modern agricultural techniques. So here I am trying to enter with the 7 potting mix. The 7 potting mix here, the 7 potting mix with the garden soil, chemical, vermicompost, coco peat, cow dung, and others, neem cake. So the table shows that the 6 potting mix, set 6, gives the best result regarding the length of fruit, breadth of fruit, number of fruit, and weight of fruit. And this is very much available, and this is very much convenient for the farmer for increased nutritional value of strawberries, and that will lead to the good earn for the farmer. And also the standardized method of cultivation will be more eco-friendly and economically viable than the contemporary method. And here I am showing some pictures. That is the one can, if anyone can grow strawberry, the two essential requirement is the soil preparation and the sapling production. And this is the very expensive method, sapling production. If anyone can learn the sapling from the stolon, so he can produce sapling in the very easiest way. And this is the strawberry flower, and this is the rooftop garden strawberry. And the sweet strawberry, these are the strawberries. These length, breadth, weight are measured. Yes, yes.
Hello, Mr. Noskar. You have 1 minute more, so please the conclusion.
Okay. Thank you. So I'll conclude my poster with these are the organic pesticide and organic fertilizer used strawberries. And here are the two news, 2018 in Evela newspaper. And this is the another news, Chadei Folche strawberry in 2019 Anandobajar Potrika. So I would like to express my heartfelt thanks to the organizer and all my teachers, and thank you very much.
Okay. Thank you so much, Mr. Noskar. So the session is open for interaction. So I would like to request the participants if they have any questions to ask.
So you have mentioned that strawberry has medicinal values. What kind of medicinal values does strawberry have?
It's a larger source of good vitamins, vitamin C, vitamin A, and also medicinal values reported in PubMed Central that it has the resistance against growth of cancer cells, and it can also resist the risk of heart attack, and it can also reduce the chance of atherosclerosis here.
Okay, thank you.
Okay, thank you.
Hello. You have shown one table regarding showing that the length of and width of fruits, and did you measure it statistically? Did you evaluate it statistically?
Yes. LSD value and CV value are given there.
Okay.
So any more
and
questions?
from that data, I will conclude that the potting mix 6 is very much essential and very much successfully cultivated in the field. And some of the farmers in Mednipur and Sundarban, they'll cultivate strawberry in their field with the organic fertilizer and organic pesticides also.
So I have a question for you. So what kind of organic fertilizer you are using for cultivating the strawberries?
Okay. Actually, I am giving in the table that the vermicompost that is homemade, and that is my preparation also. Homemade vermicompost, cow dung, neem cake, etc. These are the organic fertilizers. And after soil test, if the micronutrients are not sufficient level, so I have used some micronutrients. And the organic pesticides are trichoderma solution, and that is collected from NIMPI Ramakrishna Mission. And
okay, another one for you. Have you ever compared the nutritional value of traditionally grown strawberry with your pot-grown strawberry?
No, sir. Actually, this is the taste difference that I have done. Only the market-based strawberries that are not very much tasty because due to the excessive use of pesticides and chemical fertilizer. And the strawberries produced in my rooftop garden, and that is very much tasty. But I have not done the content analysis. Okay.
Okay, I will suggest you do a comparative chart rather. That's my point of view. Okay, to establish
And
your
and another question from my side, that how did you determine that how much amount of fertilizer you have to give? Means, have you standardized anything?
Yes, yes. Actually, in a potting mix, there is a different ratio. In my table,
Yes. How
there
have
is
you
a
determined
different
this
ratio
ratio?
is given here. No, no, this is the experimental process. This standardization taken me for 2 years. For the last 2 years, I have standardized the potting mix experimentally. This is not the one-time result. This is a 2 years experiment. One time I have used vermicompost. One time I have used neem cake. One time I have used the cow dung, and then
Okay.
last finally the mix up with the ratio, and the final result is here. Okay.
Okay, so any other questions from the participant?
Sandipon, Dr. Sandipon Ray.
Yes, sir. Yes, sir.
So, may I ask one question to my scholar?
Yes,
Definitely, sir.
Yes, sir.
Definitely.
Sometimes I forget
It's
botany,
my
always
pleasure.
busy with administrative work, but I think this is the high time. So, Chitranjan, what good question raised by Dr. Sandipan Ray, one my colleague. So, please try your level best to compare the nutritional value
Content analysis.
of
Okay.
the strawberry grown in other areas with the rooftop garden by using the another person, I think Mr. Shobik something, doctor. So this is the very pertinent question. So you should keep in mind that if you added this to one, first of all you have to compare the nutritional value of the strawberry grown in your rooftop garden. One thing, another is but using the organic fertilizer in different doses. So this is my please take this good suggestion and try to your level best to add your in experimental plot or whatever maybe. That will be
Sir, actually, due to okay,
no,
sir.
no, that
Sir,
is a
thank
different
you.
question.
Actually,
Please, no. Take their suggestion in a
yes, sir.
positive manner,
Definitely.
and you will get
Definitely,
better
sir.
result. Okay.
Definitely,
That is
sir.
my.
Actually, sir.
Okay.
Yes, sir. Actually, sir, due to pandemic situation, I have not contacted with the laboratories for the comparative
No.
or content analysis.
Okay, so,
Okay, sir.
pandemic will come again. That not the problems, but try to add this to suggestion in your work so that your work will be, I think, more precise and will very nice for present. Okay.
Okay. Thank you.
Thank you. Thank you very much, organizer, for giving me the opportunity to ask the question.
Thank you so much. Thank you so much, sir. So anyone have any question? Okay, so I think there is no question anymore. So thank you, Mr.
Okay.
Noskar. Oh,
Thank you, organizer, for giving me the opportunity for presenting my e-poster. Thank you very much.
Thank you. So our next participant is Miss Deepali Das, Conservation Biology Department, Durgapur Government College, Durgapur. I will request her to present her e-poster and Miss Jani Dhruva to get herself ready for the next one. Miss Deepali Das, are you ready?
Yes, sir.
Okay. Please
So just a
deliver
minute.
your.
Sir, is the slide is visible?
Yeah, it is visible.
Okay, sir. Okay, sir. I will start you.
Please enlarge more. Okay.
Okay, sir. Good afternoon, respected sir and ma'am, and all the participants. Myself, Deepali Das. I am 4th sem student, PG Department of Conservation Biology. Today, I am going to give a e-poster presentations on ethnomedicinal plants and uses and their tolerance level against air pollution. So, we all know medicinal plants means those herbs that have medicinal quality for curing various diseases, which are precursors for hemiparaceutical semisynthesis. And these plants plays a important role in the development of human cultures around the world. Secondly, going to discuss about its aims and objectives. Firstly, to identify the medicinal plants and their uses. Second, reasons for low uses of medicinal plants. And the third is to determine the APTI values of ethnomedicinal plant species. And the fourth is arise a questions about the conservation of traditional medicine in the highly polluted area in Durgapur. Thirdly, going to discuss about methodology. The study was conducted in 4 different villages of Durgapur and GPS was used to know the exact location. And after field survey, fresh leaf samples were collected and brought to the laboratory for analysis. According to the analysis, 4 different biological parameters was used: total chlorophyll content, ascorbic acid content, leaf extract pH, and relative water content. Based on the field survey method, 17 plant species was analyzed using 4 biochemical parameters. So, the result shows that Cesium cumini is the most tolerant species, and already we know medicinal plants are essential. So, it is concluded that, and you can see the result, you can see in this table in the Botany village is the fewer use medicinal plants as medicine, and the herbs and trees are widely used in the Muhuabagan village. And you can see in this figure some images of medicinal plants and their uses, but day by day the abundance of medicinal plants also decreases because vehicles and industries are much more increases. That's why pollutions are also decreases. This is a very big problem in over the world. So it was concluded that their knowledge and culture are being lost. According to this study, most of the plant species was found as sensitive. So that can be used as an indicator, and they should be kept in pollutions free zone. And those plants are tolerant that can be artificially cultured where the plants are increased. So the pollutions can be controlled. In this way, we can preserve their knowledge and increase cultivation. So this is some references I was used in this presentation. So this is all about my poster. Lastly, I want to thank the Department of Botany for organizing this poster presentation program, and also thank to all my teachers, seniors and friends to my Department of Conservation Biology from Durgapur Government College. Thank you.
Thank you so much, Miss Dipali Das.
Hey, Dipali.
Yes, sir.
So one question, just to read out your the title again.
Sir, ethno-medicinal knowledge and uses of medicinal plants and their tolerance level against air pollution.
Ethno-medicinal knowledge of whom? You want to control the air pollution by using the ethno-medicinal plants?
No, sir. I was found
ethno-medicinal knowledge of. Now, listen very carefully. Ethno-medicinal knowledge of some people. I think you should mention the place. No, ethno-medicinal means.
Ethno-medicinal means traditional.
The ethno-medicinal knowledge of whom? That means some people, either tribal people or in a specific area, and you want to control
all the people, sir. Tribal and all both the people. Yes, sir. Both, sir. Tribal.
No, you want to control the air pollution. You have mentioned the APTI such and such. So I have a little bit of idea. So you want to control the air pollution by using such ethno-medicinally important plants.
Sir, no, sir. Air pollution is not controlled the ethno-medicinal plants, but sir, I used APTI because sir,
how many plants?
Now, sir,
you have measured APTI value of how many plants?
Sir, 17.
17 in 2 villages.
Yes, sir.
And APTI value, you have measured the APTI value of those plants, those which have some medicinal values, isn't it? Hello?
Yes, sir. Yes, sir.
Hello?
Yes, sir. Sir, in the villages I was found 28 plants, but I was analyzed only 17 plants for the APTI.
And these 17 plants, they have the medicinal values?
Yes, sir. Sir, not possible in the 3 months. So, sir, I want
that's a different thing. Ethno-medicinal knowledge of whom people. That means some people they have the ethnomedicinal knowledge, okay? Either some ethnomedicinal used by some local tribal people or whatever people, not necessarily the tribal people, they have some knowledge, and you have used their knowledge that can be used to combat the air pollution in Durgapur industrial area. Is it correct or not?
Yes, sir. Yes, sir.
Okay. Thank you.
One thing, Deepali, hello, am I audible?
Yes, sir.
Yes, one thing actually, my question is why you are trying to correlate the ethnomedicinal plants, application of the ethnomedicinal plants in air pollution. Actually,
Yes, sir. Two aspects is different, I think, no?
yes, actually, you are trying to correlate the ethnomedicinal. Why you have selected the ethnomedicinal plants for air pollution controlling? Actually, my question is that Principal Sir is also asking the same thing. I think that the two are the different aspects. One is the ethnomedicinal value. Another one is the actually. I'll be very glad that do you know that is there any APTI value high index or measure some implications of some plants which are ethnomedicine in nature, but that those plants have also the very great value for the air pollution like that. I actually that I can't understand. I'm very
sir.
Yes.
Okay, sir. I will explain. Sir, day by day, allopathic medicines are much more available. So, medicinal, I'm studying because, sir, very low few amounts of medicinal plants I can found. So, sir, I understand. I want to. So those plants I have. If we eat, I mean, if we eat those plants, I mean medicinal plants, how much tolerance level is present? How much tolerance level is present in the medicinal plants, and if we eat those plants, the pollutants go into the body. So how much affected? So, sir,
no, actually, I think that you are. Is it? Am I correct that you are trying to correlate because the ethnomedicinal plants are very much acceptable and these are very much easily grown in different areas, so that the people can get very easy accession for those plants, and as such, the plants number of the plants that means the frequency of the plants will be very high, and in that sense, the pollution could be controlled. Are you trying to approach this thing?
Yes, sir. But I am just using because, sir, I want to gather the tribal knowledge, and when we eat humans, medicinal plants eat, those plants are sensitive or tolerant. That's why I am using this.
Okay. It is nicely presented, but the aspects are quite different. Okay, but your effort is very well accepted, and it is very nice. Thank you, Deepali. Okay.
Thank you, sir.
Okay, so any more questions from the participants? Okay, I guess there are no such questions. So thank you, Ms. Deepali.
Thank you, sir.
So our next participant is Ms. Jani Dhruva, Department of Botany, Gujarat University, Ahmedabad. I request her to present her e-poster and Dr. T. Sri Vijaya Kiruva to get herself ready for the next one. Ms. Jani Dhruva, are you ready?
Yes, sir. Is the poster visible?
Yes, it is visible.
Good afternoon to one and all. I am Dhruva Jani, and my topic for presentation is phytosociological attributes as a tool for forest restoration, a comparative study of tree species from 2 talukas of Valsad district, Gujarat. As we know, the plant communities are constantly evolving. The present study mainly focuses on the quantitative evaluation of tree species from forest areas of Dharampur and Kaprada, which are the talukas of Valsad district. The major objective of the study is to carry out the assessment of tree species and to understand their distribution status. For the methodology, the whole community analysis can be broadly categorized into 3 parts. The first part was to gather the FSI 2014 map, and by stratified random sampling of 0.01%, 33 distinct plots were identified. From this 33 plots, 15 plots belonged to Dharampur and 18 plots were from Kaprada. All this 33 plots, quadrant analysis, both by listing and counting the number of species were done, and the quadrant size taken was 15 to 50 square meters. For the part 2, the documented data was then analyzed by phytosociological attributes and biodiversity index was calculated. In the phytosociological attributes, density, abundance, frequency, and the IVI was calculated. And in the last part, we also compared our field data with the satellite data of 5 years from year 2016 to 2021, and that was an NDVI data source. From this work, we found certain preliminary results which are shown in the first and the second graph. The first graph shows the NDVI value of both the talukas. We found that both the talukas had NDVI value from 0.5 to 0.7, which was near to 1, and the Kaprada taluka showed a higher NDVI value compared to Dharampur. In the phytosociological study, the highest frequency and other different values was found to be the dominated plant was Tectona grandis, followed by Terminalia elliptica, and the lowest two trees which had very less value in the quadrant analysis was Fimriana cordata and Semicarpus anacardium. In the biodiversity index, we found that the Simpson's dominance index was near 1 in both the talukas, and even the Shannon's biodiversity index was greater than 1, which indicated a good and rich vegetation. But it was found that the Kaprada forest had more species richness compared to the Dharampur taluka. So finally, the conclusion states that the main reason in which the Kaprada taluka had higher species richness is due to its topography. It is a hilly region and has less human interactions. On the other hand, the Dharampur taluka, the forest of the Dharampur taluka is converted into a crop field, a horticulture crop field, and there is more industrialization seen in such areas. And for this reason, the landscape has changed. From this analysis, we can conclude that the assessment of floristic diversity at local and regional levels can help in planning the conservation and restoration strategies for rare and endangered species, or the species which are lower in richness, and the species evenness of the forest can be the species richness of the forest can be increased rather than just cultivating monoculture trees in an area. I conclude my presentation and thank you for the platform for sharing my work.
Okay. Thank you, Mrs. Rubajani. So the session is open for interaction. So I would like to ask the participants if they have any questions. Do you have any questions? Okay, so I guess nobody have any questions now. So thank you, Ms. Dhrubajani.
Thank you
for presenting.
Okay.
Okay. So our next participant is Dr. P. Sri Vijaya Kiruva, Department of Pharmacognosy, Periyar College of Pharmaceutical Sciences, Tiruchirappalli. I will request Dr. T. Sri Vijaya Kiruva to present her e-poster and Miss Jyotirekha Chakraborty to get herself ready for the next one. So Dr. T. Sri Vijaya Kiruva, are you ready? Dr. Kiruva, are you ready? Hello, am I audible?
Yes, you're audible, Anish.
Okay. Dr. Kiruva, are you ready?
He's present in the audience, but I'm not sure he can listen to you or not. Okay.
Sir, am I audible, sir? Right now,
Yes,
am I
yes, yes, yes.
audible?
You're audible. You're audible.
So, is my screen visible, sir? Shall I start?
Yeah, definitely. Start.
Yeah. Good afternoon, one and all gathered here virtually. The topic of my presentation is phytomedicine, an emerging hub for new drugs from botanicals. Here, phytopharmaceuticals, also called as the phytomedicine, it is a complex mixture that has been derived from variety of plant sources being used in the form of medicines or what we call as drugs. They make up the good part of drugs, which are basically from the plant-based molecules, and they play a major role in maintaining a healthy individual in all his body functions. There is going to be a higher demands for the clinically proven phytopharmaceuticals. And in recent days, the field of nanotechnology plays a major role in all the areas. And when we talk about medicines, yes, in medicine also, nanomedicine plays a major role. And the botanicals, their field is also concentrated towards this nanomedicine. So nanotechnology, plant actives and extracts, nowadays there is a tremendous growth among the individuals, and especially the scientists are turning their attention towards this herbal nanomedicine. What are the perspectives of phytopharmaceuticals? When we look globally, the herbal medicines, they are considered to be an important alternative to the English medicine, that is the modern allopathic medicine. So this is one of the recent report from the US FDA, a new drug called Verigen, that is a polyphenol E ointment. It is considered as the first prescription botanical, that a herbal drug approved by US FDA under the New Drug Amendments Act 1962, and it is marketed in the US. And some other standardized extracts, namely Comifera, Ginkgo, Silybum, Marianum, Gingiber officinae, they are all quite good examples that represents the phytopharmaceutical class of drugs that are most commonly available in the Indian market. And there is the public, they are focusing their attention towards this phytopharmaceuticals. So this pictorial representation, I have just given you how the herbal drugs, in what forms they are used. They are used in the form of leaves, flowers, stems, root, barks, entire plant, juice, likewise. And when we are talking about this nanomedicine, they are used in the different novel medicines like phytosomes, neosomes, ethosomes and all, and they are used for treating the various parts of the ailments that occurs in different parts of the body. And here, this is one typical example. The common drug curcuma,
Hello.
turmeric. Yes, sir.
Hello, Dr. Kiruva.
Yes, sir.
Hi, you have 1 minute more, so please come to the conclusion.
Sure, sir, sure. So curcuma is encapsulated in the form of phytosome, and it is fried, and all know that it has antioxidant property, and it is right for myocardial infarction treatment. And here, the drugs from botanicals, when they made a survey during the year 1981 to 2002, it has been seen that 28% of all the new chemical entities are from the plant-derived drugs. And especially in the field of cancer therapy,
Out of 175 drug molecules, 85 were isolated from the plant sources. And it is estimated that in the future, that is around by 2050, the annual growth rate will be up to 5 trillion. So thereby, also we can see that the drugs, the Indian medicinal plants especially, is being included in many of the pharmacopoeias, considering the importance of the phytopharmaceuticals. So I conclude by saying that the global trends and opportunities are more for the plant-based medicines, especially phytopharmaceuticals. Thereby, the government of India has also included this under the new drug category, and especially nanophytomedicine is a new hub for the herbal industries, especially for the pharma industry, I can say. So I conclude that in near future, the phytomedicine and phytopharmaceuticals will be the driving force of the herbal industry. So I wish to say that apart from the traditional medicine, Ayurveda, Siddha, Unani and all, this particular field, that is phytomedicine, nanomedicine, will be an emerging hub for pharmaceutical industries. Thank you.
Thank you, Dr. Kiruba. So the session is open for discussion. So anybody have any question from the participants?
So, Doctor, yes, please ask your question.
Okay. Dr. Kiruba, I have, yes, actually not the question. I have a little discussion with you. As you are, it is very interesting, and you have presented the paper very well. And as you are from the branch of the pharmaceutical science, that's why, being a botanist, I have some little discussion with you. The thing is that we are always finding many wild plants surrounding us, and among these many wild plants, we know that each and every plant have to some extent the little medicinal value, but sometimes it has the great medicinal value. But I see that the pharmaceutical sciences always emphasizes the work on some already known, which are the very known highly medicinal value plants like this. Say for example, we see the plants of the Tephrosia purpurea or the Lantana camera. Those are actually wild plants, and these are the wild. Although it is the wild invasive plants, but they have the medicinal value also. So you know that the pharmaceutical sciences and the people from the pharmaceutical branches, whether they are giving any emphasis to isolate some drugs from those wild plants which are very easily grown surrounding us.
Of course, sir. Sir, as you very well said, many wild plants are having more medicinal values, as you rightly told. But as a pharmacy people, we are targeting those wild plants also as a part of our project work. When you look into the research projects, we also concentrate not only on the commonly used plants, but also we go in search of some of the wild plants, as you told, that are having medicinal value. We go for isolation every year or periodically. Work is being carried out for search of new molecules from the wild plants that are already available. So work is going on, sir.
Okay. Thank you so much.
Thank you, sir.
Okay. So next, other, please ask the question. Other participants.
So, Dr. Kiruba, I have a question. Have you done any kind of work in this field?
Yes, ma'am. Actually, our field, we belong to the Department of Pharmacognosy, that is herbal pharmacy. So we just developed some novel dosage forms. We have tried for one type of dosage form called as transdermal patches for the treatment of arthritis. And presently, we are working on phytosomes, one of the novel medicine, nanomedicine. We are in, the work is going on, ma'am. So only I have just selected this topic for my presentation. So we are working on it.
Okay.
Okay, ma'am. Thank you.
So I have a question for Dr. Kiruba.
Yes.
So actually recently I came to hear that the Indian government is actually planning for a selected dose of cannabis but to be marketed, right? in foreign country, it's already a medicine. It's already a drug, but a prescribed one. So what is the scenario right now in India for making this cannabis as a drug? Because they are very useful. They have good medicinal values.
Yes, sir. It is having good medicinal value, but unfortunately, due to the other negative impact, since it is a narcotic drug, government is still working on it, sir. So, as compared to foreign countries, we are not that much gone for its medicinal value because there is some inhibitions.
Oh, okay. Thank you.
Yes, sir. Thank you, sir.
So, any other questions from other participants? So, I guess there are no such questions.
Okay, sir.
Thank you, Dr. Kiruba.
Thank you, sir. Thank you so much.
So, our next participant is Miss Jyoti Rekha Chakraborty, Department of Botany, Shiksha Bhavan, Vishwabharati. I will request Miss Jyoti Rekha Chakraborty to present the e-poster and Mrs. Madhushri Das Datto to get herself ready for the next one. Miss Jyoti Rekha Chakraborty, are you ready?
Yes, good afternoon everyone. Can you hear me?
Okay. Yeah, you're audible and visible too.
Yeah, okay. Just a second. Okay, is it visible, my poster?
Yes. Clearly visible.
Okay, thank you. So, good afternoon, everyone. So, I am Jyoti Rekha Chakraborty. I have been conducting a study in Murshidabad district of West Bengal to understand the present status of vegetable cultivation in the district. So, West Bengal is one of the leading vegetable growing states in India, and it contributes about 15% of the total production of vegetables. So Murshidabad district, it is considered as the crop museum of West Bengal because it represents almost all types of crops cultivated in India. So River Bhagirathi, this is the map of West Bengal, Murshidabad district. So River Bhagirathi divides the district into two parts. Eastern part is composed of alluvial and fertile soil called the Bagri, and western part of the district is having stiff clay soil, and this part is known as Rar. So generally Bagri area, it is more favorable for the cultivation of different kind of crops. So my objective and my study, it includes the descriptive research methodology, and I have conducted my study to analyze the cost profit structure of like 10 different popular vegetables in the district of Murshidabad district, grown in the Murshidabad district. And also I have tried to understand the constraints of the vegetable cultivation in the district. So I have adopted the method which I have adopted for my study is the multi-stage random sampling method, and 16 blocks of the district was surveyed, and total 480 numbers of farmers were interviewed for the present study. And I have used like a questionnaire which is standardized, and I also have informants were giving me information time to time, particularly during the pandemic situation. They have given me, they were the local people, and they give me information, and through WhatsApp, through phone calls and regular visits of the field, I have collected my data. And then I have analyzed my data, and I have found that like there were so many vegetables were there, but these 480 farmers which I have interviewed, so they have grown like I have selected 10 top 10 vegetables according to the number of farmers grown the particular type of vegetable. And then I have also analyzed the investment and net profit against the particular type of vegetable out of those 10 vegetables grown in the district. So then let's come to my result directly. So here we have seen like the total land used by the interviewed farmers, where 53% of the land is used for cultivating the vegetable crops. And generally the farmers which are cultivating the vegetable crops, they are small or marginal farmers. And then the maximum number of interviewed farmers, respondents, they have cultivated brinjal, number of farmers 432, followed by pointed gourd 354, and then pumpkin 339. So I have found that brinjal and pointed gourd, they are like perennial plants. So if one time it is grown, it will last for at least 5-6 years. So the farmers, they need not have to think about the initial investment which has to be done at the beginning to begin a particular vegetable. So that is why those are the popular vegetables. And also pumpkin, regarding pumpkin, most of the farmers, they have revealed that the pumpkin, like growing pumpkin, is like a bonus for them. Like potato, it is one of the major crop plants in the district, and after cultivating potato, they can easily cultivate the pumpkin in that same land without plowing is not needed in that case. Okay, so this is another inference I have got from my experience, from my survey. And then when we consider the investment and net profit, we have found that cauliflower is the, hello, can you hear my voice?
Yeah, you have just 30 seconds more. So can you please come to
Okay. Cauliflower and cabbage are the most profitable vegetable, and high labor price is found to be the constraint of the vegetable cultivation in the district. And we can easily increase the profit of the farmer by proper marketing facility, and also by making them aware about the organic farming or the latest improvement in the technology. Thank you so much.
Thank you, Ms. Jyotireka Chakraborty. So the session is open for interaction. Anyone have any question?
Yes. Am I audible?
Yes, sir.
Yes,
I can hear you.
Yes. Thank you, Dr. Jyotireka, for the nice presentation.
I am not, excuse me, sir, I am not doctor, I am pursuing my PhD.
Okay. You are the research scholar, so you would be the doctor. Okay. Thank you very much for your nice presentation. I have one question. Actually, I have seen your poster, and that you have selected the 10 different types of the vegetables like brinjal, cauliflower, tomato, etc.
Right. Yeah.
One thing is that so far we know that the production, that means the yield, it is greatly correlated with the different variety. Because as a botanist, I like to know, say for example, if you take altogether consideration of the brinjal production in Murshidabad district, then it is not the realistic study, because the different varieties of the cauliflower, different varieties of the brinjal, and in that case, if you consider the particular variety in the particular area during your transaction study, that means the transaction study or the random study that you have followed, what sorts of the different varieties are there, and that is the important thing. Another one important thing is that as it is the agricultural science is also related to the biology, that's why you mentioned the scientific name, that means the hybrid name of the each plants. Of course, there are some procedure of the nomenclature for the hybrid vegetables, and but if you include the scientific names and the variety of the particular species, particular vegetable, then it would be more feasible, and it would be rather very good, I think. This is my suggestion only, not any questions. Your presentation of the work is very interesting. It is very simple, but the worthwhile work. Okay, thank you. Okay.
Thank you, sir. Actually, I would like to add here, actually, I am working with the varieties right now, which are the most popular varieties. I have enlisted also, but here time is very less, and in the poster it is difficult to present. But I will definitely, carry out according to your suggestions, sir. Thank you so much. Thank you, sir.
Okay.
Any more questions from the participants? Any more questions from the participant? Yes, there are no such a question. Thank you, Ms. Chakraborty.
Okay, thank you so much. Thank you.
So our next participant is Mrs. Madhushri Das Dotto, Department of Botany, Hiralal Majumdar Memorial College for Women, Dakhineshwar, Kolkata. I request Mrs. Madhushri Das Dotto to present the e-poster and Ms. Monali Bishwas to get herself ready for the next one. Mrs. Madhushri Das Dotto, are you ready?
Yes, I'm sharing my screen.
Okay, okay.
I hope my screen is visible and I'm audible.
Yeah, yeah. It's clear.
Good afternoon to all of you. Today I'm going to speak on the cypsellar features and their taxonomic significance in two species of the tribe Arctotridae. I worked on this project under the able guidance of Professor Shobhan Kumar Mukherjee from the Department of Botany, University of Kalyani. The main objective of the project was to mainly to see what are the various detailed external and internal features of cypseli, the fruit of Asteraceae basically, in two species, that is Barkia multijuga and Barkia purpurea. Both are economically important plants under the genus Barkia and clade Barkia of the tribe Arctotridae of the family Asteraceae. We hoped at the initial phase that we would get some information ultimately which lead to a better evaluation of the family. And cypsellar features, as we all know, have a potential value for characterization of the taxa, as these reproductive characters are less variable in different environmental conditions, and they play a major role for identification of taxa when other floral features are not in hand or not available. We also know that the flowers or the inflorescence of the family Compositae or Asteraceae are more or less look the same. Sometimes they are called dam yellow composite by many authors because numerous species of this family shows the same color of yellow color of flower. As these characters of cypsella are most important tool for the success of the family Asteraceae, the diversity, evolutionary diversity of the family can be predicted by knowing these characters. So initially the investigated plant material were obtained in the form of herbarium specimen from the Botanical Gardens of the University of Zurich for morphological observation. 10 dry and 10 FAA preserved mature cypseli were taken, dipped in 1 to 5% NaOH solution for 2 to 7 days and stained with 0.1% aqueous safranin and observed under microscope. For anatomical observation, thin transverse sections were done and with the sharp razor blade, and the sections were immersed in 0.2 to 0.5% aqueous safranin solution for staining and mounted by 70% phenol glycerine solution on slides and placed under compound microscope for further study. And for SEM analysis, a minimum of 3 cypsella were mounted in the stubs and coated with very thin layer of gold. Scanning photomicrographs were taken in University Instrumentation Center, Kalyani. Basic terminology used from the Kinclova 1970 and few were adopted by ourselves. I'm coming into the, I'm just jump into the results and discussion part as there is time constraint. Both the cypsella were homomorphic in nature, bright brown in color. There are minor differences because both of the species belong to the same genus. All the characters more or less were similar, but the surface texture in case of Barkia multijuga was asperous, that is rough surface is there, and in case of Barkia purpurea the surface texture is glaucous, that is covered with bloom or slightly waxy coating was there. The rib is in number, Stylopodium is present, Carpopodium is symmetrical in both the cases, but in case of Barkia multijuga, the lobed ring-like structure is present, whereas in case of Barkia purpurea, circular complete ring-like structure is seen, and the arrangement of the cells were in 11 to 12 rows. Smaller cells were present in case of Barkia multijuga, whereas in case of Barkia purpurea, the arrangement is 3 to 4 rows. Large cells were present and square to rectangle in shape. In case of anatomical characters, both the Cypsela shown more or less the same character, but in epicarpic region, the cell arrangement were in case of multijuga, the species multijuga, the epicarpic layer was single layered, whereas in case of Barkia purpurea, it is double layered. Mesocarp was made up of parenchyma, sclerenchyma and vascular tissues. Testa was present. Endosperm was also present, and in case of in both the species, embryo was also present. So lastly, we would like to conclude that in the systematic point of view, the morphology and anatomy of Cypsela are of greater importance, and these features may be employed for better understanding of the taxa. You can definitely see the pictures or the hand drawings which were presented in the poster, and there are some photographic plates also there, and the camera photograph were also there with the scales. Also, the analysis evidently indicates that in comparison to size and shape of Cypselae, nature of the Carpopodium, presence or absence of ribs, pappus features like arrangement of pappus bristles, types of cells present in the mesocarpic region, number of resin ducts present in the cotyledon, these are all important characters which are helpful for grouping or separation of different species. Apart from taxonomists, these characters are also important for the agriculturist as well as pharmacologist for identification of weeds or detection of adulterants. Lastly, we would like to thank Professor Dr. Peter Enz, curator, and all the gardeners of the botanical garden of the University of Zurich for providing us the mature and identified Cypselae for the study. We are grateful to the in-charge University Instrumentation Center Kalyani for SEM photography, and thank you all for your patience hearing.
Okay, thank you. So the session is open for discussion. Participants have any questions?
Okay.
Hello.
Hello.
Yes, sir.
You are audible.
Am I audible?
Yes, sir.
Am I audible?
Yes, sir. Yes, sir.
Yes. One thing, madam, is good work you presented in the particular field of the plant taxonomy. Very good, and very good, and the work is also very good. So one thing, just as a botanist, I would like to know that this type of the work is very important one in the sense that the taxonomic marker. This type of the study is one of the taxonomic marker on the basis of the systematic disputes regarding the taxonomic disputes of the distributed position in spot. And like this marker, other markers are also there. The seed surface study, surface structure, pollen surface study. So I am happy to know if we can confirm that whether the Cypsela morphology or pollen study or seed surface, all these are the taxonomic tools actually. All the among these three, which one is the most important? Say for example, if you don't consider the Cypsela study, if you consider the pollen study or the seed surface study, you would be more better for solving of the taxonomic or the position of the species, or it is the more convenient, the Cypsela study is the best. That I like to know.
Sir, I actually missed the last part of your query. I mean,
yes.
last part of your query, I missed that part. Can you repeat?
Yes, ma'am. No problem.
The last part of your query. I mean, it is not audible from my side.
Yes.
Sir, actually,
last part, yes. Last part is that the final part is that actually the thing is that who is the best? The seed study or pollen?
Hello. Sir,
voice is not clear.
Sir, your voice is not clear, sir.
Clear, madam?
Hello.
Yes.
Sir, आवाज तो केटे यार sir. Your voice is not clear, sir.
Voice not clear?
Yes, sir. Voice not clear.
এখন শোনা যাচ্ছে?
স্যার শোনা যাচ্ছে। হ্যাঁ স্যার।
Yeah, it is better than before, but plenty disturbances are there.
Sir, আওয়াজ কেটে যাচ্ছে, কিছু বোঝা যাচ্ছে না।
ও আচ্ছা ঠিক আছে, তাহলে নেটের ঠিক আছে ছেড়ে দিন। ওকে, পরে আমি ম্যাডামের সাথে আলোচনা করে নেব। ওকে, আই উইল ডিসকাস লেটার অন। ওকে।
Actually, I'm asking that whether the pollen study or Shipsela, which one is better? I think he was asking.
Pollen, seed and Shipsela. Which one is better?
Actually, I can't judge which one is better because I have worked with Shipsela. I haven't worked anything with the pollen. But one thing is there that as I was saying that whenever the flowering stage is not available, because and more or less all the flowers look alike, in many cases, in case of the family Asteraceae. So in those cases, if Shipsela character could be studied, or we can at least make all those Shipsela character available, then we can easily separate all those species. That was what.
Yes. Thank you, madam. Thank you very much. Okay. It is very nice presentation. Okay.
Thank you, sir.
Any other questions from the participants? Any other questions from the participants? Okay, I guess there is no question. Thank you, Mrs. Datta.
Thank you.
So our next participant is you can mute your audio.
Yes.
So our next participant is Ms. Morali Vishwas, Department of Conservation Biology, Durgapur Government College. I request Ms. Morali Vishwas to present the e-poster, and the next participant, Ms. Prashina Rai, to get herself ready for the next one. Ms. Morali Vishwas, are you ready?
Yes, sir.
Please present your poster.
So is it visible?
No, it is not visible now.
Okay, sir. Sir, 1 minute. Now is it visible, sir?
Yes. It is visible.
Yes, visible.
Please go on.
Good afternoon, everybody. I'm Morali Vishwas, a research scholar from Department of Conservation Biology. So today my topic is survey of the macrophytes in Bokreshwar Reservoir, West Bengal, India. So first, wetlands. These are very unique creation in the world, which are covering aquatic, marshy and terrestrial habitat, are also considered as ecotonal habitat. These clearly show their capacity to hold a rich biodiversity that support aquatic, marsh and land vegetation also. Bokreshwar Reservoir is a very popular wetland in West Bengal, which is built on Bokreshwar River
built on Bokreshwar River, and a botanical survey on aquatic vegetation at Bokreshwar Reservoir was carried out during 2020 to 2022, as any detailed study on Bokreshwar Reservoir was not done previously. So my present study area is the Bokreshwar Reservoir or in Birbhum district of West Bengal. It was constructed on river Bokreshwar as a backup water supply for the Bokreshwar thermal power station under the supervision of West Bengal Power Development Corporation in 1999. But it supports a great vegetation, macrophytes, microphytes, etc. So my methodology, like random survey was made throughout the reservoir during the year 2020 to 2022. Immersed, submerged, floating, marginal plants are identified, and all plants are identified through the literature by following the literature of Koch 1996, Bora 2003, etc. So now coming to the results. So due to the present survey work, almost 112 species of aquatic macrophytes are recorded here, like Utricularia, Salvinia, Otelia, Najas, Nymphoides, etc. These all macrophytes are very, very important for the environment. Here, from the pie chart, we can conclude it, or we can see that which plants are seasonally dominant or not. So from this present study, we can be concluded that this wetland is very, very rich in biodiversity. The floristic diversity as well as faunal diversity of Bokreshwar Reservoir is very rich. Life from distribution and habit group distribution presenting the present biological spectrum here. But the main thing is the reservoir facing various type of threats, mainly from the modern agricultural practice and pesticides application, with creating threats to fishes.
Please go.
No problem. It will be very nice, Dr. Saha. If you display, okay. If you present. Okay, so dear participant, you can follow the one of our teachers. He will present your slide. Okay.
Yes, sir. Yes.
Okay.
Is it visible?
It is visible. Very good.
Yes, sir,
carry on. Carry on.
Please tell me the appropriate means magnification that you desire. Is it okay?
Yes, sir, it's okay. It's
fine.
Yes, a very good afternoon, everyone. I am Priscilla Rai, and I'm currently working as a research scholar at the University of Calcutta Botany Department. And as we can see on the screen, our topic today is variation in vascular epiphyte assemblies along altitudinal zone in temperate forest ecosystem. Vascular epiphytes are an essential component of our natural ecosystem, and it can be defined as social, non-parasitic organisms that attach themselves to and grow on the other plant, but on contrary with the parasitic plant, they do not extract nutrients from the host tree. Although the temperate forest has a significant role in species diversity, knowledge about the distribution and ecological importance of vascular epiphytes in this forest system are insufficient. Sir, can you please scroll down?
Yeah, sure.
Yeah, methodology section. The main objective of my study is to investigate overall vascular epiphytic diversity and distributional pattern in temperate forest ecosystem. We also analyze the epiphytic species richness and abundance along an altitudinal gradient with focus on the basic aspects including host tree specificity and climatic variables. For this study, several field investigations were made in different sites of temperate forest in Darjeeling, Himalaya, and the study area were demarcated into 4 sites, that is Site 1, Site 2, and Site 3. The bioclimatic variables were obtained from the wall climb 2.0. So can you please show me the visual section?
Sure.
A total of 111 species of vascular epiphytes belonging to 51 genera and 25 families were recorded, among which orchids were the most diverse with 44 species, followed by other angiosperms, that is 34 species, and wild 33 species were found. Orchidaceae was the most abundant and the diverse family, which was followed by Polypoidaceae with 19 species. The frequently occurring species were Bulbophyllum, Coelogyne, and Dendrobium, whereas the dominating other Angiosperm comprises the species of Aschinanthus, Hoya, and Agapetes, while Siligua, Lepisaurus, and Haplopteris were some of the dominant epiphytic forms. As we can see in the figure 3, the species richness shows the monotonous pattern, that is, it declined as we go, it declined with increase in the altitude. In both respect, other angiosperm ferns and orchids also decreases with altitude, and the number of families, genus, and species were also decreased with increase in an altitude. So some of the dominant host trees were also studied during the field surveys. Here we can see on the screen I have mentioned some dominant species. So can you please scroll down for me? Okay. The distribution within the host tree, we analyze the distribution of vascular epiphytes within the host tree and their correlation with the CBH and the bark pH. We found the epiphytic species richness were positively correlated with the CBH of the host tree, and it was slightly significant, or it is slightly correlated with the bark pH of the host tree. And to understand the influence of the microclimatic variables on the epiphytic diversity, we performed the CCA, and we came to a conclusion that many
That's
environmental.
right.
Yes,
You
sir.
have 1 minute left, Theo. Okay.
Sure, sir. Yes, sir. We found that there are the influence of microclimatic variables in epiphytic species richness and abundance. I would like to conclude here my poster saying that the study reflects a high diversity of vascular epiphytes and variation in species richness, abundance, and species composition along different elevation gradient. The influence of the environmental parameters in different altitudinal zones tends the vascular epiphyte to be established well in warm and humid conditions. Species composition is strongly influenced by host tree traits. The occurrence of the rich diversity, including several RET species in the study area, has contributed to the conservation value and trait status of different taxa in the temperate forest of Darjeeling, Himalaya. The conservation of such a rich diversity is of the paramount importance. Thank you so much.
Yeah, thank you, Ms. Rai. Now the session is open for question. Any participant, if you want to ask Ms. Rai anything, you can put forward.
Yes. Thank you very much. Prashina, so far I remember, are you the ex-student of Darjeeling Government College?
Yes, sir. Yes, sir.
Yes, that's why I'm correct right now. That you are the ex-student of Darjeeling Government College, and you are doing the PhD under the Dr. Muktan, Saurabh Muktan.
Yes, sir. Yes, sir.
Yes, very good, Prashina. Myself is the AB chair. Okay.
Yes, sir. Yes, sir. I have recognized you.
Yes. So when you took your admission at that year, just after taking your admission, I left the Darjeeling Government College.
Yes, sir. I remember that.
Yes. So it is very good work and the good presentation. You presented the work is very nicely. You presented the work, and the work is very good and interesting one. You have covered so many things within your work. Just I like to know that as you are the ex-student of the Darjeeling Government College, surrounding the Darjeeling Government College, in which side the Agapetes is found?
Agapetes is found in the Hooker Road mainly, and around.
Good. I understand. You have seen it. Okay. In Hooker Road mainly, that just behind the Darjeeling Government College, the Agapetes.
Yes, sir. That is Agapetes serpens is most common in that area.
Yes, and at what time it is flowering?
So it usually flowers when I last visited. It was in the flowering stage, and it was the month of March. I remember.
Yes. Okay, I understand. Okay, so you have seen physically.
Yes, sir. Yes, sir. Yes, sir.
I'm sure that you have seen physically where the agapetes is growing in your host institute. Okay, thank you, Priscilla. Carry on. Okay.
Thank you,
Okay.
sir.
Congratulations. Thank you. Okay.
Okay, Priscilla, I have also one question
Yes, sir.
regarding the sampling process.
Yes,
From
sir.
each site, how
Yes, sir.
many plants you have studied? Have you made any quadrate in each site?
Actually, sir, I
Means
have done the yes,
I think you have studied the many tree species,
yes,
many
sir.
trees, and
Yes.
from each site, suppose one single plant was studied, 3 to 4
Yes.
single plants, and from another site. So I am just asking about the sampling procedure. From
Okay,
each
okay,
site, how you have sampled or how you have selected the trees.
okay, sir. Actually, I have done the stratified random sampling, but as you have asked, for the site one, I have just enlisted the dominant host trees, and I have segregated or like just simply seeing, not exactly doing the quadrat method, but because due to the topographic like variation, so it is difficult to do a quadrat in some of the forests. So I have just counted the individual trees so that I made it equal, so equal individual number of trees, so that it will be easy, or it will be like it will be easy for me to do the statistical analysis. So for like this, I have done my sampling, because doing quadrat is in some field, in some forests it is possible, but there are some difficult areas which was not possible for me to do the quadrat. So I have just counted the individual number of species, and
Okay.
host species.
Just individual number of species. Okay, okay.
Yeah.
And what are the microclimatic variables you have
Yeah.
studied?
I have actually produced all the from Bio 1 to Bio 19 microclimatic variables from the wall climb 2.0. But for this study, I have just correlated the precipitation, temperature, and relative humidity.
Okay.
Any more questions from the participant? Okay, thank you, Prasanna. So
Thank you,
we'll
sir.
move to next candidate, next participant, Ms. Riya Dotto from Center for Life Science, Vidyasagar University, and Mr. P. Santhanan Pandey. Please get prepared for your presentation. So, Ms. Riya Datta, can you please share your slide? Ms. Riya Datta.
Yes, I'm here. I'm trying to share my screen. Sorry, I'm not able to share my screen right now.
You were not able to share it?
Yes.
Can I share then?
Sure. Yeah.
Okay, please honest representative, can you please?
Yep, yep, definitely. Is it visible?
Yeah, it's visible.
Thank you. Thank you very much.
Can you see the slide?
Yeah.
Okay, okay.
Am I clearly audible?
Yes, yes.
Yes, you are audible.
Okay.
Please go on.
Thank you. So, hi. Good afternoon to everyone. I'm Riya Datta from Vidyasagar University, West Bengal. I'm going to present my e-poster entitled Plant Growth Promoting Activity by Endophytic Fusarium Species from Chelus costa speciosus. So Basically, endophytes are hidden microflora of plant that are reside within different plant parts and do not produce any types of harmful symptoms or like disease, and these are becoming a very good area of research as because they have the potentiality to produce different types of bioactive metabolite that can enhance their host defense systems against different types of biotic and biotic stresses. So I have chosen this area for my work. The methodology that I have followed for the work, that is the collection of healthy and disease-free plant from a region of East Singhbhum district, and then the plant was stored in 4 degrees Celsius for the further processing. Then we have isolated different types of fungal endophyte from that plant. Kindly, could you please scroll the slide? Yeah, okay. So the objective of my study was to collection of a medicinal plant species and assume that plant might have some good species of endophyte, fungal endophyte basically, that are able to producing some plant growth promoting activities. So we have divided these. We are followed different types of objective to fulfill my aim, and that is the isolation and identification of plant species, screening of those endophytic fungi for the production of IAA and different other types of plant growth promoting activities, like as I shows the result in this picture, that is there is the petri plate which containing the endophytic fusarium species, and that is the microscopic photography. Then the liquid culture of that isolate. After that we have we have done the IA estimation assay of those fungal endophyte, and out of which one isolate, that is the CHER2 R2, because that isolate was taken from the root part or the rhizome part of that plant, and these isolate basically shows a greater potential to produce the IA in the culture media. After that, for the confirmation of those compound in that liquid culture, we have run this we have run the TLC thin layer. We have followed the TLC thin layer chromatography for the confirmation. And other than that, we have also followed mining ammonia producing assay and phosphate solubilization assay to characterize the proper potentiality of that isolates. So these are the result where we have applied the fungal endophytes and the fungal culture on the directly on the plant. Here I have chosen the common mung bean and wheat plant to the tomato plant to show the result, what that is the positive way or in a negative way. Please zoom in the
Which one? The graph?
There's a result part, sir.
Okay.
The graph part. Yeah. Okay, thank you. So here I'm showing that the treatment where the endophyte is directly applied on the plant, and that is showing the greater result for the enhancement of plant growth, whereas when we have applied the fungal extract on the plant, that not shows that much of a result for the growth. So here we can concluded that that the direct applications of the endophyte of such endophytes by spraying on the soil or the root associated rhizosporic part, it may be a permissible way to develop a new agricultural opportunities, because in the recent time different types of tissue culture technologies, different types of modern technologies that are used to manipulate the plant gene and also the microorganisms gene to survive in certain types of decertified areas to accommodate the agricultural land. So other than that,
30 seconds left.
Yes, sir. So other than that, if we make an interest on the use of such types of endophyte by spraying directly on the soil or on the plant pots, that may be a promisable way for next study. Here also left some work in the for the research findings on future. So that's all, and I would like to thank my gratitude to my supervisor, Professor Debilal Banerjee, and also I would like to my sincere gratitude to Department of Botany, Durgapur Government College, for giving me the chance to sharing my little work in front of all you. Thank you very much.
Thank you so much, Ms. Datta. So the session is open for question.
So Ria, I have few questions.
Yes.
One is, there are many endophytes present in every plants. Okay.
Yes.
Many types of bacteria, many types of fungi are present. So why you have chosen only fusarium? Is there any reason to choose the study of fusarium only?
No, ma'am. I am not choosing mainly fusarium. I have already worked on different types of other genera, but here in this study, this particular isolate, that is fusarium, that's showing the greater result, and I am presenting only this one, not the other. There are other isolates like Coletotrichum, Cladosporium, Alternaria. They also have these types of property on plant growth promotion. But here in my study, I basically chosen this genus Fusarium because this shows the greater result on the plant when I applied it on the plant actually.
Have you seen any kind of bacteria as endophyte in this plant?
No, ma'am. I am not working on actually bacteria. I am actually working on only fungal genera of those endophyte.
Okay.
Thank you.
Any other questions from any participant? Any other questions?
Another thing is, you have given the 2 days national webinar Botany through modern perspective in a bold font, huh? But you have given your title of speech in a very small font. So for the next time, don't do this. This is not your highlighted portion. This should be in highlight, right?
Okay.
Your
sure.
title should be in highlight.
Okay, sure. Thank you.
Thank you so much, Ms. Ria. So we will move to the next participant, Mr. P. Santanapandi. You can please share your slide. And he is from Department of Plant Biology and Plant Biotechnology, Presidency College, Autonomous, University of Madras. And I would like to notify Mr. Siddharth Shankar Ram to get ready for his presentation, as he is the next one. You can start sharing your slide, Mr. P. Santanapandi.
Yes, sir. Just a minute.
Yeah, sure.
Good evening to everyone present here. I am Sandana Pandi, doing PhD research scholar under the guidance of Dr. C.V. Chittibabu. Today's my article is Therapeutic Diversity and Species Richness of Ethnomedicinal Plants in Somagiri Hills, Eastern Ghats. India is one of the largest producers of medicinal herbs and is appropriately called the botanical garden of the world. Traditional system of medicine are endowed with the vast knowledge of tribal and folk medicine, which utilized around 7,500 species of medicinal plants called ethnomedicines. The World Health Organization records that over 80% of the world's population take refuge in the traditional system of medicine for their primary human health care. Due to the unavailability of modern healthcare facilities, poverty, connectivity with the urban center, awarenesses, etc., people in rural areas are still relying on traditional medicine for their healthcare. The study area comprises Somagiri Hills. It's located in 250 meters above mean sea level of Eastern Ghats, South India. It is herbaceous dry evergreen thorn forest. It is inhabited by Valayan tribal community. Madurai district receives an annual rainfall about 600 to 850 millimeter, temperature variation between 18 degree to 40 degrees Celsius. Data collection. The ethnobotanical data were collected according to the methodology suggested by Jain 2001. A structured questionnaires were used to collect data and interview and interaction with the 8 elderly and experienced individuals practicing indigenous medicines helped in the elucidating the data. The medicinal plants used by the practitioners were photographed with GPS data. The voucher specimens of the medicinal plants were collected for the preparation of herbarium and were identified through pertinent flora. Scientific names of a medicinal plant species were also corroborated with the online source like the plantslist.org.in and tropicos.org.in. The information on medicinal plants, local name, life forms, plant parts used, method of preparation, mode of drug administration, reported risk if any, and human illness treated were all recorded from the informants. Results and discussion. The present study has revealed that there are a total of 107 medicinal plants belonging to 47 Angiosperm families and one pteridophyte family. The plants were used to cure 19 different human ailments by the Valayan community people. Fabaceae with plants, 18 species, is found topping the list of ethnomedicines, followed by Euphorbiaceae and Apocynaceae. Among the reported ethnomedicinal plants, Chlorophytum borbelianum and Aegil marmelos gain greater conservation significance as per IUCN Red List. The image one shows life forms of plant used by the Valayan community people of medicinal plants were categorized as herbs 40%, trees 30%, both climbers and shrubs 11% each, and small trees 3%. The different plant parts used in the ethnomedicine followed by whole plant part was 27%, bark 16%, fruit 9%, latex 5%, and root and flowers 2%. Based on the drug type, it is found that juice form of drug is highest 50%, followed by paste form is 29%, decoction is 16%, and edible part is 1%, and ash is 1%. Therapeutic diversity of ethnomedicinal plants were found used to cure dermatological infections is 25%, followed by gastrointestinal diseases 14%, poisonous bites 10%, gastro general debility 9%, skeletal-muscular system disorders 7%. The topmost plants and their ethnomedicinal uses in the study area we are observed. Acaranthus aspera is commonly called as Nayurivi in the local people. 5 to 10 grams of leaves are made into paste with little amount of water. This paste is mixed with a small amount of lime and applied externally to cure dog bites. Adadada vasica. Young leaves are grounded and a half glass of leaf juice is mixed with honey and taken orally as remedy of cough. It is also digestive and good for bronchitis. Aristolochia India. The leaf juice is an antidote to snake bite and scorpion sting. Powder root is given with honey for leukoderma. Cassia auriculata. Flowers are used to cure diabetes. Leaves reduce obesity. Leaf powder made into paste is used as hair shampoo. Aristolochia bracteata. Root used to identify the snake bite. Root extracts added with Acaranthus aspera and Arvelaneta and Aristolochia indica root extract as a polyherbal formulation to cure snake bites. The results confirm that the Valayan tribal depended on heavily on the medicinal plants of the Somagiri Hills, inhibited by them for their healthcare and so stressing the need for revival of interest in ethnomedicine. Documentation of...
Excuse me, we have only 30 seconds left. Please carry on.
Okay. Documentation of ethnobotanical knowledge is a valuable effort as it may carry secret solution for cancer and emerging forms of dreadful diseases like COVID-19 and Omicron. Thank you.
Thank you so much. Now the session is open for question here. Any participant you want to ask any question?
Yes, Dr. Santanon, very nice work, and presentation is also very nice, and you have covered many things in your poster.
Yes,
And
sir.
thank you very much, and congratulations for your nice presentation. One thing that is that the you have surveyed and you have informed a lot of lot of information regarding the traditional medicine sources of from the different plants. The thing is that how did you get the information, that means the applications of the various medicinal plants in the human beings? Say for example, for the various disease purpose that you are mentioning, of the different name of the different plants for curing of the various diseases. Are you mentioning this from the taking the reference from the medicinal plants book, or directly you have consulted the Oja or the
No,
local?
sir, we are collected from the articles, sir. One of my seniors used these terms in article.
Okay, so you have got it from already from the article from published by your seniors.
No, sir, just for the technical terms. The disease categories only were observed from the articles.
Yes. I think, and if you visit some local medicine man or the Ayurvedic man or some local Kobiraj or Gunin like that, the tribal people, many tribal peoples are different places. Actually, at different places they occupy, and if you get some local information, the indigenous knowledge, then I think that it would be enriched more. If you get the direct information from the local medicine man or the Guni like this, and if you incorporate those findings in your work, then it will enrich more, I think.
Yes, sir. This is also we are directly interact with the medicinal practitioners. I saw the fourth image. The medicinal practitioners are collecting the data from the field.
Yes. Okay, very nice. And your methodology is very nice, because I think that you have studied the GIS technique as well as the flora, and that's why it is very good work. You have not followed only the flora. Taking the GIS picture study, if you have taken consideration, and now you are trying to identify the plants on online also, and that's why it is a very good effort from your behalf. So thank you very much, and you try to reach more and more in your findings. Okay. Thank you. Okay.
Thank you.
Okay.
Yes. So any other questions for the participant?
Yes, I just have one question. Santana Pandey, you interviewed some informants. So how many informants did you interview?
We have interviewed more than 20 informants, ma'am.
Okay,
9% are tribal peoples, others are traditional practitioners, local people.
Okay, so your reports are based on their first-hand information about the usage of the plants, isn't it?
Yes, ma'am.
Yes, very good. It's a very nice presentation, a very vast work. Congratulations.
Thank you, ma'am.
Any other questions from the participants? Anyone? Okay, thank you so much. Now we will move to our next presenter, Mr. Siddharth Shankar Ram from AIPH University, Bhubaneswar. And I would like to notify Ms. Snigdha Mukherjee to get prepared with her presentation, as she is the next one. So, Mr. Siddharth Shankar Ram, can you please share your slide? Mr. Ram? Mr. Ram, are you present? I guess he's not present because I can't see his name in the participant list. So in that case, we will move to our next participant, Snigdha Mukherjee, Ms. Snigdha Mukherjee from Department of Conservation Biology, Durgapur Government College.
Yes, sir.
Yes. You can please proceed with your presentation.
My presentation is visible, sir?
It's visible.
Yes.
Okay, sir. Good afternoon to all of you. I'm going to present the poster. My topic is Association of Macrophyte Vegetations and Odonates from Selected Pit Lakes of Raniganj Asansol Belt, West Bengal. We are all know the pit lakes form when open cast pit mining operations are discontinuous and dewatering cases. The pit lakes associated with macrophyte and its chemical hydrology are an interesting appearance. Pit lakes are fully differ physical from other natural lakes, and pit lakes are kept undisturbed. When the growth of macrophyte vegetations and odonates breeding increase, the pit lakes are kept undisturbed. A detailed study on association of odonates with that macrophyte vegetations in West Bengal is still missing, though there are separate work on the both. Methodology I use the random quadrant sampling for macrophyte vegetations and counting the macrophyte vegetations and point count method used for the first moving species. Here odonates is also my study objects, and this is my study site for different pit lakes. MOCP, Mosandhara OCP, Sitalpur OCP, KOCP is Komardi OCP, and NOCP is Navagram SCP. This all of OCP are under the Bankola area. And now the figure 3, PCA, using the PCA biplot showing. The biplot figure 3 also suggests that MOCP has distinct species composition, especially in spring. Thus, it a good hypothesis that during spring macrophyte growth is highest for MOCP, which is turn may provide a good resource for odonates, and they become most abundant at this season. NOCP and KOCP is similar species composition in both spring and winter. SOCP has unique species composition in winter, where two species are mainly dominant, like Bacopa moineri and Limnophylla heterophylla. NOCP has similar species composition uh, in both spring and winter. In term of terrestrial macrophyte and odonates, is no aquatic macrophyte are reported from this OCP. And now we look into this hierarchical cluster. Hierarchical cluster of 4 OCP revealed a closed association of odonate species with both aquatic and terrestrial macrophyte, including good vegetation assemblies in all studied sites. Association analysis of macrophyte and odonates is assessed using the hierarchical cluster following the nearest neighbor method. Here it was OCP observed the cluster graphs. It was found MOCP 7 odonate species like Neurothemis fulvic and Brachydiflex sorvina and Asipma panorboides, and this is also closed primary cluster and are mainly associated with terrestrial macrophyte, especially in the Dalbergia siso. It also evident that Isnura senegalensis and Crocothemis cervilia depend on the aquatic macrophyte Thypha angustrata and also the Sida acuta. And here we look into the KOCP, the macrophyte mainly Limna Hygrophila formoides and Limna minor. A different picture observed in cluster graph of KOCP. Here macrophyte mainly a different pictures of observed in cluster graph KOCP. The macrophyte mainly phlomoides and the lamina minor. Okay, and the here the NOCP aquatic macrophyte are absent for the red algae, and whom problem many due to presence than the aquatic macrophyte. Only dragonfly belongs to orthidum is seen to be associated with the terrestrial macrophyte. And here some pictures. Then I conclude that overall a well documented unexpected association is noted among the macrophyte in all OCP, and though these were unique ecosystem whom prime purpose was coal mining, but yet these pit lakes, after closer and abundant, can serve a better ecosystem for growth, the odonate diversity. MOCP is suitable for macrophyte vegetations and odonate breeding because it is very old, abundant OCP. Overall, a well documented and the presence or absence odonate is any ecosystem act as an indicator of limnological health. A detailed limnology and hydrodynamic study conducted in OCP would help understand the relationship between biotic and abiotic factor associated with the pit lakes. And this is a reference. I use this study. And lastly, I would like to thank my supervisor, Ma'am Dr. Moichi Banerjee, and also my principal sir Devnath Palit, and the author to thankful my Department of Conservation Biology, Durgapur Government College. Thank you.
Thank you so much, Ms. Mukherjee. Nice presentation, and I guess this is the single poster I came through that say about the plant-animal interaction. So the poster is open for questions, please.
Am I audible?
Yes,
Sandeepan sir? Yes,
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 sir.
Yes. You have presented very good bit of work, and it is very the work is praiseworthy, and it is very interesting also, and presentation is also very good. Thank you very much and congratulations. The thing is that you have tried to make a correlation between the interaction of different biotic and abiotic factors also in pit lake study or the macrophytic study. It's very good. So what sort of biotic factors have you considered? What type of the biotic factors? There are so many different types of the biotic factors. What type of biotic factors are you considering?
Sir, actually, here pit lakes, all know pit lakes.
Yes, actually, in pit
Typically
lake.
form the natural.
Yes, to carry on. Yes. What type of biotic factors?
Sir?
What type of biotic factors are you considering? Actually, the biotic factors and abiotic factors, these are again classified in different types. So I am interested to know that what type of biotic factors are you considering? Those are interacting with the macrophyte assemblage.
So here I'll to present the odonates group and the macrophytes relationship.
Yes.
Sorry, sir.
Actually, what type of biotic factors means? I like to know the species interactions or some interspecies interactions or intergeneric interactions. What type of interactions are there? Those are also the biotic factors. But yes, that's why. And not only the assemblage that you are seeing under the pit lakes, that is not always the factor. Some in-depth factors are also there. At the various height or altitude of the lake, the different assemblages are there, and the interactions between the assemblage of different biotic agents occupying in the different strata of the lake. Those are the more in-depth study, I think, and that I think that you will incorporate this thing in future. But in depth, you have to study this thing. One thing. Another thing is that whether is there any correlation of the anthropogenic factors surrounding the lake with the occurrence of the macrophyte assemblage? এখানে যে macrophyte assemblage দেখা যাচ্ছে তার সাথে কি কোনো anthropogenic factor surrounding the lake এদের কি কোনো correlation তুমি পেয়েছো বা সেরকম কিছু স্টাডি করেছো নাকি পরে করবে?
স্যার আমার যে স্টাডিটা ছিল সেখানে আমি এখানে একটা রিলেশন দেখতে চেয়েছি পিটলেক্সের মধ্যে ম্যাক্রোফাইটের সাথে ড্রাগন ফ্লাইয়ের এটাই যে
এই রিলেশনটাকে আরো ইনডেপথ আরো ভিতরে ঢুকতে হবে আর কি, ইন ফিউচার আরো ঢুকবে ভিতরে। বুঝতে পেরেছো? এটা আরো আরো ভিতরে একটু গভীরে ঢুকতে হবে এই জায়গাটা, মানে তোমার কিন্তু রিলেশনটা স্টাডিটা খুব ভালো, অ্যাপ্রোচটা খুব ভালো, কিন্তু অ্যাপ্রোচটা আরো গভীরে যেতে হবে, বুঝতে পেরেছো?
ওকে স্যার, ওকে স্যার।
দেখতে যেতে হবে, খুব ভালো, ক্যারি অন, Snigdha, it is very good and congratulations, carry on.
Okay sir, thank you sir.
So any other question for the participant? Anyone? So we can move to our next participant, Ms. Shomanjana Khatua from Department of Botany, Krishnanagar Government College, Krishnanagar, to present her slides, her poster. And I also like to request Ms. Srijita Ghosh to please get prepared with her poster because she is the next one. So Ms. Shomanjana Khatua, can you please present your poster?
Good afternoon. Is my slide visible?
Yes, absolutely.
Okay. Good afternoon, respected teachers, my colleagues, dear students. During our repeated field survey in the lateritic area of West Bengal, recently we were able to collect 3 morphologically unique specimens from that area. Interaction with the local people let us know that they are consuming these mushrooms through ages as health promoting food. This information inspired our research towards this area. So we collected these specimens, and after detailed study based on taxonomy, DNA barcoding and phylogenetic placement, one specimen which is locally known as Murgi Chatu appeared as a novel species, and we named it as Rusula alato reticula and published in the year 2017. Another specimen which is commonly known as Jhal Chatu appeared as Rusula senesis, although our study for the first time included or reported about the edibility of this specimen. The third specimen, which is locally known as Jampatra, appeared as a novel species, and we named it as Rusula pseudocyanoxantha and published in the previous year. To understand the medicinal prospect, then we isolated various fractions from each of the specimen. For example, we have isolated the ethanol extract. First, we wanted to know the chemical characterization. For that, we have performed spectrophotometry and HPLC. Results showed that all the fractions were mainly enriched with phenolics and flavonoids, and as a result, they showed good antioxidant activity. Besides that, the fractions exhibited strong cytotoxic property against Hep3B liver cancer cell line, where the fractions from R. alato reticulum and R. senesis fraction exhibited better activity. To verify the output, then we performed several assays like colony formation assay, DAPI staining, cell cycle profiling, effect of the fractions on reactive oxygen species production and mitochondrial membrane potential, as well as we also evaluated effect of the fraction on certain apoptosis related gene expression. Overall, our results suggested that the ethanol extracts were able to cause certain stress on the Hep3B cell line. As a result, it induced apoptosis in the Hep3B cell line through mitochondrial intrinsic pathway. Apart from ethanol extract, then we isolated hot water extracted crude polysaccharide fraction from each of the fraction. Again, we wanted to know the chemical characterization. For that, we have performed spectrophotometry, HPTLC, GCMS and other many assays. All the assays exhibited that the fractions were mainly composed of beta glucan, and as a result the fractions exhibited good antioxidant activity. Then we asked ourselves whether the fractions have any effect on the immunity or innate immunity. Then we used murine macrophage raw 264.7 cell line as the model organisms. Our results showed that the polysaccharide fractions were able to induce macrophage proliferation, phagocytic activity, nitric oxide production and reactive oxygen species production in a significant way. Then to understand the mode of action, then we go for RT-PCR, and the results showed that the fractions were able to induce expression of TLR2, TLR4, NF-kappa B, COX2, INOS, TNF-alpha, I-kappa B alpha and IFN gamma, which are pro-inflammatory cytokines. In short, it can be said that the fractions being composed of beta glucan, which were acting as ligand for the TLR2 receptors like TLR2 and TLR4, which after binding to the receptor, it induced the intracellular pathway through and mediated and induced immunity through TLR mediated NF-kappa B pathway. So in sum, our results.
Ms. Khatua, you have 1 minute left.
Okay, thank you. In short, our results not only reported about the medicinal prospect of the 3 unique, morphologically unique mushrooms from the West Bengal, but also recommends for use of these mushrooms for development of pharmaceutics, cosmeceutics, and functional food. And lastly, I would like to say that there are many mushrooms which are traditionally popular, but which are available in naturally growing in West Bengal, but world is totally unknown to world. So more and more research should be done on this area so that their power or their medicinal prospect can be used for the betterment of humankind. Thank you very much.
Thank you so much. And the session is open for question. Anyone please can ask question to Ms. Khatua.
It is very nice presentation.
Thank you.
Am I audible?
Yes, sir.
Yes. Yes, madam. It is very nice work and very nice presentation. Pertinent to this seminar.
Yes.
Actually, you have covered from the thallophytic structure of fungus and up to the molecular level work.
Yes,
and
sir.
that's why you have covered so many things in your presentation.
Yes,
and the recent methodology, the different techniques, HPLC, etc., everything you have done.
Yes, sir.
I know you that you are a very good researcher, and you are probably in Krishnagar Government College. My past college, I was for Krishnagar Government College.
Oh, okay, sir.
Yes, I know you, and you have a very good publication. I know. The thing is that one thing, just I have the queries,
yes,
that whether these fungus have any bad health impact upon the human being.
Sir, actually, as these are traditionally popular and tribal people are consuming these mushrooms from time immemorial, so these are good for health.
Yes.
Good as a food. And
no bad health, no bad impact.
No, sir. No, sir. I have not included one data, that is the nutritional composition. We have performed the nutritional composition of Russula alatoreticola and Russula pseudocyanozantha. So the results showed that they are good as nutritionally also. They are good, rich in dietary fiber and good. They contain good amount of essential fatty acids. I have not included them, but they are also good as nutritionally as well as therapeutically. So
Yes,
I have not found any bad way.
Very interesting work. You carry on, madam. You now carry on, and you are doing the very good work, and you have so many very good publication. I know from Krishnagar Government College. You are doing very good work, and continue your work. Thank you so much. Congratulations.
Thank you, sir. Thank you.
Any other question for the presenter? Okay, so I have a little question.
Oh, Shodhivar, please.
So, what kind of a compound you isolated from the HPLC?
Oh,
my favorite part.
Yes. And Shondiban, because of Shondiban, I was able to complete the HPLC part. No, really thankful to you. Till now, I tell everybody that because of Shondiban, I was able to complete my PhD. Without his help.
No, that's all. I'm just
you are
a little part.
angel for us.
I'm just a little part.
No, by HPLC we were able to found some mycochemicals like cinnamic acid and para-hydroxybenzoic acid and gallic acid, which are
and secondary metabolites.
Some secondary metabolites.
And in GCMS also, I guess you have done both the fraction, like soluble and insoluble fraction both.
Now, by GCMS, we have done the carbohydrate part.
Only
carbohydrate by GCMS, and HPLC for secondary metabolites. Yes.
Okay. Thank you.
Thank you.
Good work.
Thank you.
I know you from 2009.
Yes.
Okay, fine.
Thank you.
Any other question?
I just have a small question regarding the phytochemistry. May I please?
Please, madam.
Yes, the phytochemical analysis that you had done the GCMS only for the carbohydrate fraction.
Yes, madam.
Okay, okay. And so that means the total amount of secondary metabolites, what were the kinds of other secondary metabolites that were detected, madam?
Other secondary metabolites which are available, can be available, like terpenoids, steroids, but these were not means if we want to identify them by HPLC, then we have to first run the standards, and the standards that they have to be in pure compounds. They are so costly, and for that Sandipon helped me. He actually their lab gave us so many standards, but the standards for alkaloids and steroids and terpenoids, these are very much costly and were not available to us. And recently I found that there are certain spectrophotometric assays, so through which we can identify a total amount of terpenoid, total amount of steroids, or there are some qualitative tests also, that whether the steroids are available, present or not, but to identify them, then I didn't do that.
So what was the target compound, madam? Any structural analysis done?
Yes, madam. We have mainly focused on the carbohydrate part, because as it showed the immune boosting effect, and due to the COVID situation, we know how much immune boosting, how much it is important. So we focused on the carbohydrate part. So we are working on the isolation of the pure compound and validation of the bioactivity on either on the raw murine macrophage cell line, or we are planning to go for in vivo system. Like mice is not possible for us, but we can go for some zebrafish model or something like that, fish model. So we are
so the exact amount of your target compound that has to be administered, is there yet to be found out, or you have found that?
No, still working on it. We have just isolated, we are working on the isolation of the pure compound, that is beta glucan, but to understand the total structure, like the beta glucan, what is the glycosidic bondage and what is the total structure, so that it can be artificially synthesized, we are still working on it. But because it is mainly on the chemistry background is very much needed for that.
Yes. And that is what makes it interesting, when you start from the phytochemistry and end up with that. I know it's a laborious process, but it's very interesting. Super, madam. Thank you,
madam.
Yeah, I want to add on actually to Dr. Kritika.
Yes,
if you
sir.
want to isolate, if you want to identify the unknown compounds in LCMS, it will be your best thing actually, LCMS or LCMSMS. If you don't have the standard,
yes,
right? And ultimately, it's all about chemistry. Without chemistry, I don't think so. Nowadays, botany is possible.
Yes, sir. Very correct. Even that would have been my next question, because even for my own compound, I did the LCMS only. Then I could actually found out. So it's like you step into the phytochemistry, it keeps on going till you achieve your reverence.
It keeps on going. You isolate a single compound, you crystallize it, you do NMR. It's all about chemistry at the end of the day.
Correct. Only then you arrive at least one or two compounds, and but for the paper they expect more novel compounds to be.
It's quite hard to isolate this single compound.
Exactly, sir. Very correct. Nice, sir. Nice. Thank you.
Thank you. So we'll move to our next participant. That is Ms. Srijita Ghosh from PG Department of Botany, Scottish Church College. And I would like to request Ms. Sudeshna Nonde to please be prepared with your poster, as you are the next one. So Ms. Srijita Ghosh, can you please share your presentation?
Good evening. I hope I'm audible.
Yeah, you are absolutely audible, and your slide is also visible.
Okay.
Audible, madam. Yes.
Okay. Thank you. Good evening. I am Srijita Ghosh from the Department of Botany, and as you can see from the presentation itself, the topic, it is basically just one second. The comparative study of cadmium and sodium chloride stress in growth, development, and basically the photosynthetic parameters, which were carried out on the 10-day mung bean seedling and their possible amendments by sodium silicate. So the main objective of this present investigation was to consider the effectiveness of exogenous silicate supplementation in reviving the toxic concentrations of sodium chloride as well as cadmium chloride applied separately
on the basic biochemical growth parameters, along with that mainly focusing on the pigment content, hill activity, photosynthetic parameters such as stomatal conductance, transpiratory rate, internal CO2 concentrations, etc. Now, in this particular dissertation topic, this is actually a dissertation topic done by the two students from the department for their MSc partial fulfillment. So, movement was chosen for the material, and it was collected from State Agriculture Research Institute, and then the normal procedure of surface sterilization was done, and they were treated for 10 days in the respective concentrations of sodium chloride and cadmium chloride with or without sodium silicate in low dose, that is 2 millimolar dose, and then the further biochemical estimations were done. So this is actually a picture of the particular plants when they were subjected to stress. Their main lengths were chosen and estimated, and then basically it was observed that cadmium chloride, which we know is one of the major environmental contaminants, its concentration in low dose, that is 0.5 and 1 millimolar were chosen, and sodium chloride 50 and 100 millimolar were chosen for the test subjects, and number of photosynthetic parameters was considered. For example, total chlorophyll content by Anand method, then the carotenes and the xanthophylls were method, and the hill activity was method by was followed by the DCPIP reaction. They were all studied, and basically the just one second. Yes. The standardized protocols were followed for the photosynthetic parameters as well. That is the LICOR, that is infrared gas analyzer, was used for the studies of hill activity, the net photosynthetic rate, transpiratory rate, internal CO2 concentrations, and finally the stomatal conductance. And mainly to standardize and support our data, we went for some antioxidant enzymatic activities and stress markers. And finally, in conclusion, it was seen that both the parameters, that is cadmium as well as sodium chloride, they were severely affecting the growth in the test seedlings, 10-day seedlings, and the roots were much more affected in comparison to the shoots. Cadmium chloride, in fact, was much more toxic in comparison to sodium chloride in all these photosynthetic, in all the test parameters which were considered. Sodium silicate, on the other hand, silicon, in fact, is a tetravalent metalloid belonging to group 4A of the periodic table. It's one of the most abundant metalloids. It has been recognized as a toxicity elevating agent as it retains heavy metals in roots and inhibits further translocation to shoots. So silicon-induced reduction to heavy metal toxicity as well as salts toxicity, this has already been established from beforehand. In our result also we found that this that sodium silicate in ample amounts, we used 2 millimolar doses. It was able to overcome the stress effects of both cadmium chloride stress as well as sodium chloride stress. And finally, the basic aim of the protocol was to study the effectiveness
Ms. Ghosh, you have only 1 minute left with you.
Exogenous silicate supplementation in reviving the toxicities that was achieved. So this is the initial study. This is the first year study that was being done by the students. We are now extending our work to lab. From the lab, we have gone to the soil culture and we are testing the same parameters. So this process is still under the process. I think I've completed. So thank you.
Thank you, Ms. Ghosh. Now the session is open for questionnaire. Any participant want to put any question forward? So any question from Ms. Ghosh? Anyone?
Okay, I just want to ask one or two things.
Yeah, sure.
Firstly, I mean, what was the effect of silica treatment, silicate salt treatment on control plants? Was it estimated?
Actually, the plants were subjected, firstly, as you can see from the data itself, some of the plants were just treated with sodium silicate, and in fact we were able to find by infrared gas analyzers that the net photosynthetic rate in some of the tested parameters, some of the 10-day-old plants, it was in fact even more than the control plant. So silicon benefits that.
Benefits that, right. And the replicated values and the statistical analysis, what they done?
Actually, this is for part of the MSc dissertation project. So they were able to go for, they used 3 replicates, but only one time the experimentation could be done because of the time shortage. It's only a 3-4 month time where we could do the practical. This year we are repeating it. In fact, this year we are repeating it in soil. So we are trying to get the same results there.
Okay, thank you.
Any other question from any participant? Okay, then I will move to our next participant, Ms. Suresh Nandi from Molecular and Applied Mycology and Plant Pathology Laboratory, Department of Botany, University of Calcutta. And the next one is Sonushri Agarwal. I will like her to notify that she is the next one. Please prepare yourself. So Suresh Nandi, can you please share your slide?
Hello, is my poster is visible?
Yeah, it's absolutely visible and you are clearly audible.
Okay, thank you. Good evening, everybody. Today I'm going to present a part of my research work entitled A Natural Derivative from Ethnomedicinal Mushroom, which potentiates apoptosis and attenuates cell migration in human lung adenocarcinoma cells. The work was done under the supervision of Professor Krishnendu Acharya. We know that the deadliest disease, cancer, is a very ominous disease and it spreads. It is the main concern for all the human beings. So in this scenario, medicinal mushrooms are emerging as an alternative substitute for chemotherapy, and their exploration will unlock many more modern routes in mycomedicine. In our study, we have chosen Astreus hygrometricus, well known as a false earth star and locally known as Kurkura chatu. This is an ectomycorrhizal mushroom found in the forest floor of Shorea robusta and abundantly found in the lateritic regions of West Bengal. Interestingly, this mushroom is very much available in the different locality of Durgapur area. So I would like to tell to the juniors that you just go and grab this power packed mushroom and make the most use out of it, as the tribal people consume this mushroom as a delicacy because of its rich taste, texture and flavor. Apart from this, it has some ethno-medicinal value, like it has tribal people believe that consuming this mushroom will cure several age-related disorder, and it is also used for curing for healing the wounds as well as burns. Previously, our laboratory has already worked with different extracts of Astreus hygrometricus, which shows immense biological activity. This encouraged us to isolate a pure compound named astracarcurol from Astreus hygrometricus. In this study, we have mainly interested to investigate whether astracarcurol, this is the structure of the compound, whether it possesses any anti-cancer property or not. So for this, we have chosen the lung cancer cell, as lung cancer, the mortality from the lung cancer is quite high in both male as well as in females. So to consider any drug, any compound as a potential drug, it is very important to standardize the doses. So in figure A, where here we can see that with the help of WST1 assay, we try to standardize the dose for astracarcurol. Here we can see with increasing the concentration of astracarcurol, it increases the cell death of the lung cancer cells in a dose-dependent manner. On the other hand, when the same compound was given on the non-cancerous PBMC cells, it imparts almost negligible cytotoxicity, which confers that astracarcurol acts selectively against the cancerous cell. The data was further validated by the cologenic assay, where we can see that astracarcurol treatment has prevented the ability of the cancer cell to form colonies. So to confirm whether the cell death caused by astracarcurol was due to apoptosis and not by any other mode of cell death, the cell were stained with DAPI staining. In figure D, we can see that in control set, the nuclear morphology of the cell were intact with homogeneous blue staining, whereas in the treated cell, the nucleus of the cells were comparatively smaller in size. They are brightly stained, they were fragmented, there are chromatin condensation as well as membrane blebbing, which are the major hallmarks for apoptosis. This was further validated by the scanning electron microscope photographs, which shows that in control set the cells were intact with their original morphology, but on treating the cells with astracarcurol, it has disrupted the cell morphology. It causes the cell shrinkage. Further, in figure F, we can see when the astracarcurol treated cells were doubly stained with acrine orange and ethidium bromide, we observe that there is a green fluorescence in control cells, which implies that they are the living and healthy cells. Whereas in the treated set, we can see that fluorescence has shifted from green to the yellowish green to yellowish orange fluorescence, which implies the early as well as late apoptotic phase. This data was further validated by the Annexin 5 FITC PI staining method, which was presented in figure G, which shows that maximum cell population has accumulated in the second and third quadrant in the treated cell, which signifies the early and late apoptotic phase. Interestingly, our compound was also successfully freezes the cell cycle progression of the lung cancer cell at G0 G1 phase, which is very evident from the presence of the sub G0 peak in the treated set, which is absent in the control cell. So the presence of this sub G0 peak is a clear indication of apoptosis. So once we are confirmed that astracarcurol induced cell death in lung adenocarcinoma cell was due to apoptosis, we are curious to investigate
1 minute
the
left.
underlying mode of molecular mechanism. So for this, both the markers of both extrinsic as well as intrinsic pathways were observed. In the Western blot here clearly shows there is an upregulation of all the major markers of extrinsic as well as intrinsic pathway with the downregulation of anti-apoptotic marker BCL2. This causes the disruption of mitochondrial membrane potential as evident from this figure B. Further, it causes the release of cytochrome C from mitochondria to cytosol, which is evident from this immunostaining method, and this causes the cleavage of caspase 9, caspase 3 and PARP and ultimately leads to the cell death. Apart from exhibiting a potent apoptotic activity, astracarcurol was very evidently shown the potent anti-migratory property. So here we can see that we have created a wound. Then after 24 hours of incubation with astracarcurol, we can see that in control, the wound or the gap has been completely filled, whereas in the treated set, they have astracarcurol prevented the migration of the cancerous cell, and in the highest dose, the gap has rather shortening of the gap has widened up. This was further validated by the transwell migration assay, where we can see in contrary to the control cell, lesser number of cells have migrated to the lower part of the transwell, and the scanning electron microscope also shows that in control cell there are large number of protrusions, which is very much essential for migration, but in treatment there are lesser number of protrusion with smooth cellular surfaces. We try to translate our in vitro work to the ex ovo xenograft mice model.
Ms. Nandi,
Yes, I'm just about to finish. Give me 1 minute. We have taken the cheek embryos, and they were inoculated with lung cancer cells, and after 3 to 4 days the tumor were developed. We treated them with astracarcurol. Then on 14th day of our experiment, it is very visible from this figure that astracarcurol treatment has significantly regressed the size of the tumor as compared to the control. Apart from this, it has also prevented the formation of new tumors in the distal organs of the cheek embryo. So in a nutshell, we can say that how my work has glorified the astracarcurol as a very potential anti-cancer drug, which might raise better hopes for the cancer bearer in the coming future. Okay, thank you very much.
Thank you so much, Ms. Nandi. Now the session is open for questioning.
Okay, Shudeshna, very nice presentation and very good work.
Thank you, ma'am.
So have you published this work in any journal?
Yes, this was already published in Environmental Toxicology, a Wiley publication.
Okay, very good work.
It is nice work. So really, it is very nice work, madam.
Thank you, sir.
Yes, it is very nice work. But one thing just I'd like to know,
yes.
How many, what are the total number of the types of bioactive compounds in this particular species? That means the Astreus. How many bioactive compounds are there?
We have isolated through column chromatography. We have isolated 2 compounds from Astreus hygrometricus. That one I'm working with, that is astracarcurol, and the other is astracarcurone. These 2 compounds have been isolated.
Is there any impact of any of the bioactive compounds upon the activity of the natural killer cells? That means it
No,
is.
I have not checked. I have just investigated their activity
Actually,
on the cancer cells only.
you have studied it very well in
Hmm.
considering the different health aspects.
Hmm.
But when we consider the different health aspects, then of course from the immunological system, that is from immunological point of view, we should also know the activity of the natural killer cells, because for the tumor productions or anything else, the NK cells have very good impact, and that's why any of the any compounds of from this.
Actually, as we have checked it on the against we checked the anti-cancer property of this cells, so we are more concentrated in this area only.
Yes.
So we have not checked whether it have any immunomodulatory activity or not.
Yes. Okay, very good work. So I think that all the medical aspects, or that is the medical consideration, from the medical consideration of point of the view, different parameters also should also be investigated in future. I
Yeah,
think it is.
definitely, definitely. This is just a part of my.
Yes. Those who are doing work more, the expectation becomes more. That's why. Okay. And
Okay.
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 Thank
Thank
you.
you, sir.
I have a
Thank.
small query.
Yeah, sure.
Dr. Reshna. Actually, I guess initially you got lots of bioactive compounds, right, from this mushroom?
We just got two secondary
Initially.
metabolites from this. No, in HPLC, we have not done any HPLC for Astreus hygrometricus.
Okay,
It
just you
shows
have done
we
column
worked with the different extracts of this mushroom. It shows good biological activity. So we went for isolating pure compound from this mushroom through column chromatography.
Oh, in column chromatography only you got only these two compounds,
Two
right?
compounds, yes, we got only
Yes.
these two compounds.
Okay. Well, actually, I want to ask how would you pinpoint to this particular compound? Okay, I got my answer.
Okay, yes.
Okay, thank you. Thanks.
Thank you.
So, any other questions for Sudeshna? So, I guess we can move to our last participant for today's presentation, poster presentation session. That is Tanushree Agarwal from Miss Tanushree Agarwal from Center of Advanced Study, Department of Botany, University of Calcutta. Miss Tanushree Agarwal, you can please share your slide. Ms. Agarwal?
Yeah, show me what I'm doing.
Yes. Okay.
Is my screen visible?
Yes, absolutely visible, and your voice is also clear.
Okay.
It is visible. No problem.
Okay. Thank you, sir. Good evening, everybody. My topic of discussion, sorry, I'm Tanushree Agarwal from Department of Botany, and this is here where I've been working for the last 10 years. My topic of discussion today is moss, little things which can fill the huge glitch in agriculture. And in lieu with this, I would like to start by saying that Pandit Jawaharlal Nehru had said, everything else can wait, but not agriculture. So it's a known fact that India is a largely agricultural country with a wide variety of crops grown not only for domestic use but also for export. It's also a fact that farming in India is mostly unorganized and it's dependent on natural weather cycles rather than the use of modern technologies. So farmers, they depend largely on rain gods and unpredictable weather cycles, which often causes large-scale crop failures due to drought, leading to bouts of famine. So 54% of India faces high to extremely high water stress. Adding to this, there are other stress factors such as pests and crop diseases, which put tremendous pressure on farmers and their families. So in the recent past, this has also led to extreme measures among farmers, including suicides, and the situation does not seem to be improved. So India is the second largest producer of cereal grains in the world, and it's roughly hypothesized that consumption will increase to 377 million tons by 2050. So this will create a huge gap in demand and supply, and it will affect the economy of the country as well, with an added speculation of a fall in agricultural production due to drought and pests. So there's a dire need to identify new traits for developing resistant varieties and introduction of these for commercialization. Right now, according to the data of DST Government of India, we have 126 research institutes working in the field of biological and agricultural sciences, with numerous laboratories continuously identifying genes which confer tolerance traits. Here in our laboratory at the University of Calcutta, we have been involved with the identification and analysis of dehydrin group of genes, which are drought responsive proteins for its potential in drought tolerance. We chose Piscometrilla patens and moss for our molecular analysis, as it would mark the evolutionary pattern from lower to higher group of organisms for the protein group under study. So Piscometrilla is a well-known dehydration tolerant plant, and its analysis has been restricted to only a small group of researchers in crop improvement. Now, dehydrins have been widely analyzed since its first report. However, its precise mechanism of action is still unknown. With the research in our lab, we have been able to stimulate the conditions of growth for Piscometrilla, and we are now growing it in BCD media, in solid media, in liquid media, and it has been established in the lab properly. Now, from there, we have tried to identify 3 dehydrin genes, A, B, and C, and these dehydrin genes have been cloned, sequenced, and the proteins have been isolated, overexpressed in bacteria. And these proteins have been used for in vitro enzyme protection activity. There, we have found that dehydrin A shows efficient protection of lactate dehydrogenase under desiccation stress and freeze-thaw cycles as compared to dehydrin B and C. So a comparative analysis has been drawn between these 3 dehydrins, where dehydrin A has been found to function very effectively under drought conditions. On the other hand, dehydrin C was found to have antibacterial properties, which was established by MIC IC50 and SEM analysis, where we found that under normal conditions or in normal bacterial cells, in comparison to that, the cells which were treated with dehydrin C, they showed membrane disruption with large lesions and exudation of cellular material. Also, our NPN assays showed that there was membrane damage, as the fluorescence was observed to increase when the cells were incubated with dehydrin C. This was observed for the bacteria Bacillus subtilis. Now, for the comparative analysis of dehydrin A, B, and C, all these dehydrins were cloned in Dcambia 1301 binary vector, and they were mobilized into agrobacterium and transformed into tobacco, and the transgenic lines were raised. And they were put under different types of stress conditions, like sorbitol stress, heat stress, cold stress. Here only the graph of sorbitol stress has been shown. There dehydrin A was again found to have the highest effectivity, followed by dehydrin B and then dehydrin C. Several biochemical assays were also performed.
Miss Agarwal, you have a minute left with you.
Yes. So several biochemical assays were also performed for establishing this, like chlorophyll content, soluble sugar, proline, MDA, and relative electrolyte leakage was also measured. Now, in order to study why dehydrin A only is showing higher effectivity, functional dissection of this protein was carried out, where we identified that that this protein has multiple copies of a particular amphipathic alpha helical segment. Now, dehydrins have a characteristic feature that they have actually 3 conserved segments, which are known as Y, S, and K. However, here we have identified the another segment that is marked in green, D segment, which is actually present in multiple copies in the protein, and it has a conserved sequence. So whether this sequence has any role in its protective activity, for that we developed different types of deletion mutants, and we found that as the number of these amphipathic alpha helices increases in the protein, its protective activity also increases. So actually, dehydrins have a particular K segment, which also can form an amphipathic alpha helical structure, and which is mostly important for dehydrins to function. So here we have identified a novel amphipathic alpha helical segment, which functions like the K segment. And as the number of copies of these segments increases in the protein, its protective activity also increases. So this was again established with the enzyme protection activity. And further assays were done with this. The transgenic tobacco plants were also generated with all these mutants. I have not been able to show this in the poster. And further work is being carried out to actually establish these segments to actually make a tire of these segments into a particular protein, and we can use that protein as a supramolecule, transform it into rice plants. That is the work which is going on. And if we can generate drought tolerant rice plants, or we can take it into crop plants. So this work, a part of this work has been published in Planta and JBC, and that's all. Thank you.
Thank you so much, Ms. Agarwal. Now the session is open for questionnaire. So if anybody wants to ask,
Hello.
Yeah.
sure.
Yeah, I have a question.
Yes.
Yeah, Ms. Agarwal, you have submitted that in abstract that it was Piscometrium.
Piscometrium.
Yeah. So
The name has been changed to Piscometrium lately.
so these two are synonyms, right?
Same, yes, same
Okay,
species. The
okay,
name
okay.
has been changed to Piscometrium lately.
Okay.
So any other question for the participant?
I just want to ask one thing. You studied the conserved sequence of the studied the conserved sequence of the dehydrogen, right?
Yes.
Did you what was the bioinformatic approach that you underwent? The first figure it is shown over here. Did you underwent for meme study? And
No, actually, here we were identifying whether these segments are actually present in a repeat pattern. They have
right.
similar themes, very same sequence. Like if you can see over here, it's D1 has QEGI, MD, KVK, NAV. D2 again has QEGL, VDK, AK, DAB. So the sequence is very much similar, and there are about 11 sequences like this. So we isolated those sequences, and then we were trying to identify that which are the amino acids which are changing from D1 to D11, and what is the whether they belong to the same group or whether they are belonging to the different groups.
okay. You did it from your
Yeah,
experiment,
yeah.
right. Okay, thank you. Nice piece of work.
Thank you.
Any other question? So thank you, Ms. Agarwal.
Thank you so much.
I'm again calling the name of Mr. Siddharth Shankar Ram. Is he here with us? Mr. Siddharth Shankar Ram, are you here? Okay, I guess not. So we'll conclude this poster session 1 for today. And from my side, this is a wonderful session, and we have witnessed some path-breaking work today that will certainly going to change the research scenario, research thinking, and altogether the research path of what we are doing, so-called science, for our whole country. To all the dear participants, please keep up this good work. And now with that, I would like to request our honorable convener and head of the department, Dr. Ashok Bhattacharya, to please summarize the outcome for the day 1 of the webinar.
Okay, so I think that nobody will be there when I will finish the summarization, because
No,
already the
we are
participants are trying to leave. So actually, thank you very much to all of you and all the participants who have kept their patience since 10 o'clock today, and they have give their full support to us for convening and organizing these whole day hectic schedules in a very smooth way. So in today's national webinar, today is the first day for all of us, and the first day it was started by the inaugural session, and just as 10 o'clock we have started our session, and now it is 5:40, and we had the expectation to finish it by 5:30, but I think that we are quite disciplined because only 10 minutes we are late, and that's why we are going to finish it in proper time. 10 minutes is not any nothing else. Statistically, it is any significant delay. So today the lecture was started at 10 a.m. Through the welcome address given by our respected principal, Dr. Devnath Palit. Then the inaugural speech, it was given by the IQAC coordinator, Dr. Abhijit Mondol, and our Teachers Council Secretary, Dr. Swapun Kumar Ghosh. He also gave a short speech as the greetings of the today's event. And then the keynote speech started, and our teacher, respected teacher, Professor Sudhendu Mondol, elaborated his views in different aspects, starting from the medicinal plants on the conservation, as well as the different historical aspects of the plant systematics, classification, everything, covering the all aspects of the angiospermic groups, and not only the angiosperms, he also mentioned some name of the some pteridophyte fern also. So considering all the aspects, the ethnomedicine, sociomedicine, sociobiology, and botany, plant science, environmental, everything he covered, tried to cover, and it was ended in proper time, the keynote speech. Then the plenary session started exactly at 11:30, and our first plenary speaker was the Dr. Jogesh Misro. Jogesh Misro, first one who delivered his lecture on the particular topic, how does the Anabina species PCC 7120 deal with the acute cadmium toxicity at molecular scales and get recovered out of it during post-stress recovery. It was very subject specific speech at the molecular level, and it was very interesting, although the topic is the from the algae to the angiosperms, but it covers a particular in-depth analysis of a particular species of the Anabina, and at the molecular level, the study was at the molecular level, and he particularly molecular genetics as well as the cadmium toxicity, he correlated this thing in his study, Professor Jogesh Misro from Banaras University. Then we got our respected sir, Professor Krishnendu Acharya from University of Calcutta. He gave an elaborated lecture on a biotechnology based organic farming system for empowerment of the farmers. Actually, Professor Acharya, he encouraged us not only for the academic point of view for the study of the plant science with reference to the mycology and plant pathology. He stressed on and he emphasized on the fact that the job opportunity and the empowerment is nowadays the one of the problem, and that's why he tried to encourage all the students, researchers, and the teachers also to make something so that the job opportunity could be enhanced. And that's why from the lab to land, how the practice could be established. That's why his main target by Professor Krishnendu Acharya. Then we started accordingly the competition, that means the abstract presentation, and the abstract presentation, it was started, and in this session, in the post-lunch session, the total number of the abstracts which was presented 19 total number of the abstract is was presented 19, but one was absent. In presence of our respected judges, you see that the respected judges are also here, and they are also giving their full support to evaluate and the judge. Now we will not mention their names, but among these participants, the judges are there, and with the help and cooperation obtained from our respective judges, the participation presentation started accordingly, which started at the subject, that means the ultrastructural, physiological, and the thylakoid proteomic analysis of the Anabina species started here. Then the micromorphological studies of some mangrove eco-friendly method of the strawberry cultivation at the rooftop garden. It was very nice presentations. Every presentation was very nice. Ethnomedicinal knowledge of the uses of the plants with reference to the tolerance, air pollution tolerance level, phytosociological attributes, and the phytomedicines, then the present status of the vegetable crops, the ship-cellar features and their taxonomic significance, then the survey of the macrophytes, and the variations in the vascular epiphytic assemblage, then the plant growth promoting activity of the endophytic fusarium species, therapeutic diversity, and so far I remember that title, that is the trees along the roadside, association of the macrophytes, and a hope from the newly discovered edible mushrooms, comparative study of the cadmium and the sodium chloride, stress physiology and the antioxidant activity, natural derivative from the ethnomedicinal and the mushroom potentials. It was a very good work. Every work was very good. And the mosses, and the last one is the moss, the little things which can fill the huge glitch in the agriculture. So these 18 different abstracts, these were presented in today's technical session, and all the abstracts embodied a good bit of work. It informed a lot of us to enrich our existing knowledge and to increase our unknown knowledge. Okay, and that's why all of the participants from this platform would be very much appreciate. They would very much appreciate, and they would be very much benefited from the knowledge sharing from the participants from today's lecture. And everyone is satisfied, I think, and on behalf of the organizing committee, I wish to extend my thanks and congratulations from all the organizing committee, as well as on behalf of the Department of Botany, Durgapur Government College, and we are very grateful to all our respected judges present over here in today's evaluation process. So thank you very much, and thanks all.
Thank you so much, sir.